I think that everybody is here, so we can start our symposium now. But before starting, I will give some wishes of our president and our rector, and then I will give the speech to our dean of the Faculty of Medicine, our international relations director, and then we will continue. So first of all, dear professors, distinguished guests, and esteemed colleagues, welcome to the Handshaking of Fundamental Sciences and Clinical Sciences Beyond Boundaries Symposium. I am delighted to have all of you here, whether you are joining us virtually from near or far. This event is a unique collaboration between İSTANBUL AYDIN University, Faculty of Medicine, the Health Policies Resource Center, and the Anatolian Education and Culture Foundation, and I couldn't be more exciting to open this remarkable day of knowledge sharing and innovation. So our first speaker was to be our president, Professor Dr. Mustafa Aydin, but as he is abroad, I'm going to give a short brief on behalf of him. So on behalf of him, the entire organizing committee, he said that it's my distinct honor and privilege to welcome you to the International Symposium, Handshaking of Fundamental and Clinical Sciences Beyond Boundaries. Today marks a significant moment as we gather here to explore the intersection between the fundamental sciences and clinical sciences transcending boundaries in pursuit of greater knowledge, collaboration, and innovation. This symposium serves as a platform for intellectual exchange where experts from various disciplines converge to share insight, discuss breakthroughs, and forge new connections that will shape the future of science and healthcare. I extend my heartfelt gratitude to Associate Professor Dr. Mustafa Aydin, Chairman of the Board of Trustees, İSTANBUL AYDIN University, for gracing us with his precious wishes and delivering the opening speech and wishes. His leadership and vision have been instrumental in bringing this symposium to fruition. Furthermore, his dedication to advancing education and research at İSTANBUL AYDIN University is truly commendable, and we are grateful for his leadership. And another opening speech was going to be Professor Dr. İbrahim Hakkı Aydın, but he is not here in the campus, so he said his wishes and the pleasure that we come together today. So under his guidance, İSTANBUL AYDIN University has emerged as a beacon of knowledge and a hub of groundbreaking research in various fields. His unwavering dedication to advancing education and research has made a lasting impact on the academic community and beyond. So another one that we already, in fact, organized this symposium in collaboration with the Anatolian Education and Culture Foundation on behalf of Elif Aydin, Vice Chairman of Board of Direction of Anatolian Education and Culture Foundation. She extends her sincere appreciation to all of invited speakers and participants for their participation and contribution to the symposium. So now, Professor Dr. Zafer Aslan is here with us. She is our International Academic Relations Coordinator of İSTANBUL AYDIN University, and we work with her for a long time. So now it's my privilege to introduce a distinguished leader and academic, Professor Dr. Zafer Aslan, Vice Rector of İSTANBUL AYDIN University. With a wealth of expertise and passion for advancing education and research, Professor Aslan brings invaluable insight to our symposium today. So welcome, Professor. Maybe you can open the microphone.
Now it is okay?
Yes, it's okay.
Yeah. Thank you very much, Professor Armani. It's really a great honor for me to join with you at the initial stage of the important international symposium. Dear presidents, rectors, dear deans, dear distinguished colleagues and students, I would like to say welcome to the important international symposium on handshaking of clinical sciences and medical sciences and in between the boundaries. So today it is very well known that healthcare marketing, by offering many opportunities especially in hardwaring, softwaring activities, services, technologies, in engineering applications like machine learning, advanced data mining, and learning natural language and processing, even simulations and their applications in medical image processing and diagnostic analysis to declare about the potential of the risks of the problems for patients. And this analysis and their importance have a great growing ratio. And it is expected that this marketing value is estimated to reach up to the $148.1 billion until the 2029. So really it is great numbers. And then this is why we are together with you here. And the process, of course, needs to reduce the healthcare costs, improving computing power and declining the hardware case costs. So how we can do it? The rising number of the partnerships and collaborations, like here in international meaning, with the cooperations of more than 10 international countries. We would like to very much oblige to all of you to join with us here. It tries to imbalance in between healthcare work and patients analysis. Even in our university, at the graduate institutions, we try to have some advanced applications regarding with the advanced image processing and early detections of the problems and right classifications to have the surgery field in hospitals. And even risk analysis, they will help very much to the medical sciences by using advanced machine learning techniques. To reach the reliable and comparable data, this is another important issue. So even early detection and reliable classification of the risks are very critical issues in that field. I'm sure that this important meeting will be very much helpful and have a great contribution in this field. And it will increase our close national and international cooperations, partnerships. So these kinds of the close cooperations and partnerships, it will remove all the borders in between institutions. I'm very happy with this feeling. I would like to thank to all chairs, co-chairs, organizing committee members, and the faculty members, deans, and all colleagues, and also especially invited speakers and participants, and for students, for your valuable contributions and for your questions to discuss these important topics in this meeting during the whole day. I wish a very great, successful, and very useful sessions, enjoyable sessions to all of you. Thank you very much. With my best wish, regards. Thank you.
Thank you very much, Professor Aslan, for your valuable speech and time being with us. It's our honor to have you here. And now I would like to introduce the Faculty of Medicine at İstanbul AYDIN University. Professor Enver's leadership and expertise in the field promise an enlightening and enriching opening speech. Yes, the speech is yours. Evet hocam, söz sizde.
Teşekkür ederim.
Hoş geldiniz tekrar.
Başlıyorum. Kıymetli hocalarım, değerli konuklar, değerli meslektaşlarım ve katılımcılar. İstanbul AYDIN Üniversitesi Tıp Fakültesi, Sağlık Politikaları Uygulama Araştırma Merkezi ve Anadolu Eğitim ve Kültür Vakfı AKEV işbirliğiyle düzenlenen Temel Bilimler ve Klinik Bilimlerin İşbirliği Sınırların Ötesinde adlı saygın uluslararası sempozyuma hepiniz hoş geldiniz demek benim için bir onur ve ayrıcalıktır. Günümüz hızla ilerleyen tıp dünyasında temel bilimler ile klinik uygulama arasındaki sinerji hiç bu kadar önemli olmamıştı. Bu sempozyum uzmanların çeşitli alanlarda bir araya gelerek son teknolojik araştırmaların keşfetmelerini, acil sorunlarla çözüm bulmalarını, sağlık hizmetlerinin geleceğini hayal etmelerini sağlayan kritik bir platform olarak hizmet etmektedir. Tıp Fakültesi Dekanı olarak Bugün burada toplanan bu çeşitli ve seçkin katılımcı grubu görmek beni son derece memnun ediyor. Katkılarınız ve görüşleriniz kesinlikle tanışmalarımızı zenginleştirecek ve yenilikçi işbirlikleri ve çözümlerin yolunu açacaktır. Sempozyum boyunca nadir hastalık ekosistemlerinden gelen düzenleme teknolojilerindeki en son gelişmelere kadar birçok konu değerli konuşmacılarımız tarafından ele alınacaktır. Her sorun ve tartışmanın yeni bakış açısı sağlayacağına ve fikir alışverişinde verimli olacağına inanıyorum. Bu sempozyumu düzenleyen hocalarımıza, saygıdeğer konuşmacılarımıza katılımınız ve tıbbi bilimin sınırlarını ilerletme konusunda bağlılığı için her birinize içten teşekkürlerimi sunarım. Thank you very much.
Thank you, Professor Enver, for your speech. So for the audience, the participants that are here, I just would like to translate in English the opening speech of our dean. So his speech is like that. Dear Professor, distinguished guests, esteemed colleagues and participants, it is my honor and privilege to welcome you all the esteemed International Symposium for Fundamental and Clinical Sciences, organized in collaboration with İSTANBUL AYDIN University Faculty of Medicine, the Health Policies Research Center, and Anatolian Education and Culture Foundation. In today's rapidly advancing world of medicine, the synergy between fundamental and clinical sciences has never been more crucial than before. This symposium serves as a vital platform where experts from various fields come together to explore cutting-edge research, address pressing challenges, and envision the future of healthcare. As the Dean of Faculty of Medicine, I'm thrilled to see such a diverse and distinguished group of participants gathered here today. Your contribution and insights will undoubtedly enrich our discussion and pave the way for innovation, collaboration, and solutions. Throughout the symposium, we will delve in topics ranging from rare disease ecosystems to the latest advancements in gene editing technologies. I'm confident that each presentation and discussion will inspire new perspectives and foster fruitful exchanges of ideas. I extend my sincere gratitude to our organizers, our esteemed speaker, and each one of you for your participation and dedication to advancing the frontiers of medical science. So he says, best wishes to all of us, and wish a wonderful symposium with valuable discussions that all of our esteemed speaker will be today. So thank you again, Professor Enver, for your speech. So now I will give the turn to our valuable Professor Mine Ergüven. As now I would like to say that I have the privilege of hearing from, and it is with great pleasure that I introduce our speaker, Professor Dr. Mine Ergüven, whose invaluable dedication and expertise as the organizer of this symposium has been instrumental in bringing us all together today. As a distinguished member of Department of Medical Biochemistry at the Faculty of Medicine English Program, İSTANBUL AYDIN University, Professor Dr. Ergüven's leadership and vision have guided us through the planning and execution of this event. Please join me extending a warm welcome as she shares a profound insight with us. Yes, Professor Ergüven, now is your turn. We don't hear you.
I'm sorry. Thank you very much, Dr. Armani. I'm very happy to hear these wonderful sentences from you. With respect to their chairman of the Board of Trustees and members of the Board of Trustees, their rector and vice rectors, the president of the Anatolian Education and Culture Foundation, their dean and vice deans, their colleagues, and their lovely students. I would like to welcome you to Turkey as its intersection point of Asia and Europe, to Istanbul and our symposium. I'm very happy to organize this symposium themed on the intersection of fundamental medical sciences with clinical sciences and their results for human being health with my colleague, their Dr. Arta Armani. We organized this symposium on behalf of İSTANBUL AYDIN University, the Faculty of Medicine, the Anatolian Education and Culture Foundation, and the Health Policies Research Center. In this symposium, we are shaking hands with distinguished, prestigious experts from several countries under the umbrella of their currently working unique subjects and years of accumulated experiences. We appreciate our speakers for their acceptance to give talk. I hope this symposium will give rise to new collaborations, new innovative projects, and new friendships. And it is very valuable for us. And I wish a very plantful, fruitful symposium. Thank you very much.
Thank you, Professor Ergüven, for your speech. And the last is mine, but I talked a lot. I think I do not have so much time because after 3 minutes, we will start our symposium with our first valuable speaker. But as your chair of this symposium, together with Professor Ergüven, it's both an honor and a pleasure to stand before you today. My name is, as written here, Art Armani, and I'm deeply grateful for the opportunity to facilitate our discussion and exchanges over the course of this event. Before we delve into the rich array of topics that await us, allow me to express my heartfelt appreciation to all of our distinguished speakers, organizers, collaborators, and participants. It's your collective dedication and enthusiasm that have made this symposium possible. And I'm confident that together we will foster an environment of learning, collaboration, and innovation. So over the course of the coming sessions, we have the unique opportunity to explore the intersection between the fundamental and clinical sciences, unlocking new pathways to discovery. So through engaging presentation, through provoking discussion, and fruitful interaction, I'm excited to witness the exchange of ideas and insight that will undoubtedly propel our collective understanding forward. So thank you again for being here, and we are honored to host you, all of you here. And just before starting our session, first part session, I would like to give two announcements that before each speaker presentation, me and Professor Ergüven will give a very brief information about the academic and research background of each speaker. And of course, we have one part of the student, in fact, and academician that are in the conference hall in our campus. So at the end of each presentation, if they have any question, they are going to send to us. So welcome you again, and thank you for all of our opening speech speakers that were here. Our dean, the previous one, Metin Kapan Hoca, that already stimulated me and Professor Mine Ergüven to start organizing this such symposium. And thanks for all of being you here. And let's start with our first speaker, Professor Dr. Kenji KADOMATSU. Yes, Professor Ergüven, it's your stage now. I just will make a sharing of our first.
Excuse me, I would like to remind you about the introduction video of our university. Would you like to share this video before?
I sent it to you, but I couldn't upload it now.
Yes, I just did it. And
okay, so you can share it from there.
We are very sorry for the speakers. And please excuse us. We will first of all start our introduction about university, and then we will start our symposium. Kind of you. Yes, I'm sharing.
İSTANBUL AYDIN University for Your Dreams and Tomorrows. Established in 2003 by the Anatolian Education and Culture Foundation, AKEV, has taken its place in higher education with 18 faculties and colleges. Our university, which has gained renown both in Turkey and internationally for its scientific research, is a complete city university with its experienced academic staff and campuses located in the heart of Istanbul. Established with the aim of preparing competent physicians to meet the challenges of healthcare in the 21st century, our Faculty of Medicine is a modern and large institution providing education, research, and healthcare services with its own university hospital located on a large campus area. Our Faculty of Dentistry prepares future dentists with contemporary approaches and modern technology, thanks to our dynamic team and experienced faculty members in each department. Our Faculty of Health Sciences trains young people who will serve in the field of health in our country. We take pride and honor in being with you as the Faculty of Pharmacy. The primary goal of our Faculty of Education is to educate our students as teachers who can think differently, be free in their individual choices, confident, respectful of human values, tolerant, self-sacrificing, and share the ideals of the Republic so that we can entrust future generations to them. Our Faculty of Arts and Sciences aims to educate graduates with an understanding that closely follows contemporary developments and constantly renews itself. Our Faculty of Fine Arts, which is aware of the meaning and value of art in human and social life, aims to train well-equipped graduates. Our Faculty of Law continues to make significant contributions to legal education and scholarship since its establishment. The Faculty of Economics and Administrative Sciences educates students with the knowledge and experience required to raise the Turkish economy and society to advanced standards at all levels. Our Faculty of Communication aims to cultivate communication professionals who embrace the world they live in, possess a holistic perspective on events and phenomena, are always questioning, have strong critical and analytical skills, and can anticipate the world of tomorrow from today's standpoint. In our Faculty of Architecture and Design, we instill artistic sensitivity, creative power, and self-confidence in our students and provide them with the technical skills necessary for them to perform their professions. Our Faculty of Engineering has achieved MUDEC accreditation with all its departments, completing the infrastructure requirements for theoretical and practical education. The Faculty of Sports Sciences offers undergraduate, master's, and doctoral programs in physical education and sports to train teachers, sports specialists, sports scientists, and academicians. The Faculty of Applied Sciences aims to train human resources with vocational education, which is increasingly needed both in the world and in Turkey, parallel to the increasing competition driven by science and technology. 24/7 Open Information Center. Anadolu Bil Vocational School. Vocational School of Health Services. Prep School. We have a total of 165 applied laboratories serving in many fields, from computer science to food, from automotive to health, from textiles to chemistry, from mechatronics to physics, from molecular biology to civil engineering. Medical and dental education is provided in 36 research centers. Named after our Nobel Prize-winning famous scientist, Professor Dr. Aziz Sancar, our Techno Center is Turkey's most prestigious multidisciplinary technology center established in a single enclosed area. Our university cooperates with 352 universities from 52 countries and Erasmus Plus with more than 500 world universities. Technology Development Center and Technology Transfer and Project Management Office contribute to the economy by transforming innovative ideas into products, along with the Entrepreneurship and Incubation Center. İSTANBUL AYDIN University is the only university in Turkey with 2 UNESCO chairs hosting the Education and Cultural Diplomacy for Sustainable Peace and Governance and Education chairs. Student clubs. University's think tanks engage in discussions and exchanges of views on political, economic, social, cultural, religious, and current issues. Expert individuals are invited to these meetings at regular intervals to benefit from their expertise. With 6 conference and seminar halls of varying sizes, our university can accommodate up to 3,500 people simultaneously. We host dozens of national and international events every year. Our dormitory is in the class of a new generation living complex with a warm home atmosphere and hotel comfort.
Yes, thank you very much. We can start, I think.
Yes, Professor. Now is your turn. You can start.
Thank you, but yes.
Do you see my screen?
Yes,
Okay.
but I must do some arrangements right now. Thank you very much. Today's the morning session first speaker is Professor Dr. Kenji KADOMATSU, Vice President of Nagoya University, and he will give a speech about Human Genome Atlas project and infrastructure development project for life sciences. I know Professor KADOMATSU since 2010. I'm also one of the Mitkin research scientists, and he's co-inventor of Mitkin. So I'm very excited to host him here. And I would like to give a brief introduction about his history, and we will continue there, Armani. Thank you. And first of all, he got his MD degree at 1982 from School of Medicine, and Kyushu, from Kyushu University, Japan. And he got his pediatric surgeon title at 1988, again from Kyushu University, Japan. And he got, I think, a PhD biochemistry, medical biochemistry, PhD degree from previous, thank you, at 1989, and again, Kyushu University, Japan. Yes, please. And when we look at the academic career of Kenji KADOMATSU, he got his degrees from assistant professor to professor from Nagoya University, Graduate School of Medicine. He is right now at biochemistry, medical biochemistry department. Yes, please. And in addition to research and research activities and academician titles, he has also administrative duties, several administrative duties, trustee, director, dean, and at last is vice president of in charge of research at Nagoya University, Japan. Yes, please. And this slide cannot explain his working, wonderful, unique publications and activities and experiments, and only very, very brief explanation about field of research can be life sciences, medical biochemistry, glycobiology, and neurobiology. He has also awards and honors, and at 1997 he got the Japanese Biochemical Society Young Scientist Award, and 2001 he got Walker Buzzer Award shared with Dr. Lee, and at 2023 he got Chunichi Culture Award, and as I told you, he is the co-inventor of Mitkin protein with Professor Takashi Muramatsu. And you look at right now you are looking at research activity in view of age index and and stations. But as I told you, he's much more than these slides and these numbers. Yes, please. Yes, right now it is my pleasure, it is my honor to invite for his talk Professor Kenji KADOMATSU. Yes, you can start the KADOMATSU. Nice to see you again. Thank you.
Thank you, Mine. Good to see you again. Yeah, it's a long time of friendship, actually. And hello or good morning, everyone. My name is Kenji KADOMATSU. May I share my slides?
Yes, please. Yes, you can write to share your slides.
Oh, probably I cannot. You have to permit me to share.
We already promote
did.
you to the panelists, so you can now share from the screen on the middle of the maybe bottom of the page. Could you see anything?
No, actually, it's not permitted.
Okay, I make a co-host again to make sure that does it work. One more possibility. Can you check it again, Professor?
Okay, now, yeah, probably.
Okay.
Can you see it?
It is starting. Yes.
Yes.
Yeah, okay, thank you. So maybe, okay, you can see it, right?
Yes.
It's now full screen, right?
Full screen.
Yeah, okay. So my name is Kenji KADOMATSU, and actually in Japan now it's 4 o'clock in the afternoon, so it's a good time for me actually. But somehow, thank you very much for this wonderful opportunity. And I'm from Nagoya University in Japan, and also I'm serving as a director of the Institute for Glycocore Research, IGCORE. I will present today the Human Glycome Atlas project, which is a very important infrastructure development project for life sciences. Okay. Since I'm only Japanese among the speakers today, so I will just briefly introduce you Nagoya and my institute first. Just in case if you don't know, this is Japan. I'm sorry, Turkey and Europe is over there. And then as you can see, Nagoya is located between Tokyo and Kyoto or Osaka. It takes only 1 hour and 40 minutes to Tokyo and 40 minutes to Kyoto by Shinkansen. And as you see, I'd say Nagoya is the center of Japan. And also the greater Nagoya area include the Gifu, Toyota, and Ise, right? And then about the 500 years ago, these 3 shogun unified Japan. So therefore, they are very, the most famous shogun in Japan. or the Toyotomi and Tokugawa. And then the most important thing is they are born in Nagoya area, all born. The Nagoya Castle is a symbol of Nagoya, and then was built by Tokugawa. And the Ise Shrine is the most important shrine for Japanese because it's a symbol of the Japanese religion Shinto, and it's always new because it's rebuilt every 20 years for our emperor. And so many industries gather in this area. Indeed, the Nagoya port is the #1 trading port in Japan. And the Toyota City is next to Nagoya. And as you know, it's a hometown of the automobile company Toyota. And this area is also good for in the agriculture. Some of the products actually the #1 production in Japan, right? And then finally, the Nagoya University is proud of 6 Nobel Prize laureates. The important thing is these guys were here in the Nagoya area. So this fact is very encouraging for us. We indeed feel something like, yes, we can. And this is our, institute vision, IG-Core. IG-Core is the glycoscience, that is science of the sugar chains for the future life sciences. So to this end, the 3 centers focusing on physics, chemistry, medicine, biology, and data science will cooperate to create the future glycoscience. And the connection between the human and future glycoscience gave rise to the Human Glaucoma Atlas project, which I will talk about today. And we do cover not only the human, but also other organisms such as animals, virus, plants, bacteria, and so on. I think these all activities should be linked to the creation of the future life sciences. So by incorporation glycans into life science, we will come to see the brilliant and precise future of life. So incorporation or development of the new technologies and also the collaborations with many other scientific fields will facilitate our activity or movement. So our institute indeed are conducting 2 national projects. The first J-Glyconet and second Human Glycom project. The J-Glyconet create the all Japan supporting system for glycoscience. And then it will facilitate or help the activity of the Human Glaucoma Atlas project. So this Human Glaucoma Atlas project will create the glycan and knowledge base TOSA. Okay, so the information of glycans stored in TOSA will be used by the research community, including not only the glycosciences, but also some other fields such as neuron or cancer or genome proteins and so on. Okay, now I'm just entering today's topic. I will just introduce you the glycans first to you because probably most of you may not familiar with the glycans. And then I will address the Human Glaucoma Atlas project. Our body consists of 3 major life chains: the genome or nucleic acids, proteins, and glycans. As you see, the glycans cover the every cell. Without exception, every cell is covered by the forest of glycans. Therefore, when the cell meet other cells or other particles such as a virus or growth factors, the glycans are always the forefront. So from here, I'll just introduce some biological roles of glycans. First, the glycans determine some cell identities. As you know here, the ABO or blood types, glycan determine these blood types. Say, for example, in my case, I'm A, so I have this kind of glycans. But if you are B, you have glycans different from mine. And then glycans also play an essential or critical roles in the virus infections. So, for example, here, the influenza virus, okay, our airway epithelium harbors glycans, and these glycans actually is essential for the virus entering as well as releasing. So the drug Tamiflu was developed targeting these glycans. If you think about the COVID-19, as you know, the spike protein and our body's ACE receptor, these two guys must meet each other. Otherwise, they cannot, the virus cannot, infect us, right? And then if you look at this, both the spike protein and ACE receptor covered by glycans. And it reported that the glycans play an essential role for these bindings. And also, glycans are involved in the immunities. Say, for example, here the antibody, all antibody, IgG, is harboring the N-glycan at the Fc fragment. And then often it harbors also fucose. We call it core fucose. Okay. And then so that it can suppress or control the hyper response to the immunity. However, if you remove this fucose, it increases the binding between the IgG and Fc receptor on the effector cell by 100-fold. So now the effector cell release the harmful cytoharmful cytokines and then kills cancer cells. This technology is called POTELGENT. It's actually used in clinically used. I thought this is just artificial technologies, but recently I realized that from this paper, it actually occurs naturally. So this paper, compared non-ICU patient and ICU patient. The ICU patient, most of them harbor the IgG without fucose, so that this binding will increase or induce the cytokine storm. That's why the ICU patient show the severe condition. This is discussed in this paper. And also, as you may know, the glycan serves as a good disease markers. Say, for example, the MAP2 binding protein glycosylation isomer. This glycosylation is different from the ordinary glycosylation. So this aberrant glycans are specific for the liver cirrhosis as well as liver cancer. And CA99 is a very famous marker for the cancers, and itself is glycan itself. It's good for the pancreas or other cancer markers. And as we saw in the case of the Tamiflu in the influenza, glycan also serve as the therapeutic targets. So these guys, including the Carolyn Bertozzi, who won the Nobel Prize, founded the Paleon pharmaceutical, and then they announced that they, developed the antibody enzyme conjugate designed to trim the immunosuppressive thioglycan coating cancer cells and exhausted T cells, so that it can restore the anti-cancer immunities. Right. Glycan also works as a co-receptor for growth factors. In general, growth factor can bind to its own receptor and then transduce the intracellular signaling. But often they require the co-receptor. Some of them are glycans. Say, for example, as Mine introduced, in my case, I've been working on the growth factor midokine. This is a heparin binding growth factor. So the midokine can bind the heparan sulfate glycan. And then, this is also the case for, say, for example, the basic FGF and other many growth factors, right? From here, using several slides, I will show you another work in my laboratory. Okay, so a big question is our, central nervous system, adult central nervous system neurons cannot regenerate after injury, especially the axons. The proximal region of the axon after injury can survive, but cannot regenerate. That means cannot reconstruct the neuronal network as previously. So this is a problem. So there are two reasons for this. One is a low intrinsic capacity of regeneration of adult central nervous system neuron axon. And second, the emerging inhibitors of regeneration. So the representative inhibitors are long sugar chains, glycans. So condotin sulfate and kertan sulfate. However, although it's also the long glycans, heparan sulfate is a promoter for the axon regeneration. And the further question is the condotin sulfate and heparan sulfate share the common receptor PTP sigma. So although they share the common receptor, they show the opposite effect on the axon regeneration. This is our specific question. So for example, if you can see this one, this is just a control rat. It shows the almost complete paralysis of the hind limb after spinal cord injury. However, if you administrate the keratan sulfate, the digesting enzyme, or condotin sulfate, digesting enzyme, you can ameliorate the functional disturbance. So these movies apparently suggest that the condotin sulfate and heparan sulfate are the strong inhibitors. On the other hand, the Santiago Ramon y Cajal, who is a father of the modern neurology, he left a very interesting drawing as shown here. So these are injured axon tips. So they show the ball-like structure, very bizarre structure. We call it dystrophic end ball. And we first checked whether he is correct or not. Actually, we found that, sorry. So this is what we found. Almost all the axon tips after injury show the dystrophic end ball formation. So without understanding the dystrophic mechanism of the dystrophic endocyte formation, we cannot cure the neuronal injury. That's our thought. So this is just to recapture the dystrophic endocyte in vitro. So left is the control. The axon just goes forward without hesitation. It looks happy. And the tip of the axon shows the growth cone. On the other hand, on the right side, we have chondroitin sulfate gradient on the substratum. In this case, axon just go forward, but at some point, it forms, stops and forms dystrophic endocyte and then retract. It's just a repeat this kind of phenomenon. This movement would also happen in our body after neuronal injury, such as brain injury or spinal cord injury. So if you look at the, tip of axon, left shows the growth cone, and then right shows the dystrophic endocyte. And then not only the shape, but also the content are different, right? So there are so many vesicles in the dystrophic endocytes, and we are curious, what are these? So to make a long story short, those are autophagosomes. In the default state, the heparan sulfate can oligomerize the PTP sigma. This receptor type tyrosine phosphatase is now turned off. So the phosphocotactin is over there. So this cotactin phosphorylation actually is essential for the fusion between the autophagosome and lysosome. So just this autophagy flux goes normally. As a consequence, axon regeneration can be promoted. On the other hand, once the axons are injured, chondroitin sulfate emerge. So now it's monomerized the PTP sigma, and then this receptor enzyme activity is turned on. Since this is a protein tyrosine phosphatase, the cotactin is now dephosphorylated. So this fusion between the autophagosome and lysosome is disrupted. That means this autophagy flux is disrupted. As a consequence, this dystrophic endocyte is formed and axon regeneration is inhibited. This is the underlying mechanism we found. Let me go back to the story of the glycans. So the glycan science gave several Nobel Prizes, such as Karl Landsteiner, which I already showed you the finders of the blood type ABO. And recently, as I also showed, the Caroline Bertozzi won the Nobel Prize in chemistry for her finding in the Greek chemistry. But her main research field is Greek science. Now I will introduce the Human Glaucoma Atlas project. More than 10 years ago, I conducted a project named Glyco Neuroscience. At that time, I proposed that although the neuroscience showed the striking progress these 2 or 3 decades, but the main players were proteins. However, if you look at the tissues, there are so many glycans. So without understanding the glycans, you cannot understand completely the role or function of the nervous system. And then actually this project was very successful. But on the other hand, we felt that we don't know the landscape of the nervous system as well as other tissues, landscape of the glycans, right? So I propose that we will have a kind of a comprehensive structure capture by the project named Human Glycome Atlas Project, which was just started in Japan. Although, as I told, glycan have a very important roles in our body, but almost no people don't know the glycans, and many scientists avoid glycans. This is there is a reason. Is just one reason is the glycan are difficult to study. This is a structure that from a short to long sugar chains, and then they often have branching. This structure, I mean, is quite difficult to elucidate the precise structures. And also, if you think about the glycoproteins, the DNA to RNA, RNA to protein, these biosynthesis depending on the template. However, the glycan formation is not dependent on the template. And furthermore, there are so many sites for the glycosylation. So you have to consider both the various glycan structure and various glycosylation sites. So this combination makes the structure complex. However, we are now entering an era when these problems are overcome. So thanks to the development of the methodology such as mass spectrometry and also the informatics. So this is the aim of the Human Glycome Project. So comparing with the genome of proteins, the information of the glycans is significantly limited. This is why the many scientists in other fields avoid glycans to explain life. So HGA, Human Glycome Atlas Project, will increase the volume of the glycan information to the level of the glycan and, the genome and proteins, so that every scientist can use the glycan information without hesitation, then innovate the life sciences and medicine. So if you think about the glycomics and glycoproteomics, here is a glycoproteins. If you'd like to do the glycoproteomics, you will cut the core protein with enzymes such as trypsin. So you can now obtain the glycopeptide. So after enriching the glycopeptide, you will subject these samples to the mass spectrometry. On the other hand, if you'd like to do the glycomics, you will cut off the glycans from the core protein. So only the glycans can be, collected and then subjected to the mass spectrometry. We will do both. So this comprehensive glycoproteomics is a method of the in-depth glycoprotein structure analysis. So finally, we will create the two-dimensional glycopeptide map. Okay, and then using this 2D map as a reference, we will perform the autoglycoproteomics. So this is a high throughput quantification of plasma or other sample glycoproteins. Okay, so we are now almost completed the construction of the auto analyzers. If you'd like to do the glycomics, the glycans can be categorized into 5 classes: N-glycans, O-glycans. These are linked to the proteins. And then glycosphingolipid, this is a glycolipid. And then glycosaminoglycan and free oligosaccharides. All these 5 categories can be captured by a method called total glycomics. Okay. This is the annual plan of a Human Glycome Atlas project. This is a 10 years project. In the first 5 years, we will focus on the dementia and aging by analyzing 20,000 blood samples. In the next 5 years, we will expand our target to the cancer, diabetes, and other diseases by analyzing 200,000 blood tissue and organs samples. We will also develop the auto analyzers as well as informatics, and we will elucidate the biosynthetic mechanisms of the glycans. Right. So here I will show you the history of the infrastructure development project for 3 life chains. So as you know, the most prominent project was the Human Genome Project. It changed life sciences, but society as well. So now we are now entering an era when the genome medicine is daily used. Regarding the proteins, Protein 3000 and Human Protein Atlas greatly contributed to the progress of the life science. And finally, glycan have come, right? The Glyco Science Society in US, Europe, and Japan tried to promote the Glyco science through making the roadmap as well as database alliance. And then now the Human Glycome Atlas project has been commenced in Japan. And Japan is one of the leading countries in Glyco science. For example, more than half of the glycogenes were discovered by Japanese, and we will do our best for the success of this project, and also call for collaboration to many other countries, right? So this is a global science network, Glyco Science Network. We have a MOU and also Quadrico program, and also academic cooperation and research exchange meetings. In addition to these, we have a database alliance between the Japan, Europe, and US for the Glycospace Alliance. And furthermore, the European Glyco Science Society, CARBOMET, and Canadian Society, GlycoNet, supports the Human Glycome Atlas project. And finally, the many foreign scientists gather in our institute to help the Human Glycome Atlas project success. Right, this is the final slide. This is the future of the Human Glycome Atlas project. It's an infrastructure development project for the future of life sciences, and is expected to provide benefit to the public, such as disease prediction and prevention. Thank you very much.
Thank you very much, Professor KADOMATSU. Very fascinating work and very promising work for the future. Especially, I think that it will also be good for the treatment of Alzheimer's disease. And it could be because, you know, although it was told that the reason of Alzheimer's disease is because of amyloid structures, but last publications showed that it is not only the reason, so it could be cure for the treatment of Alzheimer's disease. And I would like to ask the audience if they have any question to Professor KADOMATSU.
Dear Professor Ergüven, there are two questions to Professor in the question part. Maybe you can read from there, from the students that are in the class.
Yes, I have also two questions. And also I would like to tell about their questions. First of all, question from students in the conference hall. And dear Professor Kenji KADOMATSU, given the vast potential of the Human Glycome Atlas project to revolutionize our understanding of glycan structures and their implications for health, how do you envision bridging the gap between the fundamental research and clinical application, particularly in terms of translating this knowledge into diagnostic and therapeutic interventions? This is the first question. Yes, please.
Yes, as I told, the glycan has so many roles or functions so far known. Especially say for example, the what should I say, disease markers, as indicated, Alzheimer's disease. We don't have very good marker, very early state of the Alzheimer's. That's why we cannot, what should I say, treat the disease actually. That is a problem clinically. So this kind of marker finding, this project will give the fundamental information. This is the first thing. And also, as I told, the Tamiflu case or Bertozzi's, the startup ventures case as well, could be, the glycans could be a target for the therapeutics, right? So this would be also connect this project and clinic. Yeah, this kind of things that we are now thinking about.
Thank you very much. And second question from audience. As the Human Glycome Atlas project progresses, how do you plan to address the challenges of data standardization, harmonization, and accessibility across diverse research communities worldwide? Additionally, how will the project ensure the ethical considerations such as privacy and consent are adequately addressed in the sharing and utilization of this valuable data source?
Yes, and this is a very important question. Probably this is from the, expert scientist. I think actually how should I say, on the same platform, on the with the same method, and then with the same how should I say, interpretation of the raw data. These kind of things are very important to make this project or data from this project available for all of the scientists in the world. So this is actually, we are always thinking about it. And then now I also show we already have the global network. We will discuss this kind of issue. Yeah, this is very important things. Yeah, and then sharing and utilization data resource. This is certainly be open to the scientists worldwide. But at this moment, we are now constructing the kind of a data base. At this moment, it's not available, but in the future or in the near future, it will be available. that is our duty, I think. Yeah.
One more
And
question.
also the other countries will help us. We are we will calling for collaboration with other many countries. Yeah.
Thank you very much. And one more question. How do you foresee leveraging this multidimensional data set to uncover novel biomarkers or therapeutic targets for diseases where glycosylation plays a significant role, such as cancer or autoimmune disorders?
Excuse me, could you just repeat that again?
Yes, please. Of course, my pleasure. How do you foresee leveraging this multidimensional data set to uncover novel biomarkers or therapeutic targets for diseases where glycosylation plays a significant role, such as cancer or autoimmune disorders?
Yeah, I mean, in my presentation, I already give us some examples, but somehow we will just, I mean, give you not necessarily only the biological, I mean meaning or data. We are just provide you the fundamental fact or fundamental data you can use, I mean, freely if you'd like to compare with the normal people and then Alzheimer people, or normal people with the pancreas cancer people, something like that. So you will find, or we will find, some difference. This kind of works should be done. I mean, then so this kind of cooperation will make this project successful. Yeah, that is our thinking. Yeah.
One more question from me. Did you have a chance to check nuclear factor kappa beta activity during this process? You know, it is very important, and you know that it has diverse effects. So did you have a chance to check the activity of it?
Oh, actually, not yet. Maybe we will actually approach more or less, I mean, comprehensive approach, not necessarily the very specific target.
Yes.
So if it is included in our data of the comprehensive approach, that we will just go forward. But at this moment, we don't have I mean, such a specific target at this moment. Yeah.
Okay. Thank you very much. We are very glad to host you in this meeting. And in future, we need to meet again, of course, for the collaborations, for new studies, and for new symposiums. Thank you very much for giving time and sharing your fascinating knowledge, experiments with us. Thank you.
Thank you very much. See you again.
See you.
Bye-bye.
Yes, the second speaker is another distinguished speaker is Associate Professor Dr. Guan-Da, and he is from National Cheng University, and he will give a speech about developing multiplex immunoassays for monitoring human immunity after viral infections. And right now it is also a very common subject. You know, we continue to live with COVID-19, so I think that it will be okay for us. Yes, please. I would like to give brief introduction about his history. First of all, education. He got a bachelor degree from environmental engineering, minor in life sciences at 2005 from National Cheng University, Taiwan. And he got PhD degree from Graduate Institute of Basic Medical Science at 2012, and then again from previous, please. Dr. Arman, thank you. National Cheng University, Taiwan again. Yes, please. Thank you. And he has academic career starting at National Cheng University, and it's also continued at National Cheng University. At last, he got his associate professor doctor degree, and he got this degree from the Department of Biotechnology and Bioindustrial Sciences. He got this degree at 2023, and Conrad's their professor, and from National, previous please, Dr. Armani, from National Cheng University, Taiwan. Yes, please. And he is also very wonderful field of research and variety of research issues, and biotechnology and bioindustrial sciences, host and microbial interactions, protein macroarray, high throughput technology, virology, and bioinformatics. And of course, we know that these numbers are not reflecting the researcher. The researchers are more than these numbers. And you know that right now you are seeing his research activity in the view of H index, I-10 index, and stations. And he has several awards and honors. And I would like to tell the last two ones, a lot of, and Conrad's their professor. And he got Future Technology Exhibition Award at 2022, and he got National Innovation Award at 2022. Yes, thank you very much again. We appreciate, and I would like to welcome you to give your talk, and also thank you very much for your patience. I know that you wait a little bit, and kind of you. Thank you. Yes, Doctor, you. We are waiting for your talk.
Thank you, Professor Dickelman. Can you hear me clearly?
Excuse me?
Can you hear my voice?
Yes, very good, kind of you. And
okay, thank you.
We are seeing screen, yes.
So thank you for your kind introduction. Today I'm going to share the recent research about the multiplex immunoassay for monitoring humoral immunity after viral infections. And Taiwan is kind of neighborhood to Japan, so I'm going to follow the topics from Professor KADOMATSU about the COVID-19. So our department is biotechnology and bioindustry sciences. So we are more focused on the biotechnology part. And this is the typical image of protein microarray with separate the spots in the distance and to form a high throughput screening. So here I want to briefly introduce the functional protein microarray. So the functional protein microarray is by expressing high throughput purification and high content printing to form a screening platform usually on the glass. So each protein are printed on the glass and to form the functional format with the native conformation. And this protein can be used to screening for the signal transduction events in the cells or the antibody serum profiling. So as you may know, after immune response, they will generate antibodies. So using the protein platform as an antigen platform, we can screen in the immune response or antibody specificity on the array. So one other application is screening the small molecules because protein have a conformation, so we can screen in for the small molecule specificities. And we also can use the protein microarray to find out the enzyme and substrate interaction and identify what's the enzyme substrate, such as what's the substrate for the kinase, what's the substrate for the ubiquitination. So in this topic, I want to mainly focus on three relative to the infectious disease. First is development and application of coronavirus protein microarray. So as you may know, the coronavirus, there's several subtypes. There are some common cold coronavirus named 229E, NL63, HKU1, OC43. Those are commonly distributed around the globe and account for 15% of the common cold. And there are some SARS-CoV and SARS-CoV-2, which have a case fatality rate with 9% or from 1 to 6% in the early years. And the more severe case is called MERS-CoV. It have a case fatality rate 37%. Among those coronavirus, they have a similarity in the genome and proteins. So we want to know whether the newly happened SARS-CoV-2 can generate cross-immune reactivity. So later on, we fabricate the protein microarray contain all the human coronavirus. We call it HCOV protein microarray. And this array have a high reproducibility with R² 0.984. And later on, we want to test the detection limits of the protein microarray and by using the anti-S monoclonal antibody. And by serial dilution, we can find out the limit of detection is around 19 picogram. And this means very sensitive compared to other methods such as ELISA or a rapid test. And we also want to prove the protein on the array are functional. So the best way to prove it functional is by binding to the host receptor. So we use the host receptor ACE2, and that will naturally bind to the SARS-CoV. So as you can see, with the serial dilution, we can see the open circle is the SARS-CoV-2. It will bind to it. And the open square is SARS-CoV spike protein will also bind to it, but not MERS-CoV. Because MERS-CoV, the host receptor is DPP4 instead of ACE2. So this array demonstrates the function of the protein immobilized on top and also show the specificity. And later on, we want to ask whether the COVID-19, the specificity of the COVID-19 after infections. So we collect the patient and healthy controls and profile their human IgG and human IgA. IgG is the most abundant hemoglobin in the serum, but human IgA is also important in the mucosal immunity. So with the profiling, we can find some markers to distinguish the COVID-19 from healthy control. With this heat map, you can see IgG and IgA in the COVID-19 patient. They will show that markedly increased. And they don't show much signal in the healthy control. And we find out some specific marker or combinatory markers to show the high sensitivity. Beyond this, we also want to know what about the cross-reactivity. So SARS-CoV-2 always cross-react to SARS-CoV because they have the around 75% identity compared to the SARS-CoV-2. So that's why they can generate cross-reactivity. How about the other common cold coronavirus? Is something like HKU1. Although they only have 29% identity, they still generate cross-reactivity. And same with the OC43. Even though there's only 30% similarity, they also generate significant cross-reactivity. And across our country in Taiwan, we find out because Taiwan is a high population density, about 86% of healthy people carry the serotype OC43 positive, and about 50% of the Taiwanese carry the HKU1 positive. So this means in the crowded population in Asia, there are more excuse me, they are more popular with those common cold coronavirus. And this may reflect in the COVID-19 pandemic or endemic because there's less severity in Asia with the COVID-19 compared to Europe or America. So because there's the mutation is ongoing, because it's an RNA virus for the SARS-CoV-2. So there's alpha, beta, gamma, delta, Omicron waves, and generate waves of transmission. And the method to vaccinate it or after infection, they will generate neutralizing antibody. And this neutralizing antibody is meant to block the infection. So it's usually is meant to bind to the spike protein from the SARS-CoV-2 and inhibit its infection through the ACE2 receptor. So that's the meaning of the vaccination or after infection. However, during the early stage, the vaccination is only focused on the wild type. So we want to run a study to show whether the vaccination efficacy throughout the different variants. So we designed a protein microarray from the wild type to Omicron spike proteins and printed on the slides and showing the reproducibility of R-squared 0.974. And later on, after we build the protein microarray, we want to build an in vitro assay to compete the human ACE2 with the human antibodies. So in here, let's take a look at the bottom images. Let's focus on the red part first. The red part is the ACE2. It's showing the is to mimic the infection. So naturally, spike protein will bind to the ACE to show the red. But if there's existence of neutralizing antibody, which label in green, and more green will inhibit the red. So it's a protection against infection. So we can show in the high throughput way to show the serum protectivity against multiple variants. So next we run the clinical trials. First, we use the unvaccinated subjects. And as I highlight in the box, in the unvaccinated subjects, they are showing in the red because it demonstrates the infection. And the vaccinated subjects will show in green or orange because they generate a human antibody and protect from the infection. So by using this assay, we can quantify the after AstraZeneca vaccine 1 dose and AstraZeneca's vaccine 2 dose. And we can see that in the AstraZeneca vaccine is generate huge person-to-person variations. And the average protection or inhibition is around 40% in the first dose and 60% in the second dose. And for the Moderna, the messenger, the mRNA vaccine, in the first dose, it averaged to generate 80% of protectivity. And in the Moderna 2 doses, generate around 97% of protectivity. In the unvaccinated subjects, it generates around 15% of the protectivity. Because of most of the people are infected with the common cold coronavirus, as I mentioned before, and generate cross-reactivity to contribute some neutralizing activity. So further, we analyze with AZ2 dose subjects and showing in the different variants. In the wild type, it averaged with 60%, but in the Omicron, we can see it only have 20% of neutralizing activity, which is no significant difference with unvaccinated subjects. That's why we always get a breakthrough infection for the Omicrons. So how to generate better neutralizing antibody through the traditional vaccine. So we also run a clinical trial with AZ 2-dose, Moderna 2-dose, and mixing with AZ and Moderna. And we demonstrate the mixing of AZ and Moderna generate the best protectivity against Omicrons with 80% versus 60% or 25%. And we also want to demonstrate the use of high throughput technology in the drug development. So we demonstrate with two small molecule inhibitors for ACE2. One is called Benadryl and one is called Ramipril. These two small molecule inhibitor shows dose-dependent inhibitor to the infection. However, when the variant or mutation accumulates in the delta, the dose-dependent response is blunted, and in the delta plus, it will vanish all the dose-dependent effects. So this demonstrates the power of the high throughput technology and demonstrate the mutation, the influence of the mutation in vaccines and small molecule inhibitors. So here I want to give you a brief summary about the two types of array, human coronavirus array we can use to profile the serum antibodies, and the coronavirus variant protein microarray to profile the neutralizing antibodies. And that is about one of the problem in the tropic countries is a flavivirus or called dengue. So in the dengue, there's it's distributed in the tropic or subtropical. Even in Taiwan, we have thousands of dengue cases per year. So in every year, people get infected with dengue, but a small number of people get infected will show severe symptoms, and 5% of people get severe symptoms, and among these 5, 20% of the people will die because of the severe symptom. And the severe symptom is called like a hemorrhage and cause the multi-organ failure. Why the severe symptom happen? Because there's a non-specific immune response and attack multiple organs to generate the hemorrhage response. So in order to show or diagnostic the severe symptom in the dengue, we want to generate the protein microarray to show the cross-reactivity of the antibody and to quantify the severe response. So in here, we generate a protein microarray cover all the family virus that can infect humans. And we do a duplicate and show the R-squared with 0.96. And the limit of detection is around 172 picogram. And on the array, we can show the serotype specific antibody, such as anti-serotype NS1 and only response to serotype 1, etc. So on the array, we want to ask what's the immune response difference in the different severity. So we collect the healthy control, mild symptom, hospitalized non-severe, or hospitalized severe. These two difference is because some patient will hospitalized, but they come out, they recover after 1 week, become non-severe cases. And some is the cases is deteriorate very fast and they administrate to the ICU. So this is called hospitalized severe. So in these 4 different groups, we can see in the human IgG, the more severe case like a group C show the more cross-reactivity across all the flavivirus proteins. So in the quantification, we also demonstrate this C group is generate high response to the cross-reactivity. And I want to emphasize the time of the sample collection, because those patients are collected in the day 2 of the fever. So it's in a very early stage before they administrate to the hospital. So this will be as an early predictor after they will become into a hospital severe or not. So same here with different flavivirus, even not the dengue virus, they will generate cross-reactivity in the group C. And in other study, we focus on the different phase of the fembrile phase or convalescent phase. And we find out in the fembrile phase, we can show the cross-reactivity. Means in the very early stage of the fever, we can show the cross-reactivity as an early predictor for the severe symptom. So they are all published last year. And the last part I want to talk about is about the human GPCR protein microarray. G-protein coupled receptor abbreviation as GPCR is one of the most important receptor in humans. It accounts 40% of current FDA-approved drugs target to the GPCR. And GPCR involve your hearing, your eyesight, your neuron transmission, inflammation, or cytokine chemotaxis. They are all using the GPCR as a sensor. That's how important they are. However, GPCR is a 7 transmembrane protein receptor. It's hard to study because it's required special way to purify it. So it needs a way to purify it because it's a transmembrane receptors. Also, because they're diverse transmembrane domain, their ligand is very diverse, and from the small molecule like ATP photon to big molecule like protein ligands. So because it's too diverse, 25% of human GPCR are still classified as an orphan GPCR without any known ligands. So to facilitate the study of the human GPCR, we build a very special technology. It's called virus display technology. By using the HSV1 is a human herpes simplex virus. It have an envelope. When it in the life cycle of HSV1, you will grab a piece of the Golgi membrane to form the lipid envelope. So we use this to carry our GPCR on the virus envelope, and we clone every genes into the HSV1 vector and produce the virus carry the GPCR and print it on the array. And we cover 85% of the human GPCR on the array to form the largest GPCR array in the world. And first, we want to demonstrate the specificity of the biologicals, like antibodies. Many antibodies are antibody drugs are targeted to the GPCR. So we use some CXCR2 antibody to show the CXCR2 antibody is really specific, only bind to the CXCR2 target. But there are some antibodies not that specific, such as ACKR3. They bind to other 2 targets. So it's called off targets. And some antibodies totally fail. They generate weak signal to the target. And later on, we find out only half of the antibody commercial available are good and really specific to the target. And next, we demonstrate the ligand specificity on the array by using the opioid receptor using dynorphin A as a ligand, which shows the specific binding to the opioid receptor 1. However, there's a non-specific ligand called somatostatin releasing factor 14. It should bind to the SSTR2, but the top ranked receptor is GABA B2. So this, we use some other methods to demonstrate GABA B2 also bind to the SRIF14. Although we show the downstream signal is not turned on, but it's still bind to the GABA B2. So the last part I want to show is the demonstration in the infectious disease. Because GPCR is a rapid turnover receptors, once it activates, it will internalize. But if the GPCR being a door for the pathogens, once it bind to the GPCR and internalize and invade the cells, that's our hypothesis. So we use a group B strep. It's a famous neonatal meningitis bacteria, and we label it with fluorescence and incubate with the GPCR array. And we find out they actually bind to several specific GPCRs, such as GPR101, GPR148, CYSLTR1, etc. And those receptors are either orphan GPCR, only with number of orphan GPCR, we don't know how to study it. But there's one GPCR is very interesting. It's called cystin leukotriene receptor 1, because it have a FDA-approved drugs specifically target to the receptor. So we first validate this with a drug called montelukast. This drug is for the asthma treatment. And this drug can dose-dependent block the GBS, group B strep invasion. And with this drug administered to the mice, even with the group B strep existence, it will block the meningitis in the brain. So this is my summary with the GPCR microarray. We can screening for highly specific biologicals, screening for ligands, and screening for pathogen interaction receptors. So these are all my related publications. And I would like to thank all my groups from the pictures. And one of the students called Batu is from Turkey. So he did a nice job in my lab. Thank you.
Thank you very much. This fascinating talk, and it is also very convenient for this current time. As I told previously, we are living with COVID-19, and we will live. It will be in our life like a flu virus. So your experiments are very important to give to lighten the current situation, and also to create new treatment modalities. And I would like to look at a question part. Yes, there's a question.
Yeah, I see like two questions.
Two questions.
So
yeah.
One is about the challenge of the cross-reactivity between antibody targeted different pathogens, and what strategy you use to employ the ensure specificity.
Dear Professor, maybe it will be good to read to audience
accuracy
and
of
to you. Question from Professor Aslı. Professor Guan, in developing multiplex immunoassays for monitoring humoral immunity, post-viral infections, how do you navigate the challenge of cross-reactivity between antibodies targeting different pathogens, and what strategies do you employ to ensure specificity and accuracy in detecting immune responses?
Yes, this is a very good question. So in a protein microarray, the beautiful of it is because it's used spatial to differentiate different proteins. So they usually generate very low cross-reactivity between spots. So if you are asking about the different protein we immobilize, usually they don't cross-react to each other. We have tested before. So, but the point of our study is to show the cross-reactivity between different pathogens. And in these pathogens, because they have similarity in the protein, in the different level of similarity. So because in most immunological disease, because such as a dengue, if the severe case is due to the loss of specificity of the antibody, so they can attack multi-organ or generate a very enhanced immune response. So in order to quantify the non-specificity of the immune response, we need a multiple antigen to quantify the cross-reactivity. That's the use of the protein microarray to show the cross-reactivity between the similar proteins. Hope this can answer your question. So the other question is about the aptamers. Yes, we do a lot of aptamers on the array, which we don't have time to show it. So we also generate some aptamer to neutralize the SARS-CoV-2. But to be frank, aptamer is really hard to put into the clinical use because it have a charge issue and there's also a specificity issue. So we rarely can have an aptamer as specific as antibody. So that's why all the approved drugs are antibody rather than aptamers. That's the current situations.
Thank you very much. Let's look. Yes, okay. Yes, thank you very much, dear Professor.
Thank you.
Nice to meet you and nice to know you. And we will continue with another speaker. Next talk will be given by Associate Professor Dr. Frank Hernandez from Linköping University, and he will give talk about engineering of oligonucleotides for developing diagnostic and therapeutic strategies. We would like to give brief information about his history. Yes, please. And he got his bachelor degree from microbiology and clinical laboratory in 2003 from University of Antioquia, Colombia. I'm sorry for my pronunciation, dear Professor Hernandez. Thank you very much for your patience for my pronunciation. And he got his PhD degree about bioengineering and chemical and process engineering at 2008 from University Rovira Virgili, Spain. Yes, please. When we look at his academic career, he got 2 postdocs and about nucleic acid-based technologies and cancer and infectious diseases. And he's researcher at University of Basque Country and Biodonostia Health Research Institute in Spain about diagnosis of cancer using hybridization systems based on nucleic acids. And he got his assistant professor degree at Department of Physics, Chemistry and Biology, Linköping University, Sweden. And at last he got his associate professor degree title again at Linköping University, Sweden, at Department of Physics, Chemistry and Biology at 2022. Yes, please.
Okay, thank you very much for.
I'm sorry, I haven't finished yet. Excuse me.
Okay.
You have also administrative duties. We cannot skip this. And you are Chief Scientific Officer at Teredia, Sweden, about biotechnology research, and it is still continued. And you are also group leader, Linköping University, Sweden, at Nucleic Acid Technologies Lab, and it was started in 2020. It still continues. And when we look at your field of research, you have several publications and experiment about therapeutic approaches, advanced drug delivery systems, as that is oligonucleotides and toucan technology, and personalized antibiotics and chemotherapeutics. And when we look at the research activity, Ash index 18, and you have over 1,236 stations. Of course, it doesn't reflect your capability and your wonderful experiences. Yes, please. Yes, we are welcoming you to our symposium, and we are waiting for your speech. Thank you very much.
Okay, thank you for this nice presentation. I'm really happy to be here. I'm trying to set out my. Can you see my screen? That
Yes, Professor, but I request you to make it full screen, please.
Yeah, it's like it's not working before.
Right now we cannot see, but here, if it is possible to make full screen, it will be okay, Professor.
No, it was that. Yeah, now.
Yes.
Okay. So sorry for the technical situation. First of all, thank you very much for the invitation. It's a real pleasure to be here. For me, Turkey is a very nice country in so many ways. I really like the places, the food, but most importantly for me is that I have great collaborators there, and you're gonna see during my presentation. So thank you for the organizers for this invitation, and I will go immediately to my presentation. So today I will talk about how we can do diagnosis and therapy using some enzymes that call nucleases. And I always like to start with this slide, a small animation in this slide, because this is exactly what I'm doing in my lab. In blue, you can see what is a nuclease, which is an enzyme that can degrade oligonucleotides. And this molecule that is coming and be cleaved by the nuclease is a short oligonucleotide. And actually, this is what I'm doing. This is my work, is try to design a specific sequence that can target these nucleases. Going into the specifics of this technology, I would like to describe briefly, what is important and how we can do, or how we can make this technology to work. So, with the idea of the previous slide, so in this for this presentation, I'm gonna use bacteria as an example. Imagine that you want to develop a specific oligonucleotide for a bacteria. What we need to identify is an oligonucleotide that can be cleaved by that specific bacteria, but at the same time, the same oligonucleotide needs to be stable for any human condition. Like the previous speaker mentioned about aptamers, that is difficult. They are difficult because sometimes they are not stable. But in our technology, we are making them very stable to be able to be used in the clinic. So, but not only this, we also need to incorporate some detection mechanism to these oligos to be able to see the cleavage when the nuclease is cutting. So to do so, we have an oligonucleotide here that is modified with a fluorophore here, a dye, and also a quencher. When they have in this conformation, the quencher eliminates or blocks the signal of the fluorophore. But when the oligo recognizes the nuclease, what is happening is that this break the oligo, the fluorophore part diffuses away and recover the energy. So here, or the emission of the light. So in this presentation, we're gonna associate the fluorescent signal with increase in nuclease activity. But this kind of system has been there for a long time. What is new, what we're doing that make our technology different is that we modify these oligonucleotide sequences with specific chemical modifications. And these chemical modifications provide a specificity to the oligos that we are selecting here. So in the case of bacteria, for example, what we are doing is trying to culture a specific bacteria. In this case, I'm going to refer to Staphylococcus aureus, where you can culture in the lab. After culturing, we expose these specific bacteria to different sets of library sequences that we have with different kind of chemistries and sequences. And in that way, step by step, we can identify what is the right sequence that we can use for detection and therapeutics. So here, I don't have time to describe the entire process, but I will tell you the story about one of the successful sequences that we have, and it's this one that can identify Staphylococcus aureus, which is a bacteria with a high clinical importance. And what you can see here is that our oligo was able to target a specific infection. So to start the experiment, I need to describe that the bacteria that we used was genetically modified with a luxoperon that produced light, luminescence, and we can monitor that the bacteria is alive and is metabolically active. Here in this picture, you can see that we injected the bacteria in the muscle, and after the injection, after 24 hours, we were able to see the infection localized exactly in the inoculation point. But what is interesting here is that here you can see our fluorescence channel, what can measure if the oligo is activated or not. Here you can see before injecting our probe, and here you can see after injecting our oligonucleotide systemically, right, tail vein. And you can see that it starts to accumulate very well up to 45 minutes. This experiment was really important because it demonstrates two important factors. The first one is that our molecule was able to go all over the place looking for the infection. And the second is that has the capability to accumulate in the area of the infection. With this experiment, we were able to demonstrate that this technology is ready to try to find implementation in the clinic, for example. Before going into the main topic of this talk, I will tell you that using this oligonucleotide, we have identified, like I previously mentioned, a different kind of biosensor using fluorescent detection, but also we can modify our detection to MRI, for example. Fluorescence is a very useful technique, but it has two important limitations: the tissue penetration and the background signal. So we believe that if we can identify other detection mechanisms, the translation of this technology into the clinic will be easier. Here you can see how we modify two contrast agents, and we were able to demonstrate that we are specifically detecting the presence of the Staph aureus using magnetic resonance imaging. It's a technique that is implemented in the clinic, and in the future we believe that we can develop probes for diagnosis using MRI. But also there are other detection systems that are used for screening, for example, like a lateral flow assay that in the pandemic we realized that was very useful, something they use also for as a pregnancy test. And we were able to incorporate our system into this kind of detection here, detecting Staph aureus as well. So with this, we just want to show the flexibility of oligonucleotides to be incorporated in different detection systems. The other aspect or another detection that we have reported is using electrochemistry. Actually, this is in collaboration with one of the professors, Murat Bakrop in university, when we were able to demonstrate that for the first time that we can measure nuclease activity associated with electrochemical transduction. And with all this evidence, we believe that nuclease activity can be targeted with the oligo that we're generating, and we can provide different kind of solutions using several transduction mechanisms. But to follow up and concentrate on everything that I would like to tell you about therapeutics, and coming back to the previous experiment that I presented to you, Here you have our full experiment of what we did previously in vivo. So I want to tell you that if you don't have any infection and you inject our prop, what is going to happen is that you're not going to see any activation of the prop. To complete this experiment, we also eliminate the quencher that it was there blocking the fluorophore. And here you can see how the fluorophore can go everywhere, right? Like I mentioned before, a GPS that is going and identifying the place of the infection. Here you can see the image that I described just before, when we can see a specific targeting using our oligonucleotide for Staphylococcus aureus. And here we have a very important control, which is we knock down the gene that produces the nuclease of Staph aureus. So there is no nuclease there, and what you can see is that if there is no nucleases, the signal is not really there, indicating that we were able to identify one nuclease and associate that nuclease activity with one bacteria. So with this in mind, and we want to translate this into diagnostics, into therapeutics, our idea was, okay, now we know how to go to the place of the infection. What if we add a drug and we try to kill the bacteria? And that's this is how the Toucan project comes alive. So Toucan is basically an oligonucleotide that can recognize a bacteria and can transport a drug. Now that you have this little background about nucleases and oligonucleotides, you can understand that Toucan stands for therapeutic oligonucleotides activated by nucleases, right? Obviously, I'm not talking about this beautiful bird toucan. My toucan looks more like this, right? And what is important of this structure is that we have one area that provides protection, right? This molecule is not degraded in blood because we have this area that protects from any non-specific degradation. We also have a region that can identify the bacteria by the nuclease, in this case, that is this area in yellow. And we have one area that is the drug. In our case, it's a nucleoside analog that facilitates the synthesis of this compound. So here, I will give you an idea how we envision that technology is going to work. I will tell you step by step what's going on, but this is how we envision that this technology is going to be applied in the clinic in the coming years. Start. So Toucan technology will be indicated for localized infections such as osteomyelitis, endocarditis, or soft tissue infections. What is happening is that when an infection starts, there is this abscess formation where the bacteria is growing, but at the same time is secreting these nucleases. These are our biomarkers. Toucans will be administered systemically to the patient, and we go all the body will be resistant to any human nuclease because of this black region protection, but specific for nucleases from Staph aureus, which will degrade the molecule, release the drug will go inside the bacteria and will kill it. And this is the way that we expect to cure infections in the future. The good news about this is that it's happening. And actually, the main researcher that did this job is a Turkish postdoc in my lab, Baris Borsa, very talented researcher. And we were able to demonstrate that what I just, it was shown in the video, is possible. So here you have an infection after 24 hours. You can see how the infection manifests with different symptoms of inflammation and the redness around the infection. Here you can see this group of mice were injected with PBS, placebo. And here you can see our toucan, which was able to cure almost 100% the infection after 24 hours. That's a fantastic result because, again, we were able to demonstrate that we can inject something systemically, go to the area of the infection, and kill the bacteria. We also used the drug alone, but we didn't see any improvement compared to the initial placebo, indicating that toucan provides this accumulation of the drug in the specific tissue that is infected and provide a therapeutic effect. So here we just validate our model using here you can see by histopathology where you can see how it's an uninfected that we use the left leg of the mice as an infected. You can see how the tissue looks like. Here in the placebo, we can see infiltration, inflammation around, markers of neutrophils infiltration and everything. And you can see how, with our toucan treatment, the lesion was reduced and almost controlled. With the drug alone, we didn't see any improvement, like I mentioned before. And importantly is that we quantify how much bacteria was left there. And what I can tell you is that we were able to reduce 90%, 92% of the bacteria with our treatment, which is a great result when you compare with the standard antibiotic that is going to be around 80, 88 in the case of vancomycin, for example. But we also measure the immunological response where we measure the amount of neutrophils, the percentage of neutrophils there. And you can see here that for our toucan system, it was very close to the uninfected mice, indicating that the immunological response was reduced because the infection was controlled. And we have a significant difference with the other two groups. So all this indicating that this technology has the capability to deliver a specific drug to where it's really needed. And that's why we believe that we have a great opportunity to translate this into the clinic. But to demonstrate this even further, we have we were asking ourselves what would be the limits of this technology, and we were trying to do this dose response experiment that they are needed to translate this kind of technology into clinical trials. So now what we know now is that using 10 times dose of what it was used in the previous experiment that I showed you, that was around 92% performance, we were able to reach even higher, 99% elimination of bacteria. This is something really, really important for our technology, since we didn't see any antibiotic performing at the same rate. So indicating that we have features that can be very useful for clinical implementation. So, but when we talk about, I'm telling you about one of the bacteria that we can target, that is the virus, but there are many other bacteria out there that they are very dangerous and need to be treated as well, need to be identified and try to find the way to cure this kind of infections. So in my lab, we are focusing on the escape pathogen, which are the most dangerous pathogens, and we have identifying probes that can target any different degree. So another opportunity that we have is not only target only the bacteria, we can also target the immune system. And this is something that is under evaluation in my lab. And another application of these probes would be, how is the tumor microenvironment? The tumor microenvironment is very complex. We know that it involves the response of the immune system. And in a similar way that we could do it with bacteria at some point, we are planning to translate this into cancer therapeutics. In this case, we are just at the beginning, but we have very promising data indicating that we can identify tumor tissue from healthy tissue. This opens the avenue for us to develop toucans, incorporating a drug, and imagine in the future chemotherapeutic treatment without side effects. So that's going to be the goal of this research lab. And with all this, and I know that time is a little bit limited right now, I would like to finish here, and I will open and happy to answer any question. These are the founders and the people in my lab working to make all this possible. And thank you very much for the invitation and your attention.
Thank you, Professor, for your presentation. I think that Nina Hoca is here. Yes, she's here.
Yes, of course,
Yes.
I'm here. Thank you very much. Wonderful presentation, again, fascinating presentation. And I would like to ask audience, do you have any questions to Dr. Professor Hernandez? Yes, this one, I think. Dr. Armani, the last one? Okay.
Yes, but some, it's like an anonym. Some do not write the students their name. Maybe they forget to write that.
Oh, yes, okay. Please let me read for you, Professor Hernandez. And considering the rapid advancements in genome editing technologies like CRISPR-Cas systems, how do you foresee the integration of engineered oligonucleotides in precision medicine approaches? And what are the key challenges and opportunities in harnessing these tools for targeted manipulation of gene expression or genetic variants implicated in disease pathology? Very long question. Thank you very much.
It's okay. I mean, this is a great question, actually. I mean, CRISPR is another nuclease. It's a nuclease. It's editing DNA. It's the definition of a nuclease. The thing here is that we use the other way around. CRISPR, the target is go to a specific area of the genome and make the addition there, right? So the target is the oligonucleotide. Our technology is the other way around. We target the nucleases that they are associated with the disease, right? In our case, it's easier to use our oligos directly in blood because they're going to identify the nucleus and they're going to release the drug at the moment of it. So what we have is a drug delivery system using nucleases as activators of our drugs or prodrugs. CRISPR, on the other hand, everybody knows, is a gene editing machine system, which is amazing, but we have the problem of delivery. Right? So deliver a specific CRISPRs to a specific cell is still a problem that is not solved yet. We will need a genius that is going to do it for us at some point, but we don't have the capability to do that. We can get cells, for example, and we can do blood, we can modify blood cells or something like this, but targeting is still a very difficult issue. So how we can integrate it? I mean, this is a great question. It's something that we are planning to explore in the future. We believe that our technology can target right now. We demonstrate that we can transport one nucleotide, right? I mean, this is enough for us and it's doing the job. So we need to start for something that is. But in the future, we believe that we can transport bigger molecules. And bigger molecules, we're talking about 23, 22. That is going to be the size of the antisense oligonucleotides. So there is a possibility there. Obviously, for us, is another avenue of development that we have in mind. But right now, we don't have a clear evidence that we can do that. It's on the way, but we are not sure that we can do it at this moment. It's one of the things that is in our mind.
Okay, thank you very much. We have another question from
yes.
Professor Dr. Çetin is asking, as the field of nucleic acid therapeutics continues to evolve, how do you envision overcoming the barriers associated with systemic delivery of target effects and immunogenicity to ensure the clinical translation and widespread adoption of oligonucleotide-based therapies, particularly in the context of treating chronic or genetically complex disorders?
Another great question. So I agree. I mean, nucleic acid therapeutics is advancing. What we offer here is a different view of how to use oligonucleotides, not as therapeutics, but as drug delivery systems. We are focusing on cancer and infectious diseases at the beginning, but autoimmune disorders is something that really is associated with nucleases, and we have a really interest in that field. By the way, if anyone is working on autoimmune diseases, just contact me. I'm happy to talk about it. So in the area of chronic disorders, I mean, like I said, right now we are able to deliver small molecules. So we believe that we're going to get there, but right now we don't have the tools in place to do this kind of work. But the topics are exciting, and I think that with time and a little bit of support, we can get there at some point.
Thank you very much. Thank you very much for sharing your experiments and results and with us. Kind of you. Nice to meet you. And we wish to meet you again at future time. Thank you very much.
Thank you very much. Have a good day.
Okay. Yes. We would like to welcome another speaker, Associate Professor Dr. Nina PEREZA from University of Rijeka, and she will give talk entitled "Is there a place for developing educator competencies in biomedical sciences?" I would like to give brief information about the history of Professor PEREZA. And Professor PEREZA got her MD degree from University of Rijeka, Croatia, Faculty of Medicine at 2009, and she got SPC title again at same university, and also she got a PhD title from medical biology and genetics again at can you please thank you from University of Rijeka, Croatia. Yes, please. And when we look at her academic career, she got assistant professor title from Faculty of Medicine, again University of Rijeka, Croatia, and at last she got the title of associate professor doctor again at same university. Yes, please. She has also administrative duties. She is co-founder and head of the Center of Genetic Education, the Center of Improving Teacher Competences and Communication Skills, again at the same university. And she got awards and honors, and she got 2 awards. One of them is the best young scientist of the medical faculty in Rijeka. I'm sorry for that pronunciation. And another award is Teaching Excellence at the University of Rijeka. And when we look at the field of her research, we are seeing that she focused on medical biology and genetics of human reproduction. You know, I'm also a clinical embryologist, their professor, and genetic education and medical education. And when we look at her research activity, she has H index 16, I10 index 19, and over 701 stations. Of course, it doesn't reflect her potency and experiences. Yes, our pleasure to invite for her talk Associate Professor Dr. Nina PEREZA. Thank you very much. Dear Professor, we cannot hear you. And also we cannot see your screen. We need your help. Right now, we are seeing your screen, but I request you to make it full screen, please. And still, we cannot hear you. Okay. Your audio is blocked. Let's see. Okay, please just go to your microphone, and at the right side you have one arrow. Please choose it, and when you choose it you will have menu, and please choose again microphone, and afterwards do the same thing, and please go to this menu, and please test microphone by using the same menu. And one not working. What about the proposal from Dr. Guan? He requested you to choose. Okay, okay. It is also my proposal, another proposal. Yes, please. You know, in Turkey, we tell this negative energy. You know, we have also sample eyes in order to prevent negative energy coming to a person or the environment.
Well, hello, and thank you for this wonderful introduction. I still have to share my screen with you. Let's
You
do
are
that.
looking great. You are looking great.
Oh, thank you.
I'm also
I'm under stress now, but okay. So
I'm also colleague with Professor Spandidos, and I also came to Croatia to give speech.
It's okay. It's okay. The important thing is that we've worked everything out now, and I hope that you can hear me, and see my presentation.
Yes, yes. Wonderful.
Okay, great. So again, thank you so much for having me. I'm very privileged to be here. And as you mentioned, I'm very attached to my university, the Faculty of Medicine in Rijeka, University of Rijeka. I'm actually in a dual role because I'm both a medical doctor and a medical geneticist, but I'm also specifically trained in the methodology of medical education. And when Professor Armani asked me to join this symposium, the first thing that came to my mind was like, is there a place for this topic in this symposium? And I think that the title of this presentation kind of imposed itself on its own. So I would like to talk about whether there is a place for developing educator competencies in biomedical sciences from the position of both a medical geneticist and the head of our Center for Developing Teacher Competencies and Communication Skills. And this is a question that I would like to address from three perspectives, and I would like to give you an overview of the results and experiences of the Faculty of Medicine in Rijeka. So first and foremost, I would like to show you the answers that our students and medical educators provide to this question. So let's see their actual needs or the needs of our target groups. And then from our perspectives as experts in the field, I would also like to talk about what we think about this question. And finally, from the perspective of faculty management, do we have the support of decision makers for making this question or the answers to this question work? So let's start with
Professor.
Yes.
I'm sorry to cut your speech, but we have a request from you. Can you please make it full screen, please?
It is full screen.
I don't know. It is not.
Well, to me it is.
Okay, we will continue like this. You can continue. Thank you very much.
Do you see it's not full screen?
No, it is not. We are seeing slides, and we are seeing only one slide, and the slide menu at the right, at the left. So because of this, I requested you to make it full screen for us.
Do you see?
Yes, wonderful. Thank you very much.
All right, so I changed the settings now. Okay, so let's start with probably the most boring definition about what teacher competences are. So or educator competences. So these are the sets of knowledge, skills, and values, attitudes that we need to kind of conduct the role of a teacher in the healthcare professions, or what we call a medical educator. Of course, there are different categories and different levels of these competences, and some of the most basic categories include the planning of teaching and learning, selecting learning methods, and so on. And when we talk about students, or I don't know if you have the same experience, we probably do, because students across nations have the same, what we call subjective challenges in medical education, and they'll usually tell you that, well, lectures are not interesting, I don't know where to learn from, there's too much information that I need to learn. So but what does this actually mean? So this is kind of a conversational challenge that we hear in medical education. And from the perspective of teachers, again, I could use the same images and say that, well, students are not interested in my lectures, they don't know enough. And if we talk about specifically this symposium and the topic of science, well, what we usually hear from our university teachers is that, well, if I dedicate my time to education, I've got less time for science. So to kind of surpass the subjectivity of challenges, we tried to objectify challenges in medical education. So we conducted two different scientific research from the perspective of students and also from the perspective of our teachers. So in June 2022, we did a large research on students of all 6 years at our study of medicine about the quality and structure of teacher competences and medical studies in general. And also in continuity since January 2023, we conduct similar research on our medical educators or teachers that actually come into our program for developing teacher competences. So we ask them before and after they finish education, what is their kind of self-reflection on a set of and a number of teacher competences in the general field of medical education methodology. And I'll show you some of the most interesting results. So first and foremost, we asked students to say how important is, so starting from the basics, so how important is the course program or the syllabus to you to successfully navigate a course? And of course, the average grade is like really high. But when we ask them to assess the quality of currently available syllabi, of course, the grade is much, much lower. But the fact is, when you ask teachers, whether they be basic teachers or clinical teachers, how competent they feel in writing a syllabus, the grade is even lower. So they are aware of the fact that they do not have education in this field. Also, we asked students to kind of assess how much the aims and learning outcomes are aligned with course contents, and also do teachers explain the aims and learning outcomes at the beginning of a teaching unit. And of course, you can see that the grades say everything, no further explanation is needed. And of course, teachers actually agree with students, so they do not feel competent enough to design and write aims or learning outcomes and contents for courses and teaching units. And also, what you can often hear from students is that lectures and seminars feel the same. So we ask them to assess how much does the concept of lectures and seminars different in methodology currently. Of course, the grade is not that great, so it's 2.8. And of course, teachers also agree with students because they do not feel competent enough to select learning methods according to learning outcomes of a teaching unit, nor do they know how to use the appropriate learning methods in medical education. We also asked students to determine the order of importance of the following components for successful medical education, and of the four, the feedback, participation, individualization, and relevance of content. I don't know which one you would choose, but our students, of course, chose that relevance of contents for future profession is like the most important component for successful medical education. But then we asked them to assess the current situation. So how much are the aims, learning outcomes, and contents of courses aligned with the actual needs for future profession of students? And here the grade is again very, very low. And of course, teachers also agree that they are not competent. So it's around 3. The grade is around 3. They don't feel competent enough to actually design a course or a teaching unit that would reflect the authenticity of medical education. And also something that we usually talk about, again, subjectively, we try to objectify this. So we ask students to assess the current level of interactivity in lectures, seminars, and practicals. And I'd say that the grades are really, really low for all 3 components or all 3 teaching units, especially for lectures 1.96, and even for practicals, having a grade of 4 is not that great. On the other hand, teachers assess themselves really high. So when we ask them, do they feel competent enough to establish interaction with students during classes, they actually made a grade of 4, so the average grade of 4, which can be simply explained by the fact that us teachers probably perceive interaction as, how are you feeling today? Did you prepare for class? So we kind of see interaction as a basic communication skill, whereas students see interaction as a way to facilitate their learning. So our perspectives on what interactivity in the teaching process is doesn't match students and teachers. So we also ask students to, when we talk about clinical teaching, we ask them how much are different teachers unified in their teaching of clinical skills in the same course. And I think you might be surprised with the results. So it's very, very low, meaning that in the same course, different teachers teach clinical skills in quite different ways. And also we ask students to assess how much different teachers have unified instruction for patient presentation in the same course. The grade is again really, really low. And surprisingly, teachers, when asked to assess their capabilities of teaching clinical skills, actually give like the entire grade above what students graded them, meaning that probably because perhaps, for example, I do perform clinical skills every day in my practice, and I kind of think, okay, if I know how to perform a clinical skill, that automatically means that I know how to teach someone to perform that clinical skill, which is not always the case. And you can see huge discrepancies between students and teachers in this point. Also, we ask students to assess the current level of presence of case-based learning in medical education and clinical reasoning. Again, the grade is quite low, and teachers don't really agree with students, again, because we perform clinical reasoning every day in our practice. We know it, and probably our skill is like really high, but having a developed skill in us teachers doesn't necessarily and automatically mean that we can actually teach someone how to perform that skill in a methodological way. And finally, the final question before I move on to the second part of my lecture is, well, assessment is again quite is graded on a quite low level. So we ask students to see or to tell us whether teachers are unified in assessment criteria for the forms of assessment in which multiple teachers are involved, meaning that different teachers have different criteria and ask different questions. It's graded quite low, but teachers again here agree with students that they are not kind of educated in performing what is especially important today in medical education, those structured methods of oral assessment. Well, someone might say, now that I've presented these results, someone might say, why is she presenting this? This is kind of a weakness of their faculty. But I would actually say that this is currently perhaps not weakness, but actually a reflection of an actual situation, and that we can only turn it into our strength by admitting to ourselves, okay, we've got areas where we could do better. Now that we know where we can do better, we can actually try and fix it. So today at the Faculty of Medicine in Rijeka, we see this as our strength because now we know what our actual needs are. So in the second part of my lecture, I will show you how we use our current weaknesses and turn it into our strengths. So from the perspective of us as experts in medical education, I would like to show you how we solve the problems that I addressed in the first part of my lecture. So briefly about the Faculty of Medicine in Rijeka. So we were founded in 1955. We are the oldest and largest constituent of the University of Rijeka, and we are one of 5 medical faculties in the Republic of Croatia, of course, dedicated to educating healthcare professionals. And if you think about yourself as a healthcare professional, the truth is that we all perform simultaneously 3 roles. For example, I'm a medical doctor. I'm educated as a medical geneticist, and I had like a structured professional education to become a medical doctor and a geneticist. Also, I had education to become a scientist and to have my doctoral degree, but unfortunately I'm also not unfortunately, luckily I'm also an educator, but unfortunately I had no opportunities for an official education during my studies or even postgraduate studies. Basically, to become a medical educator, you depend mostly on lifelong learning, which is often unsystematic and unstructured. There's a nice exception in the European Union, and it is our Faculty of Medicine in Rijeka. So we have separate organizational unit, which is called the Center for Improving Teacher Competencies and Communication Skills, which was founded in 2017 with the aim of providing systematic support of the highest quality for the professional development of teacher in the methodology of medical education. And again, I'm really proud to say that we present rarity in the European Union, unlike the United States of America, where these centers, usually called centers for or offices for faculty development, are really present and highly represented. In the European Union, we are really one of the exceptions. I'll just walk you over or through some of the activities of our center and how we actually address professional development of our educators. So we perform many different activities, from educational activities to organization of scientific and professional meetings. We perform scientific activities because medical education is also a science. We publish a lot, and we perform many different other activities. Just briefly walk you through each of these activities, and we kind of divide our educational activities to pre-COVID and post-COVID era, because COVID kind of made a natural stop, I'd say, or a break between how we teach and learn before and after the pandemics. And these were just some of the educational programs that we offered before COVID, but we were faced with a lot of challenges because usually these programs were mostly on-site. They were full of lectures, so passive learning methods. You could not gain any skills. There was no mentoring, no individualization. And of course, during COVID, such way of teaching and learning spontaneously ceased. So we turned off all of these programs. And during 2021, we entered a phase of huge reformatting of our educational offer. And today we have a much wider educational offer, and we have several programs which are completely different in methodology and contents, and I would just like to show you some of them. So this is our basic program for improvement of teacher competencies or education competencies. It's called Modern and Practical Medical Education. It's mentored, it's individualized, and I'm proud to say that the current management actually recognized this field as really important for our faculty. And to finish this program, it's actually a mandatory condition for the promotion to the position of assistant professor. It is a very long program. It lasts for 4 months, but by the end of this program, having called it practical medical education, you actually have like applicable and actual products at the end of the program that you can use in everyday practice. So the program is very large. It has 3 thematic modules. It has 7 courses that will teach you about everything you need to know in medical education. And there's a second module that's really important, where we divide our basic teachers and clinical teachers, because they have different needs and these forms of teaching are quite different. This is a nice program because it is 50% on site and 50% online. And basically, especially for the clinical teachers, it is really difficult to, I don't know, listen to a course for 7 days, 8 hours a week. So all of our e-contents or contents for learning are present as e-courses. So you can take them whenever you want, wherever you want. And when you come on site, you actually do something useful for your own practice and kind of apply the knowledge that you learned online to the on-site practicals. And these are our first teachers that were promoted last year. So 11 of them today, they are mentors in our program, and they contributed significantly to educational materials. This year in July, we'll have 3 times more promoted teachers, and we'll have some other, we have promotion of teachers from other courses. So this is, we're really proud of our center and how it works at our faculty. And also because Modern and Practical Medical Education is a modular program, we are able to take out smaller satellite courses from the entire program. And this is our offer currently at the faculty. So both for teaching with clinical simulation and EMI, and a lot of different other courses that are present as satellite courses. Also, what is new this year, this May, as you can see here, so less than a month away, we've got another new course, which is called School in Medical Education. It's a 3-day course that we wished not to focus just on our faculty, but also everyone who is from Croatia and neighboring countries can come to this course. And it is a course that will be conducted on a wonderful island, because we are a Mediterranean country. We try also to promote our country in this way. So it will be on the island of Tres in a wonderful palace. And I'm really proud to say that our Croatian Medical Chamber always follows us as organizers and helps us promote all of our activities to medical doctors and other healthcare professionals in the Republic of Croatia. I'll just walk you briefly through our second activity, the organization of scientific and professional meetings. I'm proud to say that I actually met some of the professors from Istanbul AYDIN University when they came to our international MEDRI conference last year. We also have an internal symposium and a new professional symposium that will be taking place this May. I would just briefly remind you of the first international conference on teaching and learning in medical education that took place a year ago, and Professor Haki Dalcik from your university was here in Rijeka, and we had a wonderful time. This was a huge conference. I won't be going into many details why we organized it, but we were able to kind of gather all the deans of medical faculties in the Republic of Croatia and neighboring countries in this conference. And it was a huge conference. We had more than 1,700 participants from 11 countries, a huge number of student volunteers. So we're really proud. And next year we're going to have the second edition. And of course, I will invite you to participate in this conference again. Again, this is an internal symposium. It's Symposium on Medical Education that we perform every year at the Faculty of Medicine in Rijeka, and we try to introduce innovative teaching and learning methods in this symposium. It's very popular, so hopefully this is also something that we will continue to do. And another satellite symposium that's new that will start this May. We have a lot of scientific activities. We participated in some of the EU-funded projects. We also applied for an Erasmus project this year. Currently, we have a smaller scientific project. We publish a lot. We publish a book of abstracts. This is kind of, it was a huge project. I kind of showed it here because it took us a lot of time and a lot of people were involved. So we're really proud of this. And also we always try to kind of gather all of the stakeholders of medical education, not just in Croatia, but in our neighboring European countries. And this was just one thematic issue of a journal here in Croatia about the challenges and the future of medical education in the Republic of Croatia. We have a lot of other activities. I won't be bothering you with these, but this is kind of a sum up of all of our activities. We are a very active center, a very progressive center, and I think that we are very, very proud of what we do, and we always say that we are proud that we are from Medri. And finally, as a conclusion, I would like to show you how important faculty management is in all of these processes. And the support that we have from our decision makers is crucial for everything that we do. So if we were to answer the question, is there a place for developing educator competencies in biomedical sciences? I will tell you that, of course, there is, but you need to have the support of the management. And this is our dean, who is our major promoter, I would say, and the person who actually blocked all of the resistance probably that we had a couple of years ago. So as I showed you previously, because we are a university, we have three roles. So we are scientists, we're professionals and educators. But as you might know, the scientific way is always kind of more like, I want to do science, I want to do less teaching, but I think that management is crucial for you to be able to push through all of these projects. And I think that positive PR is the key to show all stakeholders of medical education that this is not something that's terrible, huge pressure, but something that we need and something that is essential, because first and foremost, the quality of the healthcare system directly depends on the quality of medical education. So this is one of the rare fields, even if you do scientific research, the results of such scientific research in medical education are directly translatable to practice. So be it in basic sciences or in clinical sciences, so basic teaching and clinical teaching. Second, I'd like to remind you that our studies in most healthcare professions, undergraduate and graduate studies, do not offer us the possibilities of mandatory training for the role of a medical educator. And consequently, why you need management support is that if you want to teach someone how to teach, you need separate institutional strategies which will be aligned with the vision and mission of faculty development, both quality and the restore of teaching. And finally, all of this can be conducted only through separate structural units. And it is also important to say that it is crucial to be at the right time and at the right place with the right people, having support from the management, having professionals who can conduct the programs, and having teachers and students who are motivated enough to join you in all of these processes. And finally, what's really important is that this is especially relevant after COVID. The change in the way students learn must accompany the modernization of teaching in teachers, because the way I learned medical education has nothing to do with the way students now learn medical education, but it is not up to them to kind of adapt to my way, how I was learning. It is up to me to adapt my teaching to how they learn today. So finally, I'd just like to show you that all of this makes sense, because if you might remember from the beginning when I showed you how teachers feel about their level of teacher competences before they entered our program for improvement of teaching competences. After they finish the program, the grades rise from 1, 2, or even 3, which shows that education 4 months long can actually change and transform how you feel and how you do your teaching and how you do medical education. Of course, not everything can be put in numbers, There are some descriptional results at our faculty that are really, really important. After all of our activities, we have the reform of several courses, modernization of courses. We have what's really, really kind of staggering, some of the departments that were really dedicated to basic science and still are and will always be dedicated to basic science, and who kind of had resistance towards medical education methodology, now introduced separate meetings about medical education. Of course, creation of new elective courses, a lot of teachers got their awards for teaching excellence, and of course we have mentors in our center, so all of these teachers kind of remain in our program. Of course, all of this is a very slow process. It will take us probably even more years to completely transform the system, and improving teacher competences on its own is usually not enough to transform the system. Curricular reform must also accompany all of these processes, and of course infrastructure is really, really important. And the final two slides, this is again a sum up of all our activities. We are, as Medical Faculty of Medicine in Rijeka, Europe, really, really proud of all that we do. And finally, I need to thank a lot of people, because people are responsible for all of these activities. Me on my own would never be able to provide all of this if these people were not here. First and foremost, I need to thank the person behind all of this, my greatest support and the greatest supporter of our center. So this is our Dean, Professor Hauser, and also all of these people who create and support our programs. And as I go along with all of the activities, I no longer use names, because it is impossible to write the names of all people involved in our activities. I just use organization committee, scientific committee, and so on. And thank you so much for your attention. I would be more than happy to answer your questions, and I hope that it is a topic that finally did align with your symposium. Thank you very much.
Thank you very much. You talked about the backstage of medicine. Backstage reflects front stage. So without right education, it's impossible to continue to maintain the quality, and you cannot help the patients. Right now you have two questions.
Okay.
I'm just looking. I will read for you. One of them is coming from research assistant Zeynep. And she is asking you, given the rapid pace of scientific advancement and the increasing emphasis on evidence-based teaching methodologies, what strategies do you recommend for continuous professional development among educators in biomedical sciences to ensure they remain abreast of cutting-edge research, pedagogical innovations, and best practices in student engagement and assessment?
Yes, thank you so much. This is such an important question. And I think that the best way to kind of achieve all of this is to have an organizational unit at the faculty that will provide for all of this while you do your scientific and professional work. So for example, having courses, short and long, that will enable you to stay up to date with the latest teaching and learning methods, and also to have the organizational unit that will have shorter professional meetings. So someone who will take care of the fact that you need to stay in touch with all of what Assistant Zeynep wrote. So I think that the key for all of this is having a professional organizational unit that will help you develop in this field. For example, I didn't mention, but we also provide individual support. So teachers write to me as the head of the center with specific questions, and if they need some support, for example, if they want me to see their syllabus, whether it's written in the correct way, we also provide individual support. So I'd say that this is kind of the answer to this. I hope that I answered the question.
Yes, I think yes. Another question coming from Professor Vildan. for Faculty of Education and Research Center asks from, as we strive to foster a culture of inclusivity and diversity in STEM education, how do you envision incorporating principles of equity, accessibility, and cultural responsiveness into the development of educator competencies, particularly in addressing disparities in representation and retention among underrepresented groups in biomedical sciences?
Well, this is also a very interesting question. I have to say that in Croatia, we don't see that many, we are very, I'd say, at our faculty, we don't see such cultural differences, which is unfortunate in one way. But I think that perhaps the best example that I can give is that at our faculty, we have a special interest group that deals with especially cultural responsiveness to differences or to, I'd say, addressing disparities in underrepresented groups. And this is an issue on its own. For example, if I talk about differences, the best thing that can cross my mind is, for example, we have medical studies in English, and we see a lot of foreign students coming to Croatia from all over Europe and also from all other parts of the world. And we offer, for example, special courses in our center regarding how to teach foreign students, not just improving English competences, but also how to address different cultural aspects with these, because they are different than our students. So I'd say probably special educational programs and, I don't know, popularization of addressing these disparities is probably the best way to address this. But as I say, unfortunately, in Croatia, we're still not that, I'd say, we don't see that many differences still.
Thank you very much for your talk. Nice to meet you and see you. Good day. Yes, we will continue with Associate Professor Dr. Parina from Tabriz University of Medical Sciences, Iran, Persia. And she will give talk entitled Hepatotoxicity due to herbal medications and dietary supplements. I would like to give brief information about her history. Yes, please. She got a bachelor degree from Faculty of Pharmacy at 2012, and from Tehran University of Medical Sciences, Iran. And he got PhD degree from pharmacognosy at 2015, again from Tabriz University of Medical Sciences, again Tabriz, Iran, in Tabriz, Iran. She got at last academic degree as Associate Professor Doctor. She got this degree from Faculty of Pharmacy, Department of Pharmacognosy at 2019, and of course continued again at Tabriz University of Medical Sciences. She has also administrative duties, and she's right now head of Faculty of Pharmacy, Department of Pharmacognosy, and it is newly started. It seems she is the head of Department of Pharmacognosy from 2023, again at Tabriz University of Medical Sciences, and she started to Tabriz University of Medical Sciences, Iran, in Drug Applied Research Center at 2015, and it still continues. Thank you very much. And when we look at the field of research, you can see that she focused on pharmacological sciences and pharmacognosy, and pharmacochemical analysis, biological activity of plants, biological studies, bioactivity, antioxidant activity, and natural product and drug discovery. Yes, she has also awards and honors. And when we look at this, in 2022, she got award that is top researcher of Tabriz Faculty of Pharmacy. I think that your university loves you very much. And when you look at the research activity, you can see that she has H index 19, I-10 index 32, and over 1,079 stations. And again, I would like to emphasize that these numbers don't reflect the activity of researcher. And I would like to invite Dr. Parina for her talk. Right now, I'm sharing her presentation for her, and I request you 1 minute for this.
Hi, dear professor. Can you hear me?
Yes, doctor. Yes. I'm sharing.
Okay.
I'm sharing. Don't worry, I'm sharing.
Okay, thank you. Hi, dear professors. Good noon. The presentation of all professors were very useful. My speech is being held during lunch and break time, and this responsibility burdens me to deliver my speech better.
No, it's okay. This delivery, this delivery can be seen. Okay, it is usual. And right now, we are looking forward to hearing from you. And yes, I opened your presentation, and I'm ready to continue with you.
Thank you so much. If you'll let me, I'll close my webcam, or it's okay.
It's okay. If you
Okay.
feel
Okay,
if
okay.
you feel relaxed when you close, okay, you can close, and I will continue. If you feel relaxed,
No, it's okay for me, because not only for a problem in connecting to the internet, I told you. If it's okay, it's okay for me. Yeah.
Yes, it's okay. You can close, dear Dr. Parina.
No, it's okay.
Yes.
Now.
No, we are seeing.
Close now. Now close or open?
No, now open.
Open. It's okay for me. No problem. It's
Okay.
open. Yeah, it is better open.
Yes.
Dear professor, do you hear me?
Yes.
Okay. Yeah. I'm very proud to be here. Yeah, I'm very proud to be here among the honorable professors from all over the world. And also, in the begin with, I would like to thank all of the professors who involved for coordinating this program. Unlike other professors, my speech is based on systematic review and meta-analysis studies. Now, my presentation is about, is it okay? The first slide, my presentation is about the hepatotoxicity due to the herbal medications and dietary supplements. Dear professor, if you lose my voice, please announce me and alert me to repeat my speech, okay?
Okay, that's right. Thank you.
Okay, thank you so much. In this presentation, we will talk about the 5 steps, and my presentation also is based on 2 review articles entitled Hepatotoxicity due to the herbal dietary supplements, past, present, and the future, which is published in the Elsevier journals. And the next one is about the hepatotoxic botanicals and evidence-based systematic review. Now, we will talk about in the 5 parts, introduction parts, frequency of the next one, please. Yeah.
Okay.
I bothered you now.
Again, you want next?
The next slide, please. Yeah. And the next one, yeah, the next one, I explained about this. Yeah, and the next, yeah. Yeah, in this part, we talk about the diagnosis, treatment, and specific hepatotoxicity. Now, we want to talk about the introduction part. Survey data have suggested that the use of herbal and dietary supplements, which we can call it as a HDS, range from 10% to over 75%, depending on the country. The most common reason reported for their use is for health improvement and health maintenance. Women, persons younger than age 65 years, and individuals higher annual household income have been reported to be more likely therapies. Completely free of unwanted side effects contains products have and lead to severe hepatotoxicity. Interact with one or other with the prescription medications. Now, the next one, please. It doesn't work. No, yeah, thank you. Over 1,000 medications and herbal products have been implicated in the development of drug-induced liver injury, and the list continues to grow. A searchable database of drugs, herbal medications, and dietary supplements associated with hepatotoxicity has been developed by the National Institute of Health. We can call it as NIH. Additionally, the NIH has developed a dietary supplement label database containing over 4,000 products. The next one, please. Sorry for bothering you today.
Oh, my pleasure.
Yeah. Now let's talk about the frequency of the hepatotoxicity. The next one, please. There are no precise estimates. There are no precise estimates of the frequency of the hepatotoxicity attributable to herbal and dietary supplements. Patients often don't report the use of herbal products to their clinicians, and they self-medicate with large amounts. As a result, toxicity associated with herbal use is missed. Only fewer than fish disclose to their clinician that they are using these products, either to HDS. It is a high percent of the cases. Additionally, this proportion of DLA caused by HDS increased from 7 to 20% between 2004 and 2013. Unfortunately, contrary to our idea that as science progress and the use of multimedia such as Insta, WhatsApp, and blah blah increase, awareness should also increase. We see that disease such as hepatotoxicity are increasing, unfortunately. Others, depending on the geography, 73% of cases of DLA. And the next one, please. About the diagnosis, patients are often reluctant to discuss the use of herbal and dietary supplements or don't recognize them as medications. It is important for the clinician to discuss the use of these products with the patient in a non-judgmental manner. Patients can then be educated on the potential adverse effects and herb or drug-herb interactions that may occur. This may prevent the development of hepatotoxicity. However, the unreported use of herbal remedies must be considered as a possible etiology of liver injury. The next one, please.
This one.
Call it Healy. Yeah, Professor?
Yeah. Hey, you can
Okay, yeah. Herb is, yeah, can I continue? Keep on, please. Herb-induced liver injury, we can call it Healy, typically presents as an acute hepatocellular injury with extremely elevated aminotransferase and bilirubin and jaundice. Patients presenting with the hepatocellular patterns of injury, particularly in women, have a poorer prognosis. However, it may also present asymptomatic liver enzyme elevation, acute or chronic hepatitis with signs and symptoms of cirrhosis. Continued use of the offending products increase morbidity one's hepatotoxicity. And the patient may even increase the use of the herbal preparations to help manage the new symptoms, further worsening the liver injury. And also the next one.
This one?
The next one, please.
Next one.
Yeah,
okay.
there is no gold standard and no specific tests or serum biomarkers exist to confirm a diagnosis of drug-induced liver injury from either prescription medications or an herbal remedy. And for patients,
We cannot
the
hear you.
next one, yeah.
We didn't hear you.
Yeah, that's exactly this part. And for patients with suspected Hillary challenge isn't advised, and also
Oh. A minute.
Professor, can
Yes.
you
Yes.
hear me?
Right now,
Oh, okay.
I'm hearing you. Thank you very much.
Okay. Thank you. For patients with suspected HELI, non-specific symptoms such as developing after the introduction of HDS, such as nausea, anorexia, myalgia, fatigue, right upper quadrant pain, or pruritus may indicate HELI and should promote evaluation. And the next one, please, about the treatment. Yeah, about the treatment. The next one, please. Okay, glucocorticoids are of uncertain benefit for most forms of drug hepatotoxicity, although they may have a role in treating patients with hypersensitivity reactions. In addition, we give glucocorticoids to patients with extrahepatic manifestations of hypersensitivity reaction that warrant glucocorticoids. And the next one, please. Yeah.
Yes.
Yeah. In this part, we want, in the finally part, we want to talk about the specific hepatotoxic herbs. Hepatotoxicity may be the most frequent adverse reaction to herbal remedy. The calpure, the next one is a valerian, the next one is misteletol with the scientific name of viscum alder, and the products of the misteletol is iscador. The next one is the iscalcap, chaparral, comfrey, herbal teas containing toxic alkaloids, it. And finally, we can talk about the kava. The next one, please. Yeah, in this part, if you can zoom it, or it's completely clear for you.
It is completely clear for us.
It's okay.
Yes.
Oh, okay, no problem. Yeah, I can explain in this part. In this part, we can talk about the plants which are the side effects of the plants are hepatotoxicity. We can start with the black cohosh, with the scientific name of Actea racemosa or Simicifoga racemosa, that the roots and rhizomes of this plant is used for menopausal symptoms. The main toxic constituents of these plants are triterpene glycosides, which have side effects such as hepatic necrosis and breeding the fibrosis. The potential mechanism of this action is autoimmune, and it is very rare, which has very rare. The scientific name of Lara atridentata. The leaves of this plant is used for antioxidant and as a health tonic. The main toxic constituent of this plant is northydroguaric acid, which can cause cholestasis, chronic hepatitis, and cholineangitis, and finally cirrhosis, with the mechanism of the induction of the cytochrome P45 3A. And the next one, the next plant is prostata, with the scientific name of Sereno auripens. And the other common name of this plant is Sao palmetto, which the berries of this plant is used for treatment of benign prostatic hyperplasia, which we can call it as a BPH. And also in the hair loss products, we use this plant. The main product, the side effects of this cholestatic hepatitis, which is the
Dear Dr. Parina, we cannot hear you.
Hepatitis. And the main mechanism which is attributed to this plant as a toxic side effects is cytochrome P45 and glutathione depletion autoimmune. The next were metasticum. The roots and rhizome of this plant, which we can use as an anxiolytic and sleeping aid. The kava
Dear Parina, we cannot hear you now. Your voice is coming.
Okay, which in the part of kava?
Kava, yes.
Ah, okay, yeah, I can repeat it.
Thank you.
Yeah, I repeat it. No problem, I repeat it. The plant of kava with the scientific name of Piper metasticum. The roots and the rhizomes of this plant is used as an anxiolytic, sleeping aid. The kava lactones as a toxic constituent of this plant, which cause acute and chronic hepatitis with the mechanism of the immunoallergic factors. The next one is a Clydinium majus. The aerial parts of this plant is used for gastrointestinal tract disorders and broad range of disorders. but with the unknown mechanism which is attributed to the autoimmune, it is caused acute hepatitis and cholestatic hepatitis fibrosis. And the next slide, please.
Yes. Next one. Yes.
Okay, thank you.
You're welcome.
Thank you so much. Yeah. About the Mao Huang, in the second part, Mao Huang, as we call it, is a Ephedra sinica. We use the aerial parts of this plant in acute hepatitis, in weight loss products, but the toxic constituent of this as alkaloids is which cause acute hepatitis with the autoimmune mechanism. In the third place, we have a polygonum, and which we can use the roots of this plant in health tonic, anti-aging, and also we can use it in the hair color. The root of this plant is used in broad range of disease and have some broad pharmacological effects. However, the anthraquinone of this plant has acute hepatitis and acute liver failure side effects with autoimmune mechanism. And in the last one, yes, the Cassia angustifolia, we can call it as a senna. The leaves of this plant is used as a laxative and in the constipation diseases. But the main constituent, the toxic constituent of this plant as anthraquinone senozoids, which cause the acute hepatitis. The last of the table, menta, which scientific name of Mentapulagium. The leaves of this plant is used for abortifacient or upper respiratory tract, but the polygon of this plant, which cause the hepatic dysfunction with epileptic encephalopathy with the mechanism of the glutathione depletion. And the last one is margosa oil with the scientific name of Azadirachta indica. The seeds of this plant is used for health tonic, but the bitter constituent of this plant cause the Ray's syndrome with the unknown mechanism. The next one, please.
Yes. It is.
Thank you. And the next one, please. I summarized all of these data in the previous slide. Yeah, the next one, please.
Okay.
Next one, please.
Again, next.
Yeah, the next one. Yes. Okay, in this part. In this part, we want to talk about the germander. Yeah, in this part, we want to talk about all of the plants which can cause hepatotoxicity. We want to explain all of them in a detailed one. The first plant is germander with the scientific name of Tocreum commodorice. However, I can't find the supplement products or the hair products from this plant in all of the world, but in Iran we use the distill of this plant and the powder of this plant for decreasing the diabetes and also for decreasing the blood pressure and if increasing the and it is used as an immunostimulant. In the next one, please. Yes. Yes, of course. Thanks to the Lamiaceae family, since ancient times, germander has been used as a herbal remedy for several properties, including antidepressant, diuretic, choleritic, and also tonic. Globally, whole germander has been used as capsules or teas for the treatment of obesity and as a weight loss agent. Moreover, germander was considered safe until many cases of hepatotoxicity associated with germander ingestion were reported in France. For this reason, in 1992, the germander powder containing capsules were prohibited and banned, unfortunately, from the French market after an epidemic of 30 cases of hepatotoxic effect associated with the germander ingestion. In the next slide, we can talk about this reports. Next slide, please. Yeah. For example, Larry et al. Reported one of the cases involved a 54-year-old woman who was taking germander capsules for weight loss and developed hepatitis. However, after 6 months of the germander discontinuation, the woman showed a complete recovery of the liver function optimistically. And in the another reports, a 15-year-old female adolescent who had abdominal pain and developed hepatitis with jaundice after taking germander capsule 600 milligram per day for a few months in combination with other drugs. She recovered after 1 month of germander discontinuation. And the next one, please. Additionally, the other case is a 55-year-old woman.
Dr. Parina, we cannot hear you. Dr. Parina, can you hear me? Dr. Parina, can you hear me? Yes, I'm hearing you right now. I think that we lost internet connection and we didn't hear the information about the first case. I mean that we cannot hear you from the beginning of this slide.
Okay, yeah, I repeat them again. Okay.
Kind of, yes, thank you.
No problem. No problem. The first case is a 55-year-old woman with no history of liver disease who took germander capsules 1,600 milligram per day as a medication for hypercholesterolemia with some other drugs. She developed jaundice after 6 months of the germander intake and diagnosed with hepatitis. However, the liver function tests of the patients improved fortunately after 2 months germander was discontinued. The second case involved a middle-aged woman and which used germander 260 milligram per day for 6 months to reduce her weight and was diagnosed with jaundice, asthenia, and hepatitis. Additionally, there is a case of 68-year-old woman who developed massive hepatic necrosis following ingestion of 450 milligram day of wild germander for 2 weeks, unfortunately. In the next slide, we can take which are famous for its antioxidant and reduce the radicals. We have a broad range of products such as fulfill green tea extract from the Now company and green tea extract from the Nature's Botany. In the next slide, we want to talk about its side effects when we consume it in the chronic period. Extracts of Chinese green tea, which can call it as a Camellia sinensis, are commonly used and have been associated with hepatocellular injury. In general, drinking green tea in moderation appears to be safe and may have beneficial effects. However, numerous supplement extracts are available and have been reported to cause liver
Dr. Parina, we cannot hear you.
Period. The exact mechanism of hepatotoxicity remains unfortunately unclear. In the next slide, the green tea extracts in the green tea extract catechins such as epigallocatechin 3 gallate, we can call it EGCG, are likely to be involved. EGCG induce the reactive oxygen, especially similar to other herbs, so predisposing in this case. Hepatotoxicity is increased in those who are fasting, for example, 12 fasting or 16 fasting in the day. The pattern of injury is predominantly hepatocellular, but mixed and cholestatic presentations have been also reported as well. Liver injury generally develops within 3 months of use and usually resolve with discontinuation of the products. In the next slide, in this part, we as a scientific name of Cassia angustifolia. As you see, we have broad range of this product in the constipation disease as an emollient and as a laxative products. For example, senna from nature's ways, senna from the leaves of the senna from the now, and senna graft in Iran we use in these products before the colonoscopy. In the next slide, Cassia angustifolia, commonly known as senna or Tina Valley from the Fabaceae family. The leaves of this product and the fruits of the senna have been used as a laxative from the herbal tea. However, excessive abuse of the Cassia angustifolia has been implicated in few cases of hepatotoxicity. Accordingly, a 26-year-old female was diagnosed with toxic hepatitis after intake of herbal tea containing 10 grams of senna leaves and 100 milligrams of sennazoid in type B extract of senna fruit twice a week. And another case of senna hepatotoxicity involved a 52-year-old woman who drank 1 liter of herbal tea containing 70 grams of senna dried fruit for more than 3 years. The patient developed acute liver failure with renal impairment due to chronic use of Cassia angustifolia. In the next part, another case, moreover, another case is a 42-year-old female presented with gastric pain, vomiting, and intermittent fever, the fever after intake of 200 milliliters of dried senna leaves for 2 years. Unfortunately, the patient was diagnosed with portal vein thrombosis, as we call it, PVT. Another case of subacute cholestatic hepatitis has been reported in 77-year-old male. The patient admitted taking senna for the treatment of constipation. I want to remove all the other parts because I want to say about the anthraquinone glycoside, which are responsible for showing these side effects. In this slide, we want to talk about the Valeriana officinalis, which we have a broad range of this plant as antidepressant, as a anti-tranquil, and as anti-migraine products in all of the world. In the next slide, Valeriana officinalis, which belongs to the Valerianaceae family. The dried root of Valeriana officinalis is known as valerian, which has been used for a long time by ancient Rome and Greece as sedative and anti-anxiety remedy and for treating insomnia. Additionally, it has been used as a remedy for treating palpitations, headache, gastrointestinal pain, high blood pressure, sleeping disorder, and blah blah. And the main pharmacological effects of this plant is attributed to its anti-migraine effects. In the next slide, some hepatotoxic events associated with valerian ingestion have been reported as well. The first case of the valerian-induced hepatotoxicity were reported in 1989, where 4 patients took over-the-counter sleep aids containing valerian. Another case of valerian hepatotoxicity was described in a 27-year-old female, took 1 capsule of the valerian root extract for 3 months. In the next part, in the interaction ability, we want to talk about the interaction ability of the 43-year-old woman who ingested the valerian with iscalcalp as a Escutellaria pinnatifida and black cohosh and some other herbal preparation develop hepatitis. We can conclude that when we use 3 or more than 3 plants simultaneously, we can increase the side effects of this plant, and unfortunately, it may cause hepatotoxicity. And in the next part, we talk about the Actea racemosa. We can call it as a black cohosh. We have the very supplements of this plant in the world, because Actea racemosa's root part belongs to the Ranunculaceae family. It is used for the broad range of conditions such as colds, cough, fatigue, and constipations. In particular, Simicifuga racemosa rhizomes have been used as a treatment of menopause-related symptoms, rheumatism, and also autoimmune type of liver injury and liver failure necessitating transplantation has been reported, unfortunately. In the next part, the main active constituents of history of liver disease, she started to use black cohosh to treat hot flashes, but she developed autoimmune hepatitis. In the next part, the main another case reported mentions a relationship between the use of Simicifuga and acute hepatitis. A 47-year-old woman with no history of the black liver disease took black cohosh for only 1 week with no other medication to treat menopause symptoms. Severe hepatitis and jaundice developed following ingestion leading to liver transplant. In the next part, we can talk about the Lara tridentata. Only the 1 product of this plant which has reported in the world, and it is an evergreen desert shrub which can use as a tea to treat rheumatic pains and bronchitis. The capsule and tablets of this plant is treatment for other conditions such as diarrhea, cold, rheumatism, urinary tract infection, and chronic skin disease. The next part. And also, among the herbs, it is very important to mention the potency of the chaparral is probably the best known antibiotic treatment derived from plants. Nor-dihydroguaiatic acid has been proposed as an active constituent of the chaparral, and also the mechanism of this plant is attributed to the involving cyclooxygenase and lipooxygenase pathway. Some of the reported side effects of the chaparral include vomiting, diarrhea, rash, fever, and abdominal cramp. Hepatotoxicity is the major problem linked to the chaparral ingestion. In the next part, in this part, we talk about the many cases, such as 30 patients was 25 until 60, and jaundice was the main sign of the liver disease. In another case, 22-year-old woman, the hepatotoxicity developed during the breastfeeding period following delivery of the healthy newborn and ingestion of the chaparral. And in the next part, we talk about the comfrey or symphytum. The next part. In this part, as you know that comfrey is famous for having the allantoin part, and it is used for the broad range of disease such as hair loss or another application such as anti-inflammatory disease. But the pyrrolizidine alkaloids are a group of naturally occurring alkaloids based on the structure of pyrrolizidine. They are used for a variety of complaints, including arthritis, gout, and a number of infections. However, over 50% of pyrrolizidine alkaloids exhibit hepatotoxicity, which are the families of boraginaceae, such as heliotropium, symphytum. It belongs to the family of echinaceae, leguminaceae, and the family of scrofulariaceae. Comfrey, it is very highly mentioned that comfrey has been removed from the market in France following numerous reports of the liver damage. In the next part, alkali toxicity usually results in moderate to severe liver damage, particularly to the hepatic central vein and in a sinusoidal endothelium. Hemorrhagic necrosis, hepatomegaly, acids, and endothelial cell proliferation are also seen. In the next part, the major hepatotoxic effect of pyrrolizidine alkaloids is the hepatic veno-occlusive disease, as we call it as a VOD, and sinusoidal obstruction syndrome. Portal hypertension with severe acid is the one major characteristic of VOD disease. And in the next part, we can talk about the first report of pyrrolizidine alkaloid intoxication, which was held in 1985. A 49-year-old woman was diagnosed with portal hypertension with obliteration of small hepatic venules, which only taken 85 milligrams of pyrrolizidine alkaloids for 6 months. Another recent case involved 66-year-old woman that was previously diagnosed with chronic disease. She consumed around 1.5. I want to summarize this other cases, and I want to talk about the next part as a echinacea plant. Yes, echinacea plant, we have a lot of supplements such as supplements or now or active immune products. As you know that we have 3 species of this plant: Echinacea angustifolia, Echinacea pallida, and finally Echinacea purpura. However, the Echinacea angustifolia is the main species of this genus, which has been used as a as an immunostimulant in many diseases. Echinacea has been used by Plains Indians as a remedy for respiratory infections and has been used as immunobooster and as a therapy for common cold. And in the case 45-year-old man who diagnosed with acute cholestatic autoimmune hepatitis due to the intake of the echinacea, unfortunately. And in the next part, we want to talk about the mistellato, with the scientific name of Viscum album. The main product of this plant is Iscador, which is used in the many cases of cancers, such as colon cancer. However, in this part, we talk about its side. It is used for many conditions such as hypertension, headache, insomnia, epilepsy, asthma, and infertility, cancer, and finally urinary disorders. However, one case report involved 49-year-old woman presented with general malaise, nausea. She admitted taking herbal medications and continuing mistellato iscalcab mouthwort, and finally it is she developed acute hepatitis. It is unfortunately it's a side effects of this plant. In the next part, we talk about the kava, which can call it as a piper mysticum, which is used for complaints such as anxiety, sleepiness, and menopausal symptoms. Numerous reports of severe hepatotoxicity and liver failure have been described in Europe and the United States, but following the ingestion of at least 4.5 months. And also clinical reports of the toxicity range from the transient elevation in aminotransferase to acute failure and death. And also it is the transient elevation in aminotransferase to acute liver failure and for also in death. And also in the next part, the toxicity of a kava was due to the extraction methods. It is very important, and severe hepatotoxicity of kava leading to hepatitis, hepatic necrosis, and cholestatic hepatitis, which is reported from the 14-year-old girl, which used it as a one tablet twice a day for 8 months. It is, it's unfortunately is a bad reports from this using this plant. And also in the next part, 6 patient, in this part, a 5-year-old female. There was another case of the kava associated hepatotoxicity involved, who had hepatic necrosis when had only 210 milligram per day of kava for 4 months. And in the next part, we want to talk about the pennyroyal as a scientific name of mentopelagium, and also it is used for the broad range of therapies for upper respiratory tract and ear infections. However, in the next part, a small amount of oil, 10 to 15 ml, can cause severe toxicity like liver and lung damage. And also one case of the pennyroyal oil toxicity, around 120 milliliters of tea had fulminant liver failure, unfortunately again. And also in the next part, 6-month-old boy who took aspirin and 90 milliliters of the tea only 3 times per week from homegrown mint plant and colic. The baby had hepatic dysfunction with severe epileptic encephalopathy. Okay, in the next part, we talk about the St. John's wort as a hypericum perfuratum, which is used in some illnesses such as insomnia, bronchitis, burns, gastritis, hyperthyroidism, and insect and snake bites, and blah blah. The St. John's wort has been used as an antidepressant remedy, but it interacts with numbers of drugs which can be beneficial, like the increasing response of clopidogrel and reducing toxicity of the irinotecan. In the next part, Yeah, the some interactions of this plant with other drugs show reduction of the blood cyclosporin level, reduction of plasma level of anti-cancer and antiviral drugs, causing serotonin syndrome of unwanted pregnancies. And the case of, in the next part, we can report the case involved a 59-year-old male who presented hyperlipidemia and initially took pravastatin as a 80 milligram per day. Unfortunately, the total levels of cholesterol and the low-density lipoprotein cholesterol levels increased due to the using the St. John's wort interactions. And 29-year-old female as a another case, it used this plant only twice a day, and he reported some other side effects such as hepatotoxicity in this case. Okay, the next slide. Yeah, Chinese herbal remedies. As we talk about these Chinese herbal remedies, such as Maohang, ephedra, and also another plants that I can't say the name of say the name of the plants, such as Kaozo or Onshido, Shizo, Zizu, Z, and I can't talk about this plant, but they use as a broad range of disease, such as weight loss, central nervous system in the next part, and also in the central nervous system. But however, the hepatitis, acute liver failure, and exacerbation of autoimmune hepatitis was reported from this plant. And in the next part, which is some weight loss products such as Herbalife and Slim Quick products, which has green tea, caffeine, chaste tree, and rhodiola extract, are very toxic drugs, and also we can use and forbidden all that them. And also the next part, please. The next one. And also I explained about the hydroxycut. And the next one, please. Yeah, the next one.
We cannot hear you.
For plants which are used in the broad range. This is the final slide for my presentation. Aloe vera, senna, and borax and camphor oil are the main plants which use in the chronic periods, which can cause the reports of toxicity with the broad range of herbal preparations, and we should avoid the using of this plant in the chronic period. Thank you for your attention.
Thank you very much for your fascinating presentation. We learn a lot of things about plants, and this kind of things, this kind of plants are being used by citizens in common way, and they are prepared. So we have also knowledge about their side effects in view of hepatotoxicity. Dear Parina, thank you very much for your presentation. We run out of time, so we cannot take any question from audience. If they want, they will let us to know their questions via our email, and we will send these questions to you. Thank you very much. Right now, we are going into second part of our symposium, and this second part will be moderated by Associate Professor Dr. Arta Armani. Thank you very much.
Thank you, dear Professor Mine Hocam. for your help and support during this organization. And now we will continue with another speaker, invited speaker, valuable speaker that we meet from previous symposium, Ana Gorine. And before starting the presentation, I would like to give a brief information about the professor. She will be here today with us by her topic, which is genomic epidemiology in the context of global health, and will show us the recent data that they already obtained. So Professor Ana has a bachelor degree from the cellular and molecular biology from the Federal University of Rio Grande do Sul, Brazil, and from the same university, she has also the master and the PhD degree. Besides this one, you can see the academic career of professor, where she graduated from the University of Brazil. She worked as a researcher at the National Institute of Health. She worked also as a postdoc fellow and senior researcher in the Ion School of Medicine at Mount Sinai, USA, as a consultant in different projects, and now she continues to work as a professor at the same university in the General Cellular and Molecular Biology Department, in fact, Federal University of Health Sciences de Porto Alegre. Professor also has a lot of, and maybe now we don't show all of them here, but a former coordinator of Center of Technology Innovation in Health, and has another duty. And the research expertise of the professor is cellular and molecular biology, bioinformatics, One Health, global health approach, together with the genomic epidemiology, respiratory viruses, innovation, and intellectual property. Of course, as Professor Erguven mentioned, that maybe these one do not show all of the staff of the research activity of our speaker here, but you can see that you have the professor with over 1,700 citations. And of course, professor has a lot of awards. Some of them are already included here. In 2005, Young Scientist Prize in Brazil, and another award as a best researcher. And I would like to welcome Professor Ana Gorine, that she is here and sharing her valuable time with us. Welcome, Professor, and stage is yours.
Thank you, Professor Armani, for such a nice introduction. Thank you very much. And I'm very happy to be able to talk again with you about the global epidemiology and global health and genomic epidemiology. And let me share my screen here. Okay. So let me put on presentation mode. Can you see it?
Yes, it's a full screen. Everything is okay.
Okay, great. So I want to talk a little bit about the genomic epidemiology in the context of global health. And this is a topic that has been increasing in the past few years, especially because of the pandemic. So when we think about the global burden of disease, we always say that cardiovascular disease are the main cause of death in most countries. And how did this pattern change along the years? So many years ago, back in the 90s, we had infectious disease as the main cause of diseases. Still, cardiovascular disease was the first cause. It's still the first cause. But as we can see here in the GBD database, the Global Burden of Disease database, between 1990 and 2019, we could observe a decrease in the impact of infectious diseases. However, we still have respiratory infections and tuberculosis as one of the main causes. This is worldwide. And here, when we also compare how the situation, the scenario is, the top 10 cause of deaths, this is back in 2000, and this is in 2016. Again, we see a decrease in the burden of infectious diseases. This is for the global scenario. However, when we think about low and middle income countries or very low income countries, such as those in Africa, in Latin America, in Southern East Asia, we still have infectious diseases as the main causes of deaths in these countries. So this is clear that we still need to fight to combat infectious diseases. Now let's turn a little bit to molecular epidemiology. Even though we've been talking about this in the past 4 or 5 years, as I mentioned, because of COVID and other infectious diseases, Ebola, Zika, this term, this expression, molecular epidemiology, dates back from the 90s. So here are a few papers that bring this term, this conception of molecular epidemiology and its applications, including sequence analysis, nucleic acid amplification, and to study viral infections. I bring this example here for you because it sounds like what we experienced in the end of 2019 with the coronavirus, the new coronavirus emergence. So this says that an outbreak of respiratory illness of unknown etiology occurred in the United States in 1993. And these patients, they had a respiratory distress syndrome. The fatality rate was very high. And then they isolated the samples. They realized it was a hantavirus. And so this is very similar as what happened at the end of 2019 with the coronavirus, as I mentioned. So this was all based on the hantavirus sequencing that they were able to detect that this was a novel hantavirus. And regarding coronavirus, this paper here is back from March 2019. So since, many years ago, not many, but some years ago, we were already studying coronavirus. So this was a paper about the molecular epidemiology, evolution, and phylogeny of SARS coronavirus, the SARS coronavirus that caused all the epidemic back in 2002, 2003 in Asia. So what is genomic epidemiology? This new term, instead of molecular epidemiology, we are using genomic epidemiology. So it's the use of genomic information using mainly the next generation sequencing platforms, what we call high throughput sequencing, to investigate the distribution and spread of infectious agents and to mitigate disease in a population. And this has been used globally. Here is a map showing that there are many hotspots of emerging infectious diseases, most of them caused by viruses, but a lot of them also by bacteria, such as cholera. These were outbreaks that occurred in different parts of the world. So we have here in 2003 case of Yersinia pestis that caused the Black Death, the plague. So we still have Yersinia pestis circulating, the bacteria circulating in many countries, but it's been controlled. And a lot of cases of influenza, avian influenza. Now, back in 2023, last year, we've been having a lot of cases in Latin America, in South America. And here the Zika virus, yellow fever in Brazil, and the Zika virus in 2015 that spread to all countries of South America, some countries in the Caribbean, and it was a threat, a global threat. And then, as I mentioned, the recent coronavirus that caused COVID. So this is going back December 2019, the first announcements of those cases of pneumonia that were occurring in Asia, in Wuhan, and we did not know what it was. Thanks to the new sequencing technologies, the high throughput new platforms that are able to sequence a virus in in few hours, and also thanks to traditional virology techniques such as ultra microscopy, where the researchers, scientists were able to detect that it was a coronavirus, and also to sequence the genome of this virus. So here's the timeline from the first patients that were attended at the hospital in Wuhan until the time that the coronavirus was sequenced and reported as a new coronavirus. We had just a few weeks. However, in another couple weeks, this virus was able to spread to many countries, and it became a pandemic. This is a slide that I still have. This image, it's back from March 5th, 2020, when we're still under 100,000 cases of COVID all over the world, with less than 3,500 deaths. 3 years later, this is March 2023, March 10, 2023, we had more than 700 million cases and almost 7 million deaths. So this was the burden, how serious the COVID was all over the world. So I'm sorry to say this, it's neither the first, neither the last pandemic that we experience. This infographic here shows most of the pandemics that the human species has experienced along the history. So we have reports of pandemics from centuries ago. The main ones were caused by Yersinia pestis that caused the plague, the bubonic plague, with more than 500 million deaths. This was more than half of the world population at the time. And with the advance of vaccines, antibiotics, and also improvements in sanitation, we were able to control most of the bacterial infections. And then with the increase in human population, in the density of the population, we started to have more cases of viral infections, especially those that are transmitted by the air. So airborne infections, such as influenza, such as Ebola, even though it's not a respiratory virus, it can be transmitted by the air. So all these viruses that are easily transmitted between humans, they increased in their the severity of diseases and the number of epidemics and pandemics caused by these viruses, especially the influenza virus, as we had the Spanish flu about 100 years ago, and then different types of flus, and now the avian influenza that has been, it's been considered a very serious threat in case this virus get more adapted to humans. Here again, another graph showing the recent viral pandemics, as I mentioned, influenza, and then we have the avian influenza, Ebola. We can never forget HIV, which is also and is still a pandemic because it's been increasing the resistance to all the antiretroviral treatments that we have, and we still do not have a vaccine, a good vaccine against HIV. And we are observing increases in arboviral diseases, especially because of climate change and how the mosquitoes and other vectors get more adapted to environments where the weather is getting warmer and warmer. And with that, we have more transmission of arboviruses such as dengue, Zika, chikungunya, Opuche, and other emerging arboviral infections. Here is the global number of deaths from influenza pandemics. As I mentioned, the Spanish flu, which was one of the worst influenza pandemics so far, and then we had other recent outbreaks. And because of the importance of these influenza pandemics, since since the 40s, back in 1947, the World Health Organization has a surveillance program for respiratory infections. This program has as a focus influenza virus. But as you can see here in 2003, due to the SARS, the coronavirus emergence in Asia, the SARS was included in this program as a SARS alert. And thanks to this, that we were able to detect the coronavirus that caused COVID in 2020. And also in 2017, respiratory syncytial virus was also included in the surveillance system. So surveillance is of paramount importance. It's very important to control infectious disease. And what is genomic surveillance that we're able to do now? Not just a clinical surveillance of symptomatic patients, but also genomic surveillance. It's nothing else but collect a sample from the patient and detect, not only detect by molecular techniques, the viral or bacterial, the etiological agent causing that infection, but also sequencing that virus or bacteria or fungus. So why is genomic surveillance important? So it's important for clinical and laboratory data, for epidemiological data, and also for environmental data, because we can also not only sequence and analyze microorganisms in the patient, but also in the environment to see if like water, the soil, or even surfaces, fomites are infected or are contaminated with that specific microorganisms. Here are some examples also of the use of viral sequence data. Because I work with viral infection, I'm gonna give examples of genomic epidemiology of viral infections. So the sequencing of viruses provide information about their emergence, phylogeny, and study population dynamics, how the virus can spread in the population, how they adapt to the host and to the environment, and also understand the dynamics of diseases. This is the case of the SARS-CoV-2. With the sequencing of SARS-CoV, we're able to characterize the SARS-CoV genome, understand what parts of the genome are more conserved, what parts are more variable, that change or have high mutation rates. And based on that, we are able to understand how the new variants of coronavirus emerge, how they change, what are the hotspots of mutations along the genome, understand how the virus is spread from Asia, from Wuhan, to the other geographic areas. And so, as I mentioned, understand the dynamic of the virus in the population. This is an image from the Next Strain database that was created especially to understand the Zika emergence and the Zika epidemics back in 2015. And then it was also included for influenza, Ebola, and now it really increased in sequencing data with the coronavirus pandemic. So here we can see how the coronavirus was spread all over the world in each epidemiological week and each week since November, December 2019. So as I mentioned, we can detect parts of the genome that are more conserved. And based on these conserved areas of the genome, we can develop molecular targets. We can develop primers for PCR reactions, for molecular diagnosis. So as for instance, here, the first primers that were developed for PCR to detect the coronavirus were based on these conserved regions in the ORF, in the polymerase gene, also the envelope gene of the coronavirus, and the nucleoprotein gene. Here are one of the first protocols to sequence and diagnose COVID, and then of course other primers from other companies were developed, and all of them had to be approved by CDC and other international agencies to be able to be used as a gold standard to diagnose COVID all over the world. So this is one of the examples of the use of genomic epidemiology and sequencing of these infectious agents. I'm giving the example of coronavirus, but this can be used for other infectious microorganisms as well. And here, a lot of us has had this experience of having a swab collected from the nasopharyngeal tract and then sent to the lab, or people that have worked on this process of extracting the DNA or RNA from the virus, performing PCR, real-time PCR, and for those that want to go deeper into genomic analysis, perform all the sequencing. Also, the molecular analysis is important, as in the case of influenza, to give basis for the World Health Organization recommendations for the flu vaccines. So each year, the WHO recommends what should be the composition of the flu vaccine according to the strains that circulate either in the North Hemisphere or the Southern Hemisphere. So for instance, for 2024 vaccine in the Southern Hemisphere, the trivalent vaccine should contain a strain of H1N1, a strain of H3N2, and a strain lineage of influenza B virus. And the quadrivalent vaccine could include another lineage of influenza B virus. And for the Northern Hemisphere, only the trivalent vaccine is being considered because one of the lineages of influenza B is not circulating right now. So it's only necessary to include the Victoria lineage, not the Yamagata lineage. So this is an example of how this sequencing is important to detect which strains are circulating and how should be the vaccine, the influenza vaccine, composed. So here, not only vaccines, but also antivirals. For instance, for hepatitis C virus, we can sequence the virus and see, and because hepatitis C is a chronic disease, we can sequence the virus from one patient and observe how that virus is evolving along the time, how it's mutating in that patient, and how the antiviral treatment should be changed, should be always updated according to the viral resistance to those antivirals. So here are all the applications of the development of antivirals based on genomic sequencing of that virus. I'm going to show now a little bit of the research that we've been doing in our university together with the government. So I have partnership with the State Secretary of Health, with the Public Central Laboratory, and also the Laboratory of Genomic Sequencing that we created now in the state. And we've been doing all these genomic surveillance. We started with epidemiological surveillance of viral infections, and now we are evolving or improving to genomic surveillance. So here's where I'm located in southern Brazil. As you see, I'm in the southernmost part of Brazil. So we don't have most of the tropical diseases that are more common in the north part the country, such as malaria, Chagas. Those diseases are not very common. But because of our colder weather, we have a lot of respiratory infection. And now with all the climate change, we are also experiencing an emergence of arboviral diseases in southern Brazil. So most of my research is focused on respiratory viruses. Here's some work that we did back in 2011, 2014 with influenza virus, and we sequenced samples from the 2009 swine flu pandemic that we had. And then we also sequenced samples from 2011, all of them from the state of Rio Grande do Sul, where I live. And here we can observe the evolution of influenza virus in our population. And we can also observe the decrease in the vaccine effectiveness, the vaccine that we were using, how we had a decrease in effectiveness because of these changes in the virus. And I was also able to sequence the first whole genome of influenza virus from Brazil. So these were the first full sequence of influenza deposited in gene bank that had been collected in Brazil. And as I mentioned, this was together with our central laboratory of the state. And this was a work that I performed at Mount Sinai in New York in 2015. And all the samples are from 2009 until 2016. So here we have a representation of about 6 years of the virus in the state. And so we analyzed the samples that we sequenced, these 220 genomes, and we're also able to observe how the virus evolved, how all the phylogenetic and the dynamics of these clades and these strains along the years in our region, and how they compare to strains that circulated in South America, that circulated worldwide. And also, based on genomic sequencing, we're able to detect cases of co-infection of influenza A and also influenza B virus. And these were it was believed that the co-infection cases were rare, but in this study here, we show that it's not as rare as was believed. Based on genomic sequencing, we can see that is much more common than we thought. And in this study, we reported the first case of a triple infection that included influenza A H3 and 2, influenza A H1 and 1, and also influenza B virus. This patient here, he stayed 96 days in the hospital. So for more than 3 months, this patient was hospitalized. And as we see here, the median time, 97 days. And also for the coronavirus, we are able to collect the first cases that circulated in the state, the first 15 cases, and also some cases that occurred in the first year of the pandemic. So we had about 90 genomes that were sequenced. And with all this study here, we're able to tell the story about how the coronavirus entered, how it started in our state, and how it spread along the first year of the pandemic. And this was very important because our state government we used the flag system. So the state would advise people that we were in the red flag or the orange flag, and based on that, they would drop lockdown measures to control the pandemic. So all the lockdown weeks that we had, for instance, then the shops or schools could open, and then they had to close again because of the emergence of new variant, a new strain of the coronavirus. That was based on these studies that we are performing along the weeks. And so here is still the same study that we showed the main mutations that were observed in the coronavirus along these weeks of the disease in the state. Here is for respiratory syncytial virus, a study that we just published now at the end of 2023. It was accepted, it came online on March this year, March 2024, that we were able to sequence some cases, some samples of respiratory syncytial virus. Because of the pandemic, the virus just disappeared in 2020, especially because of the lockdown measures. Kids were not going to school, children were not attending school. And as we know, RSV is a very important virus in newborns and children up to 2 years of age. And because all the kindergarten and schools were closed, we didn't see a circulation of the virus. But then in 2021, we started to see the virus circulating again. And these are the first genomes of respiratory syncytial virus from Rio Grande do Sul, also deposited in GISAID and GenBank. And as I mentioned, because of all the weather changes and all the environmental change, not just the weather, but also biodiversity loss, and now the deforestation that we've been experiencing in southern Brazil, we are seeing cases of dengue, chikungunya in places that they did not occur before. So here are some studies that we started to perform with arboviral diseases. As I mentioned, first only epidemiological studies, and now we are also performing genomic sequencing of these viruses. So here is a study of a chikungunya virus outbreak that we had in 2021 and also 2019 in the state, without the characterization of this outbreak. And here is the recent increase that we see in dengue in the state. 2022 until recently had been the worst year so far. But as you can see, we are still in May right now, not even May, we're still in April, and we are surpassing the number of deaths that we saw in 2022. And here's a recent work that has been submitted for publication. It's published in a preprint platform, but it has not been accepted for publication yet, showing all the dynamics of dengue. And we also observed a case of a double infection. So this is a patient that has been infected with 2 different serotypes of dengue, dengue 1 and dengue 2. So this has been right now, every day on the news, we see everyone talking about dengue and how the outbreak here is very serious. And also monkeypox, as we saw in 2022, there were epidemics everywhere. And in this study here, we were able to detect a case of monkeypox infection into healthcare workers. And even though these people here, they were wearing all the necessary equipment for protection, personal protection equipment, masks, gloves, and lab coat, they attended a patient at his home. And when they opened the door and they walked in and out of the patient's house, they were not wearing gloves. They only wore gloves when they touched the patient and when they collected the sample from the patient. And then they took off the gloves and they carried the box where they had the patient's sample inside the box. And somehow they got infected with the monkeypox. So this study here, it suggests infection through the contaminated surfaces, either at the patient home or even on the box that the health workers were carrying. And we were able to observe that this was indeed a contamination from that patient based on the genomic analysis of the monkeypox virus that was collected from the patient and from the health workers. As we see here, was exactly the same strain, the same sequence, the same genome sequence. And so here, our other works from our group, as you can see, we are able here to support public health policies and guidelines, as I mentioned. We've been doing some research on the role of different types of healthcare institutions in notifying and hospitalizing cases, for instance, of COVID and other respiratory infections. We're also using machine learning to predict outbreaks. Here are some examples. This picture here I took this morning this is a screenshot from our updated number of cases here in Rio Grande do Sul in our state. That's caused by dengue. So this is the state government panel for dengue and arboviruses, and here the coronavirus in the state, and here all the genomic surveillance that we are doing together, the university together with the state government for the vigilance, surveillance of coronavirus variants. So this was the last report in March 2024. And not only these diseases and all these other emerging viral diseases that we have to keep in mind, the zoonoses, they make up about 75% of all the emerging infectious diseases. And we need to be prepared. As a matter of fact, there is an epidemic preparedness index for countries. And this index is based on several variables, several factors that contribute for a country to be prepared for epidemic or even a pandemic. For instance, surveillance, immunization, medical workforce, coordination. Remember that to control a pandemic or epidemic, it's a collaboration among different stakeholders, different professionals. And the global distribution of epidemic preparedness, as you can see here, a lot of countries are still in need of preparation of professionals, training, and develop more human resources to be able to contribute for all these diseases that are a threat worldwide. As we see, something that happens in a country regarding infectious disease can easily be spread to other geographic regions. And so, as I mentioned, ways to control and prevent infectious diseases, there are several ways. And we need the contribution of governments, professionals, and individuals from each one of us. In other words, if we consider here, how can you contribute to prevent these diseases? How each one of us can contribute to prevent the spread of diseases. For instance, if I have some cold symptoms, like flu symptoms, should I go to work? If I really need to go out, why not wear a mask? I should wear a mask. I should prevent transmitting that infection to other people, even in my household. And this, everything that I'm talking about here is a way to contribute with the Sustainable Development Goals of the 2030 Agenda of the United Nations. As we see, good health and well-being is goal number 3, but infectious disease and epidemics is related to all of these SDGs. For instance, poverty, we know that vulnerable populations are more populations with low income and no access to food and sanitation. They're more vulnerable to infectious diseases, and they can also spread the disease to everyone in the community. So the health is in the center here of the 2030 agenda. It's related to all other sustainable development goals. And this is the decade of action: global action, local action, as I said, and people action. So think globally, but act locally, act individually. And this is my question to all of you. How can each one of us contribute for the agenda? Like being consuming less, contributing for the environment, contributing for everybody's health. And here are some pictures of my town. Even though these are very nice pictures, it's always good to remember that we also have problems regarding sanitation. We have a lot of poverty. We have problems of diarrhea in children living in the periphery of the city. But it's still a nice place to live, and I invite all of you to visit us sometime. So thank you very much, and maybe I hope to be with you in person at some point. And I'm open for questions.
Thank you, Professor Ana Gorine, for your valuable time and being with us, which is such a wonderful presentation and a lot of recent data, which is very important for us. And now I would like to ask participants if they have any question. And I think there are two questions here from the chat part. Maybe you can see already there. So I will
How can I stop sharing?
Yeah, you can stop that.
Yeah, I can't see how I stop sharing.
It's in the middle on the red
color. Oh,
column.
No, it's right there. Okay. Yes.
So I will read the first one, which is Professor Rijeka, in the realm of genomic epidemiology applied to global health, how do you address the challenges of data equity and accessibility, particularly in regions with limited genomic sequencing infrastructure or resources, to ensure that all population benefit from the insight gained into disease transmission dynamics and pathogen evolution? In fact, you talked a little bit about that, but maybe students didn't see that one, but maybe you can answer that.
Yeah, so that's one challenge that we do have here in Brazil. So as I showed you, we've been doing a lot of studies like that. But for instance, just to give you an idea, our state with the pandemic, they put all the efforts to give support, financial support for researchers involved in this type of study. And I was one of the few researchers to get one of the proposals approved for sequencing. And that the study, the money that they gave me was for 3 years of research. And in dollars, it was about $25,000. And for those that work with sequencing, know that with $25,000, you can barely do anything. So it's really a matter of collaboration. I had to ask collaboration from other scientists that were able to use part of their research budget to help me buy reagents for sequencing. Instead of sequencing thousands of samples that we see most developed countries doing, I was able to sequence 90, less than 100 genomes. But still, it helped to understand how the virus was evolving. So I think scientists need to raise their how do you say, their point. They need to require from governments meetings, more attention, more value to their research. And communication is one of the best ways to do it. And also, I know that many, most of that's, I always say that Brazil is a little bit in the limbo when it comes for international budget, because we are seen as as a developing or a developing economy or a middle high income country from the United Nations point of view. But when it comes for local problems, we are still undeveloped. We're still low middle income country. So it's very hard to get budget from international agencies because we are not considered low income countries. So for low income countries, there are many calls for, like, for instance, governments, United Nations, the NIH, Bill and Melinda Gates Foundation, and other international agencies that give support for this type of research. And so also collaborations is a way to do it. But it is a very hard question to answer. And
Exactly.
improve, have scientists, if they cannot do it in their country, like I did with the influenza, I spent 1 year at Mount Sinai in New York sequencing my samples. So I was able to get a fellowship. It was easier for the government to give me a fellowship for a 1 year research than giving money to sequence the virus. So, and I'm glad to see that with the new sequencing platforms, the price for sequencing is also decreasing a lot. It's going down. So it's much easier now to sequence a viral genome.
Thank you, Professor. And for the second one, as I know you already mentioned about that during your presentation, so I just was wondering, I'm asking this question now. You mentioned that you were the first that already uploaded some of the 200 data of the influenza A virus in the gene bank as a database for your sequence. And of course, now approximately up to 8 million data of the COVID-19 are uploaded in the Usher, ultra-fast replacement, which has already extincted trees that enables the real-time phylogenetics for the SARS-CoV-2. And is there any study in your country that the scientists would like to take all of this data, which is 8 million up to, and work with the computer scientists in order to predict the coming mutation in the future? So because some computer in the artificial intelligence here, there is a group of scientists and we come together to predict, the aim of the project was to predict that what can be the coming mutations in the future. So is there something in your country related with this one? Or collaboration?
Yeah, we do. I'm not doing this, but I know there are some groups that do this prediction. But for instance, with influenza, that has been studied for many more years than the coronavirus. And I've been to many meetings with people that are focused in influenza, like the options meetings, the option to the battle against influenza and other respiratory viruses. It's really hard to predict. It's very hard, even with all these computer prediction and based on previous mutations, it's still very hard to predict because there are a lot of factors that contribute for mutation and for the viral adaptation to new hosts. So we can predict mutations, but we cannot really predict how that new mutation can in fact cause an epidemic or cause more higher severity. It's still really hard to do it from my experience.
Thank you, Professor.
And I was this question that was raised.
Professor Erdogan also has raised, and I would like to say that now they are another speakers, Professor Fatma ATALAR and Associate Professor Peter James here. Maybe they would like to ask something, and we are all together now for the second session. Yes. Yes, Professor Ergüven.
Okay, thank you very much. Thank you very much for your fascinating presentation. And I know that there are a lot of big data about this issue, but my question is, I'm asking my question in the view of biology. And for example, do you think that there is a race between viruses? For example, I have read that influenza virus can inhibit the activity of COVID-19. So, but I haven't confirmed this via publication. You know that we have a wonderful source as PubMed, but I haven't confirmed this. But I'm wondering that, what do you think about this?
I didn't really understand the question.
I'm asking that there is. I have read that influenza virus can inhibit the activity of COVID-19. And do you think that there is a race between viruses in order to maintain their viability? And do you think that viruses can be used as a treatment modality?
I'm not sure if virus could be used. We have, for instance, adenovirus that because adenovirus is a family of viruses, and only some adenovirus can cause respiratory infection, as some adenovirus are used as vectors for vaccines, as vectors for a lot of things, because they can easily infect human cells without causing any symptoms, except those adenovirus that do cause respiratory infection. So regarding influenza inhibiting COVID, it would be more like we've seen cases of co-infection, a patient infected with influenza and coronavirus. So usually what happens is after you get infected with a virus, you have your immune system responding to the viral infection. So for that acute phase, you could be a little bit more protected against another viral infection in that specific scenario. Like it's all a matter of timely infection between each virus. Now, if you could use viruses to inhibit other viruses, I believe in vaccines more than the use of those viruses.
Okay.
Virus only for the purpose of being vectors for vaccines.
Thank you very much.
I see this other question here on the side regarding the interventions and policy decisions, what collaborations or partnerships are essential for facilitating the timely sharing of genomic data across borders. So that's another problem that we face, because, for instance, here we have the human health surveillance system separated from the animal and environmental surveillance system. And it's very hard for these systems to share data. So we have different digital platforms or online platforms. And a lot of things they're done in the environmental and animal health is not really shared with the researchers and professionals that do with human health. And that's one of the bottlenecks that we have, one of the weaknesses of our system. The ideal would be an integration. Think about the One Health approach would be an integration of people that analyze environmental samples, animal samples, and human, non-human animal samples and human samples, and combine this information in the same platform. And for instance, also the people that sequence the sample, so we have the genomic data combined with the medical clinical data. So we can really have information about how that specific viral molecular characteristics may be associated with certain clinical symptoms, clinical data, or age, or other characteristics of the patients. And if it's hard to do it locally, you can imagine globally, because then you have all ethical issues, you have different country policies, depending on the countries. And again, collaboration is the key point to improve each of these situations.
Thank you, Professor. I think that we are at the end of Professor Sanagorini presentation, and we would like to thank again you, because you are very far away, it's morning there, and hope that we could host you in our campus in the future, because it's the second time that we meet virtually in such symposium. So hope that we will meet face to face. So thank you again.
And please, if you have any questions, you can send me emails. You can share my email. I have Instagram, Ana Gorini Veiga, if you can follow. Okay.
Thank you, Professor.
Thank you for the invitation again.
And now we would like to welcome our professor, Fatma ATALAR from Turkey. Now we have only one speaker from Turkey, so she is representing back Turkey here today. And before starting, I would like to give a brief information for our professor, like for all of us that we did. And of course, before starting, I would like to say that Professor Fatma ATALAR is one of my professors that has a very special place in my heart and in my academic career, because we know her as a third-year student from the faculty. And by her support and by her help, we had the opportunity to go to the European Congress, which was not so easy to go at that time. So we would like to thank again Professor Fatma for her support during our, in fact, studies. So let's start and give a brief information for our professor. Professor Fatma ATALAR today will be with us with her presentation, which is Analytic Systems in Rare Disease Ecosystem, Genetic Diagnostic, Metabolics, and Future Perspectives. Professor gained her bachelor degree from Faculty of Science in 1992 from the İSTANBUL University, Turkey, where she continued already in the PhD in the United Kingdom in the Faculty of Science and Health, and she has done postdoc studies in University of Sussex in United Kingdom, in the Strasbourg University, and again in another university in the Kepler University, Austria. Besides education, I would like to give a brief information about the academic career. Of course, we cannot include all of the career of our professors here, but shortly we will show you that after her assistant professorship, she was already associate professor in 2019, where already continued in Institute of Child Health in Istanbul University, then as an associate professor, and now she continues as a professor at the Institute of Child Health for Rare Diseases since 2023 at Istanbul University. Besides that, already Professor Fatma ATALAR has worked between 2040 to 2070 in Muhimbili University of Health and Allied Sciences as the representative of the institution of Istanbul University. And she had done a lot there. And maybe she is going to talk about this part. Beside academic career, professor has another duty as a managerial experience. In fact, she worked for 3 or 4 years as a director, as I mentioned, in the Muhimbili University of Health and Allied Sciences, and she is now as a head of Institute of Child Health and Rare Diseases, again at Istanbul University, Turkey. And as I mentioned before, she was the institutional representation at Muhimbili University between 2015-2018 for the establishment of Muhimbili Human Genetics Laboratory. And the field of research of professor especially is natural sciences, molecular biology and genetics, psychiatric disorder, oncology, hematology, and metabolic disorder. Of course, we cannot show all of the research activity of the professor here, but if you are interested in collaboration, you can see here the papers or the articles that professor has already here. And at the end, I would like to welcome Professor Fatma ATALAR and sharing her valuable time with us. So welcome, Professor. The stage is yours, and now I will make already you are a panelist and you have the permission to share your presentation. Welcome again, Professor.
Thank you.
Maybe you can do a full screen just from FPS.
Is it okay now?
Yes, it's okay now.
Okay. Arthur, thank you very much for the invitation and for the nice presentation. Today, yes, today I'm going to talk about analytic systems in rare disease ecosystem, metabolomics, genetic diagnostics, and future perspectives. My talk will cover rare diseases and genomics, metabolomics, technologies, together with real-life examples of rare diseases, then the challenges and the future perspectives. Right, okay, so rare diseases. What are rare diseases? The rare diseases are affecting 5% of the population worldwide. And there are about more than 8,000 diseases, and 80% of these are hereditary. And mostly they show recessive inheritance pattern. And so we all know that one of the main reasons is consanguineous marriages. And there are a limited number of treatments, unfortunately, and the reach of those treatments is not very easy. Well, a third of all rare disease patients unfortunately die before reaching their 5th birthday. For me, the part that is very painful is about children and is about very severe diseases, often not very efficient treatment options. And research in this field and understanding the mechanisms and what really is causing the disease is very crucial. Well, the first hurdle that many patients are facing is the diagnostic odyssey. So it takes more than 8 years to get a precise diagnosis, and the patient pays multiple visits to different physicians and ending up having 2-3 misdiagnoses. Well, we have been talking about rare diseases about 2 decades now, and we can say that the technology is also much more improved thanks to omics, which we are going to talk, soon. And also we know the people suffering from rare diseases are more now known. We know about them more now, and we have more knowledge about the diseases. So the difficulty in diagnosis is mainly due to the phenotypic and genetic heterogeneity, which can make them difficult to diagnose even by specialists with prior experience. And often, let's not forget, the multi-system impact of the condition can be masked by common complex disease symptoms. So therefore, the precise diagnosis and treatment are not available around the world yet for all of them. And I want to share the data of Turkey. Turkey, well, the population of my country is around 100 million. And in Turkey, we have more than 5 million rare disease patients. And well, unfortunately, the consanguineous marriages rate is around 24.5% in my country. And so we have a large population, and the genetic pool is diverse. And I can say Turkey is ideal for genetic studies. So let's have a look to the term of omic, which is going to be, the subject of the next slides. Omic stems from the suffix of om, added to the many fields of biological studies, which refer to the study of something in its entirety. And so there are estimated to be over 500 omic types. I mean, I found it very surprising when I did a bit of research, on the internet. And but the most commonly known are genomics, epigenomics, transcriptomics, proteomics, metabolomics, and phenomics. And these omic types have been used to identify or you can say provide functional supporting information for candidate pathogenic mutations for rare diseases across various medical specialities, of course. And we are going to talk about genomics a bit, and genes and genomics. And for rare diseases. So rare diseases is just one end of the spectrum of human disease, and there are diseases that fall along an entire continuum of genetic architecture. So on that hand, we have the diseases that we work on traditionally here, also in my group. These are severe monogenic diseases that can be very severe or can be relatively mild. But these are monogenic in the sense that they segregate within particular families in a dominant, in a recessive, or X-linked fashion. And at the other end of the spectrum of disease, we have common complex disorders, and these are the things that cause actually the most mortality and morbidity across the human population. And I can give you the example such as type 2 diabetes, cancer, or heart diseases. So these are often, but not always, massively polygenic, meaning that in any individual case, there is a whole range of typically small genetic risk factors as well as environmental risk factors that contribute to the person's risk of getting the disease. Okay, this, I'm sure that you all know this this picture, and I will very briefly, go through that. Genetic variants that contribute to disease also follow on a spectrum, and we have down the x-axis here. So we have the allele frequency of the particular variant, and then the y-axis here, we have effect size, that is the probability, as you can imagine. So this is the probability that someone who carries that particular variant will go on to get the disease. So the top right-hand corner, yes, is basically entirely or at least very sparsely populated with variants. And the bottom left-hand corner of the screen, we have variants that are actually impossible to detect using current sample size and technology today. But the variants we can affect fall in the spectrum, yes, in the spectrum in the middle here, and where we have top up left corner, typically the very rare variants with enormous effect sizes that are associated with Mendelian disease. And down the bottom right-hand corner, we have the common variants with small effects we would see in genome-wide association studies. But the important spectrum in this spectrum is the ones in the middle. And now that we are starting to pick up as we apply next-generation sequencing approaches to more common complex disorders. So we know the Human Genome Project have changed the modern, biomedical sciences, rare and common genetic disorders, our understanding in human biology and human genetics. And so when it started 30 years ago, with the discovery of Fred Sanger and the English chemist who won his also second Nobel Prize for it. And the first human genome took actually 13 years to accomplish. It involved 6 countries, 20 universities, and it cost around $3 billion US. And now the next generation sequencing came to the scene in mid-90s. And now that we are in 2024, the personal genome can be elucidated in a number of hours, which is very handy. So let's have a look to the next generation sequencing a bit. So we know that in human genetics, various sequencing modalities are employed, each targeting distinct genomic region: whole genome sequencing, whole exome sequencing, targeted panel sequencing, and RNA sequencing. So these methods leverage both the second generation and the third generation sequencing technologies, with current trends favoring advanced platforms like accelerated genome sequencing, as an example, NVIDIA, Clara, and FlashBlade, and noted for their efficacy, efficiency, and cost effectiveness. The use of whole exome sequencing has greatly accelerated the identification of novel causative genes, and its utilization, its use has led to genetic resolution in 25 to 50% of patients, underscoring its impact on clinical diagnostics. And when we look at the whole genome sequencing, and we can definitely say that it has increased the yield with the diagnostic rate varying per disease group by extending, of course, the sequencing coverage, uncovering non-coding regions, regulatory changes, and structural and copy number variants, which are well known to be difficult to detect by exome sequencing only. So I will just give you a couple of cases to show the benefit of genomic medicine to start with rare diseases. Okay, this young boy starts to develop symptoms of paroxysmal dystonia, which means that certain part of his body would become contorted or locked for a number of times a day from around 2 years of age. And from the age of 2 to 5, the family visited multiple specialists and underwent comprehensive diagnostic evaluations, including radiological imaging, and all resulted with some non-specific findings, unfortunately, and no slam dunk diagnosis was achieved. And sorry. Yes. So, and during the course of 3 years of period, he had heaps of, different type of tests done, like nerve conduction studies, like biochemical tests. He had muscle biopsies done. He had some genetic tests, but still no diagnosis was achieved. By the age of 5, whole exome sequencing came to the scene, and so he was enrolled in one of international exome sequencing projects, and the significant gene change called PDHA1 was found. So the effect of the gene change on PDHA1 was studied by extra work later on. Okay, so we all know that PDHA1 is important because it produces an important subunit for a complex called the pyruvate dehydrogenase complex. So the functional activity was studied and found that the activity of the product of this gene was actually lower than what you would expect. So the team could give a sure further diagnosis of pyruvate dehydrogenase deficiency. So now the way this child presented would have been a very atypical way for pyruvate dehydrogenase deficiency to present. It normally occurs far earlier, and a much more severe outcome would have been expected. And so this is an atypical way of which presented, and of course that makes the diagnosis much more challenging. So then what happened? So a comprehensive literature review was conducted, and the results revealed evidence suggesting potential therapeutic efficacy of thymine. So if you give thymine to those patients, sometimes patients would have responded to it. This is what was found. So it was used, the thymine, and so almost immediately he did significant improvement, the boy. And when they increased the dose of thymine, a few weeks later, most of those episodes that he was having had stopped, and he walked home from school for the first time. And as you may all know, thymine is one of the water-soluble vitamin B. So I assume that is a very remarkable case. So I have a second one This is also interesting. So this gentleman, is in his early 60s, and he was referred with a diagnosis of Martolami, which is a very rare disorder, and he was given the diagnosis when he was 20. So, but when further examined in his 60s, the physicians didn't think that he had Martolami syndrome. Therefore, they did some more testings to make sure that it wasn't. And the tests were standard tests, such as urine tests, but the test yielded unremarkable, results. So you can see here that he had some abnormalities in his fingers, in his toes, in his skull even, and also, an abnormal morphology in his fingertips. So further doing some genomic sequencing, a gene change was found actually in a gene called Notch2, and so he actually had a condition called Hatch-Cheney syndrome, which is a very, very rare condition. What is remarkable here is that he had the diagnosis for 40 years, and he had lived with it, and doctors acted a certain way based on this diagnosis. But when he's in his 60s, he actually got the actual diagnosis thanks to the genome analysis. So this is important. Why? Because the initial diagnosis was considered to be an autosomal recessive condition, but the actual, disease that he's suffering from is an autosomal recessive one. Sorry, the first one was an autosomal recessive, but the current one is an autosomal dominant condition. So that's important because when you are canceling the family and identifying who should be tested in the family or to be offered to be tested in the family. So this is also a remarkable case. Okay, we will look, this is an interesting paper that I wanted to share with you, besides the genomic studies. Duchenne muscular dystrophies is the most common form of muscular dystrophy. Typically, affects boys due to the mutation in the DMD gene located on X chromosome. So symptoms like early onset of weakness are seen at the age of 4 or 5 that progresses fairly rapidly. In this study, a few cases remained undiagnosed despite full sequencing, of DMD gene that they performed. They also performed, MLPA for copy number variation studies, and even they did full exome sequencing. So as a next step, so they conducted a whole genome sequencing. So in the image at right, a small fraction of the gene show missing reads, suggesting a deletion in an intron, which seems less likely to be causal. Additionally, there is a complex rearrangement, an inversion, where an exon is flipped around, causing a frame shift. So this leads to the complete loss of dystrophin, RNA, and protein in the affected individual. So in summary, I would say the inversions breakpoints are distant from the exon, making them undetectable in exome sequencing. Additionally, the inversion preserves the exon copy, rendering method like MLPA ineffective. Only whole genome, sequencing would have allowed for comprehensive analysis of such complete rearrangements. So, I would say, despite the genomic advances, a large fraction of patients with rare diseases still remain undiagnosed, as you know. So toward bridging the gap, integration of other omics data with genetic analysis has proved to provide insights into mechanisms that would have remained elusive if genomic approaches had been used on its own. So, let's have a look to this one. This is also a nice study. As we have mentioned, in the last few slides, the genomic sequencing is very much increasingly in routine approaches for Mendelian disease diagnosis. So, but despite the success, it is well known the current diagnostic rate for genome analysis diagnostic tool for rare diseases, as it is the case, in this paper. So the RNA was used for genetically undiagnosed muscle disorders. What they have done, they identified a recurrent de novo intronic mutation in a collagen gene called 6A1, which significantly exacerbates the severity of the disorder compared to other patients. So what is important here is that this mutation appears to be muscle specific and is very, very rarely observed in fibroblast RNA sequencing. So, which means that it is important to use the appropriate tissue for accurate diagnosis. And the study also showed that the systematic application of transcriptome sequencing in rare disease diagnosis, revealing variants missed by conventional diagnostic methods. So, of note, of course, the mutation was not detected by whole genome sequencing because due to its intronic location and failure to disrupt or create a splice site according to standard predictive, prediction algorithms. Similarly, exome sequencing, was performed, but it proved ineffective as the variant fell outside the exome capture region. So, furthermore, we can say the study demonstrates RNA sequencing efficacy in validating splice-disrupting mutations and identifying splice-altering variants in both exonic and deep intronic regions. And, interestingly, they achieved an overall diagnosis of 35%. Okay, so genomic diagnostic typically achieve a diagnosis in 25 to 50% with suspected cases. And on the other hand, transcriptomics, yes, a number of studies show that the transcript levels may not fully mirror protein expression levels. So therefore, the need to explore and develop, an alternative omics such as metabolomics, or epigenomics or proteomics come along. In order to, of course, to improve disease diagnosis for rare diseases, in order to identify new prognostic markers, in order to identify novel treatments and drug repurposing opportunities, and also to study disease pathophysiology. Okay, let's talk about the metabolomics a bit. So what is metabolomics? It is the comprehensive study of an organism metabolites. And it is a useful tool for, for detecting biochemical changes in early disease onset and an opportunity to identify predictive disease markers for quicker therapeutic interventions. And additionally, looking at metabolomic can bring a connection to not only what's happening endogenously, but also exogenously, which means, with diet, environment, even the microbiome. Right, well, metabolites, they are diverse, bioproducts. They are involved in various biological processes, such as signaling, such as regulation of cell cycle, energy conversion, and so on. And, there are around 2,000 hemostatically maintained small molecules in human blood. And there is a diverse range of molecules. I mean, the chemical properties and of all those molecules are really quite extreme. So there's really not only one technology that allows to detect, all this diversity. So mass spectrometry and NMR spectrometry are the techniques most often used for metabolome profiling. Okay, so of course, when we are talking about metabolomics, we have to talk about biomarkers. So what is a biomarker? The biomarker is a traceable substance or characteristic of the body that can be measured as an indicator of biological processes. And so they are discovered using omics-based technologies as well as imaging technologies. So I will give you examples here. When we look at bigger picture, we rely on biomarker types that rather reflect physiologic functions, like blood pressure, which is, of course, normal for every living being, living organism, And then we just see an excess of value in hypertension, for example. And so we have histologic biomarkers like Philippine staining in Niemann-Pick-C, that the abnormality of a biochemical phenotype can be seen under a microscope. We have imaging, very important in dynamic diseases such as cancer. And we have radiographic tools, as biomarkers. And the main, the one that I'm going to focus is molecular biomarkers. So they can be enzymes, proteins, nucleic acids, such as DNA, RNA, and metabolites. And so the enzymes and metabolites are easily accessible in blood, therefore they're important as biomarker. Okay. So now I'm going to talk about the interconnection with genomics and metabolomics. As a scientist working on metabolic disorders, I'm going to give you examples from inborn error of metabolism, and then I will carry on with Fabry and Gaucher diseases. So metabolomics analysis of plasma or urine samples serves as reliable proxy for the biochemical status of an organism, because they can offer minimally invasive procedures which would be preferred for rare disease diagnosis or monitorization. And I would start saying that whole exome sequencing and metabolomics together made significant progress in diagnosis and also functional exploration of inborn error of metabolisms, IEMs. So early detection and diagnosis of IEMs are important for the management of metabolic crisis, of course. And newborn screening, or maybe many of you in your countries, you are using extended newborn screening programs. Well, they become very, very important, and they are in use in many, also in many countries, and they are particularly successful for IEM diagnosis. So the problematic thing is again the clinical and genetic heterogeneity of IEMs. And that can often lead to confusion in choosing the appropriate diagnostic tool, diagnostic test, well, unfortunately, they can delay the treatment. But the biggest advantage offered by the omic approaches and the combinatory approaches, genomics, metabolomics, would allow, collection of a comprehensive amount of information of the gene, pathway defects, and metabolites and enzyme status, which can accelerate the diagnosis and treatment of new, inborn error of metabolism patients. Okay, so I will talk a bit about Fabry disease, and this is the most prevalent lysosomal storage disorder caused by pathogenic variants in the gene called alpha galactosidase A, GLA, and these variants would result in loss or decreased function of the encoded protein enzyme. So this enzymatic defect leads to the accumulation of lyso-GB3 or GB3 biomarkers in biological fluids and tissues, followed by the progressive renal failure, cardiomyopathy, and early stroke, gastrointestinal problems, etc. Fabry disease differ, from other lysosomal storage disorders because, Fabry disease patients remain clinically asymptomatic during the very first years of life. The first clinical symptoms progress over time and eventually lead, to organ failure. So what we did, what has been doing is that in addition to GB3 and lyso-GB3 accumulation, the use of metabolomic approaches identified additional glycosphingolipids in urine of untreated Fabry disease patients. Contributing, of course, to better understanding the pathophysiology. I want to talk about two different, treatment or therapies in use. The first one is enzyme replacement therapy via infusion of the recombinant functional enzyme, which can improve quality of life and alter the disease progression. And the second one, the genotype specific chaperone therapies are in use now, and they are available and offer definitely a great promise. So I believe the success will come when we start using genomic and metabolomics data, or approaches together. And as you may you have the multi-omic biomarkers, you have the genetic diagnosis, but unfortunately, they might support, in some complex and unclear cases, each other, but there are some cases that they do not. So what we do then? You might also know from your own practice the genomic diagnostic. Of course, you can kind of look at molecular results and then assign a potential diagnosis to a patient. But however, the phenotype is often not very predictable for the genotype, and even for the very well-known, rare disease like cystic fibrosis. We know the most common mutation either would lead to prenatal termination of the pregnancy because the fetus has severe problem, or we have individuals with the same homozygous mutations that go on to their own children with cystic reproduction. And so, the mere genotype doesn't explain the phenotypic variability. So therefore, along with alpha-gal A activity, which is the commonly, measured for that, biomarkers like GB3 and related glycosphingolipids in plasma and urine need also to be analyzed. This is the key theme of various diseases, such as also in patients with Gaucher disease. Very common Ashkenazi mutation, which is N409S, is seen very mildly in some homozygous cases, individuals, while often others, suffer from severe hepatosplenomegaly with both crisis and full picture of Gaucher disease, which requires, massive treatment. Along with GCase activity, glucocerebrosidase activity, biomarkers such as, chitotriosidase need to be studied. So the precise diagnosis is based on genomics, but is not sufficiently explained by the genotype. Therefore, the other layers of predictive medicine are definitely needed to fill the gap. And Centogene, I'm sure that you all know this is a very well-known company for metabolomics. I found it interesting. This is why I'm sharing this. Centogene reported an increased diagnostic yield that when combining genotype and biomarkers in Gaucher disease, as the diagnostic yield is reported to be up to 25% through the combination of two layers, which are genetics and metabolomics analysis for biomarker measurement analysis of lyso-G1. Okay, what we do in our laboratory, this is the workflow for multi-omic analysis of rare diseases we use in the lab. So to conduct an impactful study of multi-omics and rare diseases, the careful planning need to be, is crucial. So we collect the biological samples from individuals affected by the rare disease and appropriate, of course, control samples. We use standard protocol for sample handling and storage. We extract the DNA, RNA, or other relevant, biomolecules from the collected samples using optimized procedures. So we perform sequencing by Illumina nanopore, which is currently in use in my lab, and Sanger sequencing. And we do also perform metabolomics profiling using mass spectrometry. We then integrate genomic and metabolomic data to unravel metabolic dysregulation underlying rare diseases. And then this is further that, the interpretation is done and the report is given. Okay, I'm going to give you an example, which is quite also interesting example. And this was a male born in 2015. He had neonatal onset of progressive cerebellar ataxia. He had cholestasis, hepatic failure, and mental deterioration. He had no family of disease, and clinical and paraclinical observations were suggestive of an inherited metabolic disorder. So it was suspected, Niemann-Pick C. And so what has been done is genetic testing and biochemical testing, have been performed. And genetic testing revealed, 2 heterozygous variants in Niemann, the NPC1 gene, and one was already known, but the other one was not described so far. So it would have been classified as a WUS, what we decided to do is subsequent biomarker testing. And so the subsequent biomarker testing showed pathologically increased level of lyso-SM 509. And so the second variant pathogenicity was proved with biomarker analysis. So this is how the genetic and biomarker evaluation has been performed. Okay, so what is in the future? We have all the clinical informations, we have all the imagings, MRIs, we can do biopsies, the pathology, we can do the biochemistry, we can measure thousands of different metabolites in blood, in urine, we can definitely look at, the whole genome. We do that now, routinely, and also transcription analysis. But let's not forget there are also issues or domains like the epigenome, the microbiome, or the proteome, and of course, environment. So all this avalanche of informations, there is no way that we can handle all that information on our own. So manpower is not enough to do this. So where do I see the future? All the scientific information will have to go through artificial intelligence, machine learning, and the way I see that is these will not be applied with each of these domains individually, but all together by eventually developing a common language that we can share and exchange information among the different domains and provide a final answer. Okay, who we are? We are a team of clinicians and researchers working together in the Pediatric Nutrition Metabolism Clinic, in a Nutrition Metabolism Laboratory, and Rare Diseases Research Laboratory. And we have 2 PhD programs running. One is metabolism and the other one is rare diseases. And I would like to thank you. And if you have questions, you can always use those emails below. And this is it from me. Thank you.
Thank you very much, Professor, for such a wonderful presentation. And we learned about the data that are already collected here and worked. And I was already surprised because we now have proteomics, metabolomics, but we have our phenomics. Every time as science is advancing, we have new terms, as I think. And of course, now I'm going to check for the question, if there is any question. Yes, there were 2 questions because we have part of the academician of a student in a conference hall in our campus here. So some of them are not here because they are attending in hybrid form. And let's check for the first one. Maybe you can check it on the chat part. Professor Fatmahan, is the rare disease ecosystem in the rare disease ecosystem, how do you envision the integration of genetic diagnostics and metabolomics data to elucidate novel disease pathways and identify potential therapeutic targets, and what computational or analytical frameworks are essential for extracting meaningful insight from these complex data sets? In fact, you, I think, mentioned a little bit, but if you would like to add something.
I think I have mentioned it,
yeah.
And we are also currently working, you know, on that particular project involving new discovery of new biomarkers, along with, you know, the genomic data. So, I mean, this can be, of course, you know, I mean, research is going on, and many discoveries are there. And so the spectrometries, NMR, you know, I mean, through the help of those machines, you know, I mean, you can try to discover new biomarkers and integrate it, you know, together with the genomic or whole genome data.
And I will check for other questions. I think that the other one already is related with machine learning. And
yes,
given the heterogeneity of...
We do use machine learning. Yeah, for clinical, for genetic, and also metabolomic data. We do use machine learning, well, I mean, for the early, you know, predictions.
Yes, and if you have, for the last one, as we look toward the future of rare disease research and diagnostic, what emerging technologies or methods show promise for enhancing the sensitivity, specificity, and scalability of analytic system in characterizing rare diseases? And how do you anticipate overcoming the barriers of cost, accessibility, and interpretability of, in fact, to ensure the equitable access to cutting-edge diagnostic tools and therapeutic interventions for patients with rare and undiagnosed conditions?
Yes, the undiagnosed rare diseases, they are, well, I mean, they are the ones that we are currently focusing on in Turkey. And because, but the first thing is, of course, you know, integrating into international platforms, data sharing, and is important. We need to have, you know, I mean, in Turkey, unfortunately, you know, I mean, this is, well, unfortunately, it is missing, you know, the data sharing, and also, you know, I mean, we have to have, you know, I mean, directories. We have to have system in the health ministry system. We have to, this rare disease should be integrated, and also, you know, in my case, in my country, you know, there are so many things missing as far as the health ministry, you know, I mean, is concerned, their tasks. And so us researchers, we are trying to do our best in order to, you know, I mean, to give diagnostic for rare disease patients and early diagnosis. So, I mean, this is what, you know, I mean, we are working on. Early diagnosis is a must, but the rest is with the Ministry of Health, unfortunately.
Thank you, Professor. And one question I would like to ask that, you know, everybody talk about the DNA bank laboratory. Is there any formation or establishment of rare diseases DNA bank, or the collection of the samples that, or policy?
Yes, I think we have talked with you. I've been talking about that for 20 years, DNA banking, yes, but no, there is no DNA banking. Unfortunately, in a country with more than 5 million, you know, rare disease patients, unfortunately, we don't have one. And but you know, each laboratory has got their own small DNA banks, which they are not willing to share with others. And so, but I mean, as I said, you know, I mean, we need to have a national DNA banking, you know, I mean, coordinated by Ministry of Health, coordinated with, by state universities, and, under the European regulations. But unfortunately, I have small biobank in my laboratory for my rare disease patients, and I'm ready to share with, with anyone who was willing to work or collaborate. Yes.
Thank you, Professor.
Thank you very much.
Yeah, thank you. Is there any other question from the speaker here? Professor Ergüven, do you have any other question? Or Peter or Rebecca that is already here?
No, thank you very much.
Thank you.
I thank you very much and good luck. Bye-bye.
Okay, thank you, Professor. Okay, now we will continue with our next speaker, and I will share the presentation for our next speaker from Philippines. It's very far away from Turkey. And yes, welcome, Professor Peter, and nice to see you here. So I just would like to give a brief information about you. We prepared for all of our speakers. So today, Associate Professor Dr. Peter James Gann will be with us, and his presentation is related with the gene editing technologies and medical applications. When we check about the education, we see that Professor already has a bachelor degree from animal science from Marcos State University, and has a PhD again from the University of Philippines, Los Banos, from animal breeding and genetics, and the PhD from University of Arkansas from cellular and cellular molecular biology. Professor academic career, he started as an assistant professor at Marcos State University, continue as a research associate and PhD at the same university, and the University of Arkansas, where the PhD talk about education. And now he continues to work as an associate professor at Cell and Molecular Biology Department since 2023. And beside academic career, we see that Professor has a managerial experience also, has worked as a project leader at specific project evaluator, project leader, like in the research services Los Banos, Philippines, as a project leader at the Marcos State University for 3 years, and already as a project manager at the same university. And he already continue for another project that, of course, we cannot include all of them here. And when we check about the field of research of Professor Peter, we see that he is interested in life sciences, natural, and especially bioinformatics, biotechnology, genome editing, and CRISPR-Cas9 technology, which is one of the latest and recent technologies now that scientists are working on. And besides academic and managerial experience, I would like to mention that Professor has a lot of awards and honors. And of course, I can just mention some of them because there are a lot, as you see here. Second place in graduate student oral competition, biotechnology oral presentation, excellence in research award, graduate student mentor award, and a lot of. So if you are interested in research activity of a professor, you can check from the news here that we already included here, and maybe Professor is going to mention about his research field. So I would like to thank again for being here and accepted our invitation, like all of our speakers today, and welcome again to our symposium, Professor, and now is your stage.
Well, thank you so much, Prof. Arta, for that very comprehensive presentation about me. And I would like to start by thanking you for inviting me in this conference. Hold on, just give me a bit of time to figure out how to get this in a full screen. I don't think. Yeah, I think I'm going to stick it this way, and it's going to be a single scroll because my presentation is actually in the form of PDF, but I hope everyone can see it. Is it?
Yes, it's okay.
It's okay. Okay, so moving forward, my presentation for tonight here in the Philippines, I know it's the afternoon in Turkey, is about all the genome editing technologies and the corresponding medical applications. So to capture the interest of everyone, I would like to start for you to discuss the reasons why we do gene editing. But before that, I would like to emphasize that when we say gene editing, we are actually talking about modification of a certain genome in a way of doing insertion, we could do deletion, or we could modify certain bases. And our purpose for genome editing is actually outlined in my presentation. As you can see to your left, I have enumerated here 4 reasons why we do gene editing. First, of course, is that if we want to tackle or defeat diseases, particularly those that are genetic in nature. An example that I have provided here is actually your sickle cell anemia, where you can see that a single mutation in the DNA sequence, when we have a replacement of your A into T, would actually result also in a mutation in the corresponding amino acid from your glutamic acid into your valine. And because of this mutation that we have, you can see from here that the shape of the red blood cell actually changes from being spherical into a sickle cell type. And whenever the shape of the blood or the red blood cell is actually in a sickle cell type, what happens is that they clamp together and it comes up into several symptoms. And an example of that is kind of like lower oxygen in the body. And then another example of that is that there would be clamping and then there would be less movement of blood all throughout the blood vessels. So in gene editing, this is one thing that we could address. And then aside from that, the second one is that, as you can see from this timeline, you can see that in the earlier times, the life expectancy is very low. But with gene editing, if we can actually tackle certain genetic disorders by actually changing the genome of humans, then there is a possibility that we can actually expand or extend our lifespan. And then in addition to its medical uses, I would like also to emphasize here that with genome editing, we can actually produce healthier plants that we can actually use for us to have a more holistic being. And then lastly, you can see in number 4 that we can actually also improve the characteristics of animal food sources that we have by improving their growth. So you can see from here that at the same age, say for example, for this one, they were grown for 50 days. You can see that when you compare it from 1957 and the size of the chicken in 2005, after growing them for 50 days, you can see actually a very significant difference in the height. Well, we know that the use of gene editing actually has several advantages. But on the flip side, we can see on your right screen some of the limitations or some of the disadvantages of using gene editing as a technique. And I would like to start with the ethical dilemma. So one thing that we do understand is that there are so many movements all over the world that don't like gene editing, and they consider anything that's gene edited to be unfit or something that we should not be using because of certain concerns that they don't like. Aside from ethical considerations, the second limitation of using genome editing as a technique is about safety concerns. Well, people are always very idealistic about gene editing, and we always think, oh, it seems like a very perfect technique. But one of the concerns of gene editing is that since the human genome is actually a very big genome, when you try to target a certain sequence, there is a possibility that aside from hitting your target, you will actually also have your off targets. And these off targets that are actually being hit could also be very important or very essential for certain metabolic processes. So you might have fixed a certain genetic disorder, but you might be hitting other proteins or other genes that codes for proteins that are also essential for certain metabolic activities. And then aside from that, number 3, another concern is that, evolutionary-wise, we actually like diversity. And when we do gene editing, people are actually claiming that we are actually decreasing the diversity and lessening our chance to actually survive as a species. And then lastly, when we say gene editing, we would know that this technique, if it's applied in medicine, it's going to take a lot of money from us. And people say that it's also unethical. It's because most likely when we offer this therapy to our patients, there's a possibility that only those who have higher income can kind of like, go into this kind of treatment that they can use to treat the diseases that they have. So moving forward, I would like to show to you the gene structure as I will be mentioning to you certain basic techniques that we use in gene editing. But in order for us to be able to do that, it is very important to understand the structure of the gene. And you can see from here that the gene is basically just consists of two main components. So on the top, you can see here on your top right, you can see here the structure of the gene, which has two parts. We have the promoter sequence, and then we have the transcribe region. So it is understood that in eukaryotic organisms, for the transcribe region, we have two parts. The ones that are in gray are actually your exons or your coding sequences that codes for your protein or your mRNA. And then for your introns, these are the ones that are actually spliced out. And this particular transcribe region is the one that becomes your enzyme that will actually be coding several activities in the body. And then aside from the transcribe region, the switching on and off of the gene, this will come into play later. It's going to be very important for you to understand, is that in the upstream region of your transcribe region, we actually have the promoter region. And this promoter region is kind of like the switch of the gene. It tells how many transcription factors will bind to it, and it explains to you how a gene can be activated, switch on, or switch off. So when we want to edit genes, the next questions that we have to ask is, what genes do we want to edit? So I am presenting here for you like 4 classes of genes. So first, if we want to control the transport of certain molecules, then we could consider genes that are coding for several transporters. Aside from that, if we want to control or facilitate digestive activities, then we can go for the enzymes that are coding for certain biochemical pathways. And then lastly, or thirdly, if we want to alter signaling pathways, then we can actually code for the genes that codes for receptors in a signaling pathway. And then we have number 4 here. If we want to regulate the expression of the genes, then we could actually consider the pathway that is actually responsible for the transcription process. So moving forward here, we can actually see the type of regulation that we can do with a gene. So when we want to do gene editing, we have 3 different options. We can actually do a knockout, we can actually do an overexpression, or we could perform downregulation. Now, it's very essential to understand here that before we start with any gene regulation, it is very essential for everyone to understand the significance of a gene that you are trying to edit. So I would like to start with the knockout here. So it has been known from literatures that we have a gene called PCSK9, which is actually a protein that's found in the cytoplasm, which actually bind to an LDLR. So LDLR is a lipoprotein receptor. And what happens here, if we have a binding of PCSK9 with our LDLR, it actually takes lipoprotein are actually taken by the receptor, and then it forms an endosome. And with this particular process, what happens is that the fats are actually going to be dispersed in our bloodstream, thus increasing the cholesterol level inside the bloodstream. So what happens here is that what we want to do if we have this particular thing that's happening is that we actually want to knock out your PCSK9 to actually avoid the process of binding with our receptor that's actually increasing the cholesterol level in the blood. So that is an example of a knockout. And then next, we have number 2, we have overexpression. So when do we want to overexpress? So in certain conditions, say for example, in your diabetes, or in your liver or your pancreas is actually having a failure in the production of insulin, there is a possibility that we can design a construct that could be brought into the cell that would actually allow it to produce insulin. And in that way, if we produce insulin, then we can actually take a cell that's kind of like defective of not producing insulin to producing insulin, such that we have binding to the insulin receptor, and then we can bring in glucose inside the cell. So that is an example of overexpression. And then we have here an example of your downregulation. So we have a gene which we call as your TEM8, which codes for a protein that has been considered as a biomarker in cancer. So it has been found out in ovarian cancers and in breast cancer cases that this particular gene is actually overexpressed. However, aside from being a biomarker for cancer cells, it has been found out that it is also important. This gene is actually very important for differentiation. And aside from that, it's also important for cell adhesion. Then, because it is essential for certain activities, we don't want actually to knock out this gene. So therefore, it is very essential to consider downregulation. So when we do gene editing, these are the options that we can do. We can perform knockout, we can go for overexpression, or we could actually do downregulation. So moving on, when we want to do gene regulation, there are actually different options that we can do, and I have presented here 4. Of course, we have the original genome editor. We are all familiar with this. The first one is the use of restriction enzymes. And then the older genome editors that we've been used in the last decade, we have your Zaphens or zinc finger nucleases, and then we have your TALEN, and then the new platform for gene editing is your CRISPR-Cas9. So first, I would like to start with your use of restriction enzymes. Well, the restriction enzymes are actually very simplistic. They actually cut a certain sequence in the genome. An example that I have here of an endonuclease or a restriction enzyme is your EcoR1. And then you can see that EcoR1 cuts this particular double-stranded sequence, which is your GAATTC. And what happens is that when you cut this, you actually produce 2 sticky ends. And with these 2 sticky ends that you have, you can actually use this opportunity to actually insert a particular gene of interest that you want to insert into your cell. So in this example that I am showing, there is a gene that is found in your firefly, which is called as your luciferase. And what your luciferase does is actually it catalyzes the conversion of your luciferin into your oxyluciferin. And with the aid of ATP, oxygen and magnesium, what happens is that the conversion of your luciferin to oxyluciferin actually also yields light. That's why you see light from your fireflies. And we can actually take this opportunity to actually put these sticky ends into our luciferase through the process of nested PCR. And with that, we can use a ligase to bring this luciferase into our gene. An example of that one that I'm showing in here is actually a plant that is actually showing a luciferase expression. So if you put luciferin in this plant, since it is actually expressing luciferase already, then it can catalyze this process. So when you provide the substrate, then what happens is that it will convert luciferin into oxyluciferin. And then in addition to that, it will actually be emitting light. And the significance of this one is if we want to track certain proteins, if we want to track the movement of certain enzymes, we can actually tag them with luciferase, with mCherry, or we could tag them with GFP, and we could figure out where they are localizing in the cell or where are they localizing in a particular tissue. So aside from the use of restriction enzymes, it has also been introduced, like in the older times, the use of zinc fingers and your TALEN. So these 2 techniques are actually using the same enzyme to actually cut the DNA, which is actually your FOC1. Now, the disadvantage of, I'm not saying these are bad. I'm saying they are still great tools that you can use for genome editing. But the problem with zinc finger nucleases is their versatility. So when you design a platform for zinc fingers and for your TALENs, you actually have to engineer proteins. And these proteins that you are going to engineer are actually corresponding to a particular sequence or a particular nucleotide that is actually found in your DNA sequence. So as you can see from this one, we have the components of your zinc finger. We have the finger 3, the finger 2, and then we have the finger 1. And for each of the fingers, they actually correspond to certain sequences. And with this, since these are actually proteins, engineering this one actually requires a lot of time. That is why when you have this platform and you want to use any other target, it would actually be a lot of work. And now it has been introduced in the last decade. Like we have your CRISPR-Cas9, which is considered to be the most flexible. And once you have started the platform, it is actually very easy to use. So all you have to do is to have your plasmids ready. And when your plasmid is ready, when you have constructed it, then it's actually very easy to be modified. And then it can fit the targets that you are actually wanting to edit in the genome that you have. However, the limitation to this one is that the platforms or delivery systems for different organisms are not actually the same. So if you want to work on plants, if you want to work on animals, or if you want to work on microorganisms, we actually have different platforms or delivery systems that we actually use. So moving forward, we have here the advantages and the disadvantages of CRISPR-Cas9. So to your right, I have enumerated actually three pros or three advantages or benefits of using CRISPR-Cas9. First is that it is actually very simple to amend. So we actually just have two components here. When I say two components, we have our guide RNA, which is actually corresponding to the target that we are wanting to hit or we are wanting to edit. And then we have the enzyme that's gonna perform the cut, and that's actually our Cas protein. Aside from being very simple and very flexible, another advantage that we can have is that it can actually be multiplexed. So as you can see from this illustration that I have here, you can see, I told you a while ago that the guide RNA actually corresponds to the target. So from this structure or from this figure I'm showing to you, we actually have two gRNAs, indicating that we are actually trying to hit two targets with just one construct that we have. So aside from being capable of multiplexing, it is actually also cost effective. So I told you a while ago, when you have the plasmid, when the end, you have your endonucleases, so you can actually do the cut and paste. And then the cut and paste that you perform, that's actually gonna help you customize your CRISPR-Cas9 experiment. Unlike with the use of zinc fingers and the use of talents, I've told you a while ago, it actually requires protein engineering, and it's actually a very tedious process. Now, we have advantages, but we also have kind of like some disadvantages in the CRISPR-Cas9 system. And the first disadvantage is the limitations when it comes to delivery. So I will be discussing with you some of the delivery systems that we use for CRISPR-Cas9. There is a mechanical one, we use adenoviruses, but sometimes these delivery systems will have their own pros and cons, and they have their own disadvantages. So that's one, the limitations with delivery. And then the second one is that when you are starting from scratch and your laboratory don't have any platform yet for these kinds of experiments, it's actually very difficult to start with. But once you have already actually established your platform, it becomes very flexible and everything can be very easy. You can hit as many targets as you want once you already have established your laboratory for genome editing. And then the third disadvantage is something that we normally just don't discuss when we talk about gene editing, is that it is not always efficient. So I have mentioned to you a while ago that you want to hit one target, but after hitting one target, you might also be hitting other parts of the genome that you may not know. And most of the scientists that performs CRISPR-Cas9 experiments, they are only validating the edits that are actually happening in the target sequence without actually looking into the background. Because we all know whole genome sequencing is very expensive. So if we actually hit our target, sometimes most of the scientists won't care on what's happening in the background. So this is one of the problem. It's not always efficient. So moving forward, when we talk about CRISPR-Cas9, this is how gene editing actually takes place. So it comes in 5 steps. So when you have delivered your CRISPR-Cas9 construct or your plasmid into the cell that you're actually wanting to transform, there are actually 2 main components here. The top component that you are seeing in figure A is actually called as your seed. And your seed is actually consists of 3 parts. We have your scaffold, and then we have here your guide RNA. And your guide RNA actually corresponds to the sequence that you want to edit in your genome. And then we actually have here your protein that's going to do the cut, which is your Cas9. And then below here, we actually have here your DNA sequence that you are actually targeting. And now I'm introducing you to a new term, which is called as your PAM. So this is kind of a sequence that is first recognized by your ribonucleoprotein complex before it does the cut. So I will be introducing to you how to identify PAMs later as we move forward with the discussion. So when the PAM is recognized by this complex or by the seed, what happens is that it binds to your DNA, it recognizes the PAM, and after recognizing the PAM, it will actually try to fit in the guide RNA that you have to the DNA sequence. And when we have a perfect match, so when we have here extension of the pairing and we have a perfect match of our guide RNA with the DNA sequence that we have here, then the Cas9 will actually perform a double-stranded cut. But I would like also to emphasize here that it's not always necessary that the pairing is actually perfect. There can be some mismatches that can be found in certain locations, and still there is a possibility, even if you have mismatches, say for example 1 or 2 mismatch, then what happens is that the Cas9 can still do the cut. And then as you can see from here, we have partial pairing, and then there is actually no cleavage whenever there is a mismatch. So when we have so much mismatch, say for example 50%, like we have a 22 base pair of target, from this one we have 22 base pairs, and then only 15 is actually matching. What happens is that we have unpairing or uncoupling of the complex, and then there will be no cut that will actually take place. But whenever we have few mismatch, or we have complete pairing of your guide RNA with the sequence, then it will actually perform the cut. So now moving on, I have here listed for you the different steps in identifying the target for CRISPR-Cas9. So now you are wanting to design your experiment, and you wanted to edit, say, for example, the gene that is responsible for phenylketonuria. So we are actually interested in genetic disorders when we do Cas9. So the first thing that you have to do is to actually select a region in the gene for editing consisting of 17 to 20 base pairs. So say, for example, this is the catalytic activity of the gene I wanted to target. I need to identify a part of that particular gene that I actually want to target. But before doing that, it is very important to consider the type of mutation that you actually want to do. Well, if you want to do a knockout, you go for the catalytic sites, or you go for the exons. Because there is a chance that if you go for the exon, what happens is whenever you introduce a mutation, then you will form a protein with a different sequence. It's going to be misfolded, and then that protein is going to be inactive. However, if you just actually want to suppress or make the expression of the gene lower, then you can actually focus on the promoter region. So I have shown you the structure of the gene a while ago. So from the promoter region, you can actually identify certain cis-regulatory elements. And if you try to disrupt them, you can actually downregulate the activity of the gene. So when we talk about Cas9, the type of mutation that you normally get is kind of like unpredictable. Most likely, we normally get a single base mutation, and it is very seldom that you get like a big deletion. Even if you do a cut in 2 sides and you try to drop it off, most of the time we have an immediate repair. And what happens is that in 2 sides, you will just have mutations that are kind of like unpredictable, and you don't know what's going to happen. However, with the advances in gene editing, there has already been platforms for us to be able to introduce specific insertion. So after you have identified the region that you're trying to edit, then the next activity that you have to do is actually to identify the PAM sequences surrounding the preferred target region. So now, in the ones that I have highlighted in the sequence that I have shown you, I was actually thinking of this one as my target. But then you can see from here that for this location, I don't have any PAM sequence. And I have presented to you a while ago that the first step for the editing to take place, or for the Cas9 editing to take place, is that it first recognizes PAM sequences around the genome. And now you can see from here that we have actually a lot of PAM sequences. Anyway, when we say PAM sequences, these are actually your photo spacer adjacent motif. For short, we call it as your PAM. So these are conserved sequences that are found in the genome. And when we talk about PAM, we're actually talking about NGG, although there are other alternative PAMs, but the highly efficient PAMs where your ribonucleoprotein complex will actually bind is actually NGG from the 5' to 3'. N stands for whatever nucleotide, while your G is for your guanine. So as you can see in this illustration, I have highlighted the PAM sequences from 5' to 3' region, like your NGG. So an example would be this one, it's AGG. And then we have this one, it's GGG. And then we have another one here, we have your TGG. Well, you may ask, Dr. Gann, why did you highlight CCA? Then you have to remember the sequence that I'm showing you is a single sequence that's running from your 5' to 3'. So hypothetically, under this particular sequence, we have a reverse sequence that's running from your 3' to your 5'. So therefore, in the other strand, my PAM sequence would actually be my CCA, or the reverse of my CCN, or the reverse of my NGG. That is why, as you can see from here, all the CCNs that's running from 5' to 3' is actually highlighted. So this is a PAM sequence, this is a PAM sequence, and this is also another PAM sequence. And you can see from here that we have 2 PAM sequences. So now, when you have identified your PAM sequences, then the next step is for you to identify your to scan and analyze potential off-targets from the genome. But before that, I would like to emphasize also here that this one would not be a potential target because the cut will actually happen somewhere near your PAM sequences. So I will provide you examples of targets later. So in identifying off-targets, you can actually have several options. And I have identified here easy web tool materials that you can use if you want to identify off-targets in your genome, because you don't want to be hitting genes that are housekeeping or very essential. Like say, for example, cytochrome B, ubiquitin, certain genes that are very important for all biological processes. You don't want to be hitting them. So you can actually use the Cas-Off Finder. So I have the link here. And the good thing about Cas-Off Finder is you can have multiple options for target genomes and also for the PAM type. Another one that you can use is your ChopChop. I've also provided you here the link. You can go visit this one. These are very easy platforms. What I like about this one is that it has a very good visual. I'm a very visual person. So when I can see where my PAMs are and then I can see a lot of colors, I really like that. So that's the good about ChopChop. And then we have your CRISPR. But the problem with the last one is that there's only a maximum number of base pair that you can actually feed in. So if you have a very long sequence or if you want to scan the entire genome, you cannot find the other off targets. It's only very limited to the certain sequence that you're going to input to it. So that's the problem with this one. So essentially, when I try to identify sequences that can be targeted or can be considered as off target, I normally use Cas-Off Finder and I normally use your ChopChop. So after identifying the off targets, and then after identifying the gene that you want to target for CRISPR-Cas9, the next step is for you to identify or predict the cut site and the potential effect of the mutation in the cell. So now, given the sequence that I have provided you earlier, I was actually planning to target somewhere around here. But then I've showed you that this particular region is not containing any PAM site. So I'm not going to be having any double-stranded break. So now, I have to identify targets that are actually very close or very proximal to my PAM sequences. And I have identified here 2 PAM sequences. I have my CCA, and then I will have here my CGG. And actually, when you actually do CRISPR-Cas9, what happens is that the cut site will actually happen, or the double-stranded break will actually happen between the 3rd and the 4th nucleotide from your PAM sequence. So if this is my PAM sequence, I will have my 3rd and then my 4th nucleotide, and the double-stranded break will actually take place in here. And then I will have, if I have my PAM here, what happens is that my NGG will be in the other strand. So therefore, the cut will be happening from the 3rd and the 4th nucleotide that's actually gonna be here. I've told you a while ago that basically, if you think hypothetically and if you look at the sequence, if I did try to do a double-stranded break in this region, and in this region, you are expecting the entire sequence from here going down here to be deleted, right? Hypothetically, we are expecting that. But unfortunately, for CRISPR-Cas9, that is actually very rare. We normally just get 5% of deletion. Like out of 100 edits that we try to do, we normally just get 5% of a complete drop-off of the sequence that we are trying to eliminate or we want to remove. So moving forward, when you already have your target, you have identified your off targets, and then you have predicted the site that you're going to edit, the next step is for you to design a vector. Of course, when you want to design a vector, you need to have a backbone, and these are the options of plasmids that you can purchase for genome editing. And actually, you can just customize them for you to be able to perform your experiments. So in this very simplistic vector that I am showing you, you can actually see several components. So the guide RNA would actually indicate your target sites. And for guide RNA, you can see that we have several components. We have your target site, we have the scaffold, and then we have your tRNA here. And then this guide RNA, when they actually become transcribed into our pre-mRNA, it actually forms this complex. So as you can see, I've told you that we will have the tRNA component, and then we will actually have your scaffold. And then aside from your scaffold, we will actually be having your target sequence in here. And then this will just be an additional backbone. So when we have our tRNA's enzyme inside the cytoplasm, what happens is that the tRNA will actually be cleaved. So as you can see, we have here enzymes that will cleave our tRNA. And then from here, we actually form two complexes of RNAs. One is containing our guide RNA1, and then the other one will be containing our guide RNA2. Our guide RNA1 actually corresponds to the first target that we are trying to hit. And then the second one will actually correspond to the second target that we are trying to hit. So I've been telling you a while ago that we can actually do multiplexing for CRISPR-Cas9 gene editing. And then aside from that, we also have the promoter component in our backbone. So our promoter components are the ones that are in yellow. And these promoter components are actually ones that determines the transcription activity of our different proteins. So we have ampicillin resistance here, a gene for ampicillin resistance, which is our selective marker later. We have our Cas9 here. And then for our guide RNA to be transcribed, we actually also have a promoter sequence here. So normally for promoter sequences, we actually use the promoter sequence coming from genes that are constantly expressed. Example of that is that you can actually use the promoter for GAPDH. So those are certain promoters that you can have to have higher transcriptional activity. And then aside from promoter sequences, we also have terminating sequences here to terminate the process of transcription. And of course, we have your Cas9. So in this part, we have the sequence for Cas9 so that we will be able to produce this component, this yellow component here. This is actually our enzyme that will actually perform the cleavage in our DNA. And then lastly, I think everyone understands this. We have our ampicillin. This is our selectable marker, so that when you grow your cells in ampicillin, then you can select those ones that were transformed versus the ones that were actually not transformed. So moving on, when we already have our plasmid, so what we do is we try to customize our plasmid according to our need and design it according to the targets that we actually want to hit. So as you can see, these components remains the same. And what's actually changing is just your guide RNA. Because like your guide RNA will actually be corresponding to the targets that you want to hit. And that's very customizable. And moving forward, when you already have your plasmid, the next question is, how am I going to deliver this to my cells? So normally for animal cells or human cell lines, we actually have three options here. We could use viral vectors. And for viral vectors, there are other virus options, but normally in the lab, we use adenovirus. And then we have mechanical methods. And mechanical methods are the easier ones. You can use a gene gun, we can do microinjection, or we can perform electroporation. And then we can also use your liposomes, or we can use actually lipid coatings, and then we can deliver that into our cell. And in here, we actually have other options for the viral vectors. We have retroviruses, adeno-associated viruses, lentiviruses. And when it comes to non-viral vectors, we also have other options here. And then the ones that are at the bottom, these are actually your mechanical methods. So moving on here, I have here listed the advantages and disadvantages of the different ways of transformation. So the advantage of viral vectors is the fact that for transformation experiment, we actually have very high rate of transformation when we use adenoviruses or viral vectors. They can infect a wide range of human cell types. So whatever cell type you have, whatever type of tissue, they are very flexible with that. We have very high transduction efficiency. And the thing about it is that there are vectors that can only take a very small insert, but with viral vectors, they can actually take a larger insert. Like even if you have 3,000 base pairs that you wanted to insert into the backbone, you can actually do that. But then aside from the advantages of viral vectors, we also have known disadvantages. So for viral vectors, since they are known to be non-integrating in the genome, then the expression can also be just transient. And in the succeeding generations, it will actually just disappear. And then aside from that, by using adenoviruses, there is a possibility that you actually, because you are introducing a foreign material into the cell, what happens is that you can actually generate an immune response. And an example of that is chronic inflammation. For non-viral vectors, I actually focused here on the use of liposomes. One advantage is that they are easy to prepare, and there is no restriction in the size of the DNA that you can actually integrate, unlike in viral vectors, although they can take large inserts, but it could be limited to 5,000 or 6,000. If you go longer than that, it's going to be very difficult to do an insert. But when it comes to non-viral vectors, for whatever size, you can actually move ahead with your editing. And then the disadvantage is that the efficiency of the gene transfer is actually very low, and sometimes we have very low rates of integration. And then lastly, we have your mechanical methods here. For mechanical methods, I'm actually talking about the use of your electroporation. And aside from your electroporation, I'm also talking about the use of your gene GANN. So for the gene GANN, one advantage is that it is kind of like safe and very simple to use. But then the advantage of the gene GANN is that since you are putting so much pressure on the cell, there is a possibility that you can cause damage in certain cells. And then when you try to grow them, when you try to culture them, some of your cells would actually die. And sometimes the problem with the use of the gene GANN or biolistic method is that you can also have lower efficiency. And for the second method that is considered to be mechanical, we have your electroporation. The advantage of your electroporation is that the amount of processing and the amount of performing the transformation is actually in a very short time. But then the disadvantage is that since you are opening the cell, you are exposing it to the environment, and there is a possibility that when you open the cell, it can take in foreign materials, especially if you don't perform it in an environment that is actually very clean and free of any foreign material. There's a possibility that you are taking in additional genetic material or other foreign materials, which could actually result into the death of your cell. So now I'm moving forward to the medical applications for gene editing techniques. So when we talk about gene editing techniques, there are actually two methods that we can use. First is your ex vivo, and then the second one is your in vivo. When I say ex vivo, you take out a cell from a patient, and from that cell that you have, you actually perform your transformation. And after you perform your transformation, then you put it back. The advantage of this one is that you can actually have a safety check, and you can make sure that actually nothing is happening in the cell before you put it back into a particular tissue. But then the disadvantage is that it is considered to be more invasive because you are taking a tissue from a patient. Now, for in vivo techniques, what happens here, particularly when you do Cas9, we can actually do in vivo. The advantage of this one is that less invasive, but the problem with using in vivo techniques, like bringing in all these vectors inside the cell, is the fact that when you use in vivo, there's no possibility of safety check. And we are also decreasing control over our target cells. So when we do CRISPR-Cas9, one of the medical application is actually in your diagnostics. So we all know that COVID happened, and some of the diagnostic kits that we are using for the detection of COVID actually uses the CRISPR-Cas9 system. We have here the to the upper part, like from this one, we have the Sherlock platform, and then to the lower part, we actually have the detector platform. So what happens here is that the RNA is actually extracted from the patient, and then inside our tube, we actually have a guide RNA, and then we have our Cas9. So our guide RNA, which is actually fused with our Cas9, when not fused with a corresponding sequence that is actually complementary to it, the Cas9 is actually inactive. So you try to imagine that if we don't have this sequence here that's coming from our patient, the Cas9 is actually inactive. But with the presence of an RNA that will actually interact with our Cas9, what happens here is that the Cas9 becomes activated, and then inside the tube, we actually have these RNA probes, which contains a protein that fluoresces, and then we have here a quencher. So what happens here is that this particular sequence is actually being cleaved also when your Cas9 is attached with a material that's coming from the virus and it corresponds with our guide RNA, it will actually cleave the RNA probe, and by removing the quencher, we actually can observe fluorescence. And this is actually placed in a strip, and then it's like a simple like a lateral flow assay, and you can see that when it's negative, all these fluorescence probes will actually be producing one band, while if it's positive, you will be producing two bands. So it's actually the same system that they have for the detector. However, for the detector, it actually has it's not actually in the form of a strip, but it actually measures the fluorescence. So you have to put it in a machine or a fluorometer to determine the fluorescence, and a certain fluorescence signal would actually indicate that you are positive for COVID. So these are just two examples of a platform that actually uses Cas9 for diagnostic tool. But the limitations of using this one is that it requires higher expertise, particularly for the detector method. And then aside from that, it still requires sophisticated equipment, because still with your extracted RNA, you need to perform amplification before you can proceed actually to the downstream processes. So you cannot actually just perform all of this in a point of care kind of like level of perspective. So it should also be performed in the laboratory. So aside from the use of gene editing or CRISPR-Cas9 in diagnostics, so in general, gene editing can be used to gene augmentation therapy, and it can also be used for targeted killing of specific cells. So when we say gene augmentation, we are talking about augmenting a certain allele that's not actually producing a normal protein for a certain biochemical pathway. An example of that is in your phenylketonuria, which is autosomal recessive. So what happens here is that when your gene is autosomal recessive, you would want them to introduce the dominant allele. So what you can do is you can actually take the dominant form of it and then introduce it to your disease cell. And in that way, you can actually augment the problem with a person with a particular genetic disorder. Another example of medical application of gene editing is actually the targeted killing of your specific cells. So when you try to target specific cells or you want to kill them, this is particularly applicable for cancer gene therapies. You can actually introduce a toxin to target cells that you want to kill. Example would be your cancer cells. So you can actually directly introduce it. But another way to go around killing specific cells is by way of introducing a prodrug. So when I say a prodrug, it is actually a gene that you try to introduce to a cell. And then when you try to introduce a particular compound, that particular compound will be transformed into something that's going to become toxic to your cell. An example of something that you can introduce is your TK or your thymidine kinase. And with the introduction of thymidine kinase to your cell, you can then introduce your ganciclovir. And with the introduction of your ganciclovir with your prodrug, what happens is that you produce a compound that's going to be toxic to your cell, and then your cell dies. So when we talk about targeted killing of specific cells, there are also other ways. You can actually take advantage of the immune response. When I say introduction of something that uses the immune response, what you want to do is you want to introduce materials in the cell that would actually trigger the immunity of a particular organism to attack those cells, and then they're going to get killed. So an example here is the introduction of a foreign antigen into your cell. And if you can introduce that, then your immune cells are gonna attack your cells of target, and then they're also gonna get killed. In addition to that, you can also introduce cytokines into your cells. And with the introduction of your cytokines, the immune cells will also attack the cells with your cytokines, and then they will also be killed. So another medical application is actually, there are other medical applications aside from gene augmentation and targeted killing of the cells. You can also use it for, I think it's, I had something wrong with my presentation, so it was kind of like repeated. So I think I will end it here, and then I'm gonna move forward to introducing to you my lab. So this is my team, and these are the people who are working with me. They are my in-house researchers. We are located in the Philippines. So we have here actually two barcodes or like QR codes. If you want to visit our website, you can scan this one. And then this one will bring you to our FB page. And then we have here our email and then the address or location of our lab. So we are open for collaborations. And if you have questions, I'm really open to answer them now. And I will be happy to address any questions that you have in mind. Thank you so much for listening. And thanks, Prof. Arta, for the invitation.
Thank you, Professor, for accepting our invitation, in fact. And of course, I would like to thank also your university president, Shirley, because we meet with her a couple of months. They came, in fact, to visit our university here from Philippines. There was a group of professors from your university, and they advised. And so thank you again for accepting our invitation and for such a wonderful presentation, because recently these technologies are new, and scientists, especially students that are working in PhD studies, are very interested in working such techniques, which are not so easy to work. So everybody knows that. So now I will just check from the chat part, is there any question? Here are 3 questions from the students and the academician that sent to us via email, because they are in the conference hall. So we just put here. The first one is that maybe with some of them you already mentioned, but I have to read because they would like to read the question. Professor Peter, with the rapid advancement of gene editing technologies such as CRISPR-Cas systems, how do you envision addressing the ethical considerations surrounding their use in medical application, particularly in balancing the potential benefits of targeted genetic interventions with concern regarding unintended off-target effects and long-term implications of human health and social values? Maybe from the chat part, you can see also the question there. You can check from there in the chat part.
Yeah, I think I got the question, Prof. Arta. I think the question was about, like, one is about the ethical concern, and then the second point of the question is the fact that how do we envision it in the future with certain concerns still on going back on the ethics. So I think I would say that when it comes to ethics, that's something that we cannot remove. Like, we have social issues also here in the Philippines, and even in the US when I was there, we have actually transgenic organisms that we wanted to bring into the market. But the problem is the fact that some people are still conservative about it, and we also have to understand that. And when we navigate, we try to navigate around the ethical considerations, especially for human applications. We actually don't use it in embryos, and we still don't use it in humans, but we are actually limited to cell lines that we are experimenting in the lab. So we have human cell lines that we are trying to perform on some experiments. And then it's very important that, we don't move forward into, I know it's easy to do it, but, as scientists, we have our responsibility to be limited onto experiments on human cell lines or cell cultures. And when it comes to, like, the people that don't like it, it would be best to kind of like try to educate them if there are certain groups. So as people who are working in science, what I'm trying to do is I try to talk around people all over me, and then I try to explain to them the importance of what we are doing and trying to convince them that what we are doing is actually for the advancement of science, and not only for the advancement of science, but it's also going to help in the health problems that we are encountering. Because I always explain it to them in a way that I'm born with a genetic disorder. And if I'm born with a genetic disorder, that's not actually my choice. And if I have an opportunity to change it and I can live a longer life and a more normal life, why are you not going to give me that opportunity if we have the technology? But then again, it's very important to have a precaution. Like whenever you do experiments, you have to look for the off targets. And that's something that we have to consider in medical applications. Sometimes we do experiments in human cell lines and we don't know, like if it's not in an actual system, we actually don't know what's happening. We can see what's happening in the cell, but if it's a whole organism, it's really hard to predict. We cannot actually tell what's going to happen. So I think we are just still at the beginning. It's still a very young technology.
Exactly.
I hope I have addressed that question, but yeah.
Yeah, as you mentioned, that just 5% of the sum changes when you do the CRISPR technology. So we have to get rid of these side effects, then we can continue, as these are the first step in the technology. And the next one, given the complexity of multifactorial diseases and the challenges associated with achieving precise and efficient genome modifications in vivo, what strategies or innovations are on the horizon for enhancing the specificity, delivery, and safety of gene editing therapeutics? And how do you anticipate overcoming immune responses and regulatory hurdles to facilitate their translation into clinical practice?
Okay, so when we talk about its translation in the clinical practice, I think we are still very far from that, to be honest. So we are still at the beginning and we are still trying to look into the safety of the technique. And then the second question is all about the specificity and how are we addressing all the off targets and every unexpected edits that we are seeing. So I've mentioned a while ago that we already have techniques that actually provides a platform that allows you to predict the change in the bases that you can have. So if you try to imagine how your purines and your pyrimidines look like I mean your adenine and your guanine, and then your thymine and your cytosine. So your thymine and your cytosine and your adenine and guanine kind of like looks the same. And then as if you try to look at them, like at the top part, you all in one of them you will have your oxygen, and in the other one you will actually have your amino group. And by using certain techniques, like say for example, you have your deaminase. So you can actually change that and replace it with oxygen. So therefore, you can change one base to another. So we already have these techniques to use base editors. And who knows, like maybe from your group, you will be able to figure out, oh, I have this enzyme. And with this enzyme that I have, I can actually change a moiety in my nucleotide. And then now I can have a new base. So those are the advances that I can actually tell about the group. And then there has been an effort in some groups in the US. They're actually trying to increase the efficiency of targeted insertion. So they use Cas9 to do the cut. And then after doing the cut, what they actually do is they try to kind of like insert a certain sequence. But then again, I've told you a while ago, when you do a cut, you immediately get a repair. And if you look at the Cas9, it's actually, it's two types of proteins. It's two domains. It's HNH and RUVC. So my hypothesis before was like, since they are two different domains, and so I was thinking like the cut that's happening at the top and at the bottom strand, like the top strand and the lower strand, is actually probably not happening in the same efficiency. So what happens is that when you cut the top strand, and then after cutting the top strand, if it's more efficient, then the repair takes place. And then you cut the bottom strand, and then the repairs takes place, and there's not going to be a drop. So I was actually telling my advisor before, what if I engineer a Cas9 that's going to have the same domain from the top and the bottom, so therefore the efficiency of the cut will be the same, and then I'm going to have a drop. But she was telling me that's too ambitious, and you don't want to do that for your PhD because it's going to take you forever. But, you know, it's a challenge. If somebody's interested to do it, I think you can give it a shot.
Maybe one of your students may be interested, or in the coming years.
Yeah, I think we are not ready for that. I just came back to the Philippines like 6 months ago, and then I just started my lab and just putting all things together, getting all my plasmids. I have a library of plasmids now, my library of endonucleases. I just started the platform, and we are still trying to perfect it. So I think as the years go by, then we'll try to move forward with that.
Yes, thank you, Professor. And the last one, as gene editing technologies continue to evolve, what collaborative efforts are essential for fostering interdisciplinary research and accelerating the development of transformative medical applications that harness the power of genome engineering?
Yeah, I think that's a very good point, and collaboration is very important. So as a person from a different background from the clinicians, I think it's very important that you have the people from the basic sciences actually being connected with the people who are working in the hospital, because the people working in the hospital can figure out actually what are the problems that we have to address and what are the things that we have to figure out. So that's why I've been very proactive in connecting with them and then figuring out the certain diseases that we have to focus on. So here in the Philippines, in the hospital I'm collaborating with, we actually do also newborn screening. It's a requirement every time you give birth. And in the panels that we have in the hospital right now, we actually try to detect like 28 diseases. And there are certain genetic disorders that they say it's kind of like simply inherited, it's monogenic. And they're telling me that I could start with that and we could figure out what we have to do. Now, the concern is how do you translate it to medical use? So I think it's a long way to go, to be honest, Prof. Arta. We are just working on cells right now and then translating it into a medical use. It's a collaborative effort. It's a work between the people who are doing the policies in science, the policies in research, that they are very important, the people in the hospital and the people in basic sciences. So it's kind of like a triangulation. So everyone should work hand in hand. Because, with all the policies that we have, they are very limiting. So they tell us you cannot do this. So I know you also have this in Turkey. We have our research ethics committees. And whenever you want to perform something, they kind of like inhibit the level of research that you can do because they say, oh, this is not ethical because like as per guidelines, you cannot do this. So I think the people who are working in the medical field, the basic sciences, and the people who are developing the policies that we have in our own countries and even in the international community should work hand in hand so that we can move forward and then actually bring these technologies into practice and use them in medical uses.
Thank you very much, Professor. And I think the last answer of yours was related with the meaning of our symposium, as it is the handshaking between the basic and clinical sciences, and already you included the policymakers here. So I think that you already described that, and thank you for that explanation. And of course, I think there is no any question now because there are a lot about. Enough. Now we'll continue with another speaker, and thank you again for sharing your valuable time with us, Professor.
Well, thank you also for listening, and I hope I'll get to see you all in person.
Yes, here in campus. Thank you, Professor.
Thank you.
Welcome, Rebecca. Now we are going to continue with our next speaker, invited speaker, that accepted again our invitation. And one of the link between us with Rebecca was Professor April Pike and Sonia from Memorial University that we meet with them here in our campus. So Rebecca today is going to talk about the readiness for genomic-informed cancer nursing in Newfoundland and Labrador. She already completed her bachelor degree from Medical Surgery of Nursing, School of Nursing at the Memorial University of Newfoundland, Canada, and a master degree, and she already continues her PhD studies at the same university. Her field of research is nursing and genomics. Maybe everybody think about that how nursing genomics is in combination, so in collaboration. So maybe Rebecca today is going to share with us her data that already they have, and talk about nursing and genomic studies that they are doing. And besides that, she's interested in complex method, nursing and genomic, cancer genomics, person-centered care, and patient-oriented research. Rebecca is doing her PhD. She is one of our young researchers here, invited speaker. So I would like to thank her for accepting our invitation and welcome again to our symposium.
Thank you very much, Professor Arda. I'll just start sharing my screen.
Yes, I'm stopping my sharing. Then now you can share your own presentation.
Okay. And we will go from the beginning.
Yes.
Okay. Is everybody seeing that?
Yes, it's okay. It's
perfect. Well, thank you very much for your time. I'm very pleased to be invited. Obviously, it's very early in my career to this symposium of very esteemed people and to get to hear some of the very interesting bench work that's being done. And I think that was a really good segue from Professor Peter into that need for collaboration and I guess sort of a synergy figure 8 between the clinical and the bench scientists. So today I'm going to be talking about readiness for genomics-informed cancer nursing in Newfoundland and Labrador. I'm from the country of Canada, and Newfoundland and Labrador is a province within the country of Canada of about 540,000 people, so fairly small in comparison. And I'm a PhD candidate, so the work I'm going to talk to you about today is my doctoral research, not published, but collected, and I'm really looking forward to sharing that with you all here today. So as I mentioned, I'm from Newfoundland and Labrador, and if you look at this image on the map, so that's the country of Canada, and Newfoundland and Labrador is the province in red. And as you can see, it's sort of like geographically isolated. It is an island, known for its rugged coastlines and icebergs, things like that. But I point out this picture to show you that you can see that it is an isolated sort of area. And it has been known for almost over 50 years that Newfoundland and Labrador has sort of been considered this genetic, they call it a gold mine for the investigation of genetic linked diseases. This has been going back years and years because of that, what people may have formerly referred to as a founder population. There's this mosaic sort of genetic isolate population where most of the descendants had this sort of Irish and British common ancestors. So because of that, there's been many publications, including this most recent one that was published in April 2023, just confirming that there is resources here that we could use to better understand genetic link to conditions and hopefully improve outcomes for patients. Now, as Professor Arda mentioned, I do use mixed methods. So I use qualitative interviewing as well as patient-oriented research. So what that means is in my study team, when I'm designing my studies, designing my questions, I involve people with lived experience of these conditions to inform the types of questions we're asking. And I always start with something that was told to me by one of my patient partners, Vanessa, who I work with very closely. She said, Rebecca, we've been studying diseases and genetic conditions for over 50 years in this province, but yet somehow it's no easier once somebody has that genetic test result to navigate what that means for themselves in the healthcare system. And a lot of times people feel like they're left adrift. So my interest is all the incredible work that is being done in bench science. How do we ensure the continuity of care in that so that this actually has a benefit to human health and people's lives? So with that as well, in my province of Newfoundland and Labrador, we actually have, in comparison to the rest of the country, very high cancer rates, the second highest cancer mortality rates, and high instances of colorectal and breast cancer. And we know that there are a lot of monogenic single gene cancer predisposition syndromes that are linked to colorectal and breast cancer. So, and I'm certainly not attributing, our genetic architecture as the sole cause of these cancer etiologies. We know it's a very complex gene environment interaction, but we do need to have all the factors in front of us when we're thinking about how do we actually improve health, and as well as slightly increase incidence of ovarian cancer. So with that being said, we think about what are opportunities and prospects in Newfoundland and Labrador. So this first image that I'm showing you, so our national health funder in Canada is the Canadian Institute of Health Research. And within that, you know, they have their different sort of, I guess, you know, different divisions, and one is the Institute of Genetics. So in 2022, the Institute of Genetics put out their, I guess, strategic plan called Sequencing Our Future. And this were their bold predictions for what genomics could look like in Canada within the next 10 years. And you'll see, you know, they predict things like pharmacogenomics as standard of care, genomes as part of healthcare records, and having the access to them on our smartphones. I mean, arguably, are we, you know, is that a bit of a pipe dream? Are we far away from there? But we do know that it's on the radar and it's gaining momentum. And so certainly that's something that I'm trying to leverage and get a part of, and certainly get my discipline more involved in that conversation. Because it does have the power to be transformative. But again, it has to move through all sort of the sectors. Specifically in my province, it's very much in its infancy. It's not started up yet, but for the past couple of years, they've been working to build capacity for the Provincial Clinical Translational Genomics Program. So that will be an in-home lab. It is I guess it's a collaboration between the institution, the university, as well as the health authority. So the idea that it has the capacity to do whole exome sequencing as well as multi-gene paneling, which right now, a lot of the patients who go through provincial medical genetics, that testing is outsourced to commercial labs, most oftentimes in the United States. So the hope is that this Provincial Clinical Translational Genomics Program will also be able to work very closely with our cancer care program so that we can do some of the in-house testing. And we know that not just in Canada, not just in Newfoundland, but of course in cancer care in general, we are seeing trends towards mainstreaming. So instead of waiting, and actually in my province, there was a news excerpt saying that in some cases, waiting to be seen for genetic testing and genetic counseling could be a wait list of up to 7 years. That is completely unacceptable. But when we think about mainstreaming, particularly in conditions like ovarian cancer, where now the oncologists and the oncology team are ordering these multi-gene panel testing, right? Because it has implications for their eligibility for things like PARP inhibitors. So when we think about that, there are opportunities for us to get involved. But again, this is still something, and the word genomics is still very much vernacular. At least I can comment to that for nurses who I'm working with. So there are opportunities, but a lot of work that needs to be done. So I always sort of explain to people why I really got interested in this, because I'm not a bench scientist and I am a nurse. I've been a nurse now who's been practicing for about 10 years. And I first started working on a gynecology unit. And a lot of these patients were gyne-oncology patients. And I never forgot that there was always you'd be talking to patients and I remember one of them was telling me that she was having a surgery because she carried a BRCA gene. And of course, you know, Angelina Jolie, we've sort of all heard of that. But I remember it was the patient who was educating me. I knew virtually nothing about it. And here I was, I had gone through my nursing school. And I thought, if I don't know anything about this, I'm certainly not the only one. But as I liaised with some of my interdisciplinary colleagues, I realized that, you know, who was doing a lot of work in finding I don't want to say founder mutation, but a mutation that was specific in our population in RAD51C that predisposed individuals to breast and ovarian cancer. And she had mentioned to me, she said, you know, Rebecca, there's no need for these girls to be getting cancer because we know how to prevent it. And that sort of, I was hooked by that. And that sort of led me in the trajectory of my career and I guess in career development. And what I focused on in my master's of nursing was the development of a policy proposal that would provide dedicated navigation for people who carry these inherited cancer predisposition syndromes, because we know they face lifelong considerations in risk management and such. So that was sort of the catalyst. But of course, things don't just start with a one and done with a one project, and it takes some time, a lot of, I guess, advocacy and continuing to collect data, do more research. And so that is sort of what led me in the direction of pursuing my PhD here at Memorial. So, and I'm also involved with a national initiative about how to sort of improve genomic literacy within the nursing profession in Canada writ large. But while we do know that while genetic testing is entering the clinical areas in oncology in particular, they describe genomics as sort of the paragon for the application of genetics and genomics. But there's very little empirical studies that actually assess oncology nurses' foundational genomic awareness. There was one done in Turkey by Dr. Memnon Senin, I believe, several years ago. There's been none conducted in our province, despite the fact that we do have this translational program up and coming, and there's only one that's been conducted in Canada. But furthermore, there's been very few interventional studies writ large, globally, examining the nurse-sensitive patient outcomes for genomic-informed care. There is, however, a large body of literature that's saying that people who carry these conditions, people who are accessing genetic-informed care, they experience gaps in their health care. But what hasn't really been explored is how they see that nurses can work with them to address some of these gaps. So for me, I see, issues are complex, but how do we synergize these two interrelated issues to really enact change so that these formidable discoveries that have been made in genetics and genomics can be applied to benefit patients in a way that is conducive with their lives. So for my overarching mixed methods question, and today I'm really going to concentrate on sort of those patient testimonies, because that's where my heart is with with this work, is what is the overall readiness for evidence-based cancer genomic care in Newfoundland and Labrador? So in that, I've conducted 37 interviews with people who have that lived experience. They told me tremendous about what their experiences have been like, and as well as how they foresee that nurses can be involved in their care. The second part, and while I have that data, still running some of the statistics on it, I administered a validated concept inventory to all the nurses working within cancer care in the province, and asking them questions about their knowledge, attitudes, and current practices with genetics and genomics. So from integrating these two results, it's my hope to develop recommendations for policy, for education, and hopefully future interventional work of how we can move this forward in my province. So first, high level, as I mentioned, I interviewed 37 people who had that lived experience. They had a genetic test and were found to have a molecularly confirmed hereditary cancer predisposition syndrome. Overall, and I'll get into this in a little bit more, but participants told me that there are huge unmet knowledge and supportive needs under our current system of care. They did endorse a high level of acceptability for nurse-led genomics-informed care and care pathways as potential solutions to these challenges. Now, in my survey, I had a response rate of 50 nurses. And while that seems fairly small, and it is, but considering the total population of oncology nurses in the total province is about 80, fairly good response rate. And what you'll see, and I'll speak to this a little bit later, is that many patients are already looking to nurses in these care pathways to answer genomic-related information. And while the oncology nurses had fairly positive attitudes about the potential benefits of genomics, they have clearly not yet received the educational supports to feel confident to integrate this knowledge into their practice. Like I said, I have all the data completed, and hopefully I'll be finishing this up in about December of 2024. So just to tell you a little bit about the sample of qualitative participants with that lived experience who I interviewed, 37, and they represented a broad range of lived experience with hereditary cancer syndromes, with time since receiving their diagnoses, and they care, certainly heterogeneity in the sample in terms of the different cancer predisposition syndromes, some with Lynch syndrome, which of course is hereditary non-polyposis colorectal cancer, CDH1, so predisposition to gastric cancers, in particular, highly penetrant monogenetic disorder, BRCA, which is actually less common in Newfoundland and Labrador, and as well as some other lesser known. So there was a lot of different experiences within the sample. And again, fairly even representation from both rural and urban regions of the province. And both with those who actually had a previous history of cancer and those who did not. So what we would sometimes refer to as provivors. So in how I've analyzed this qualitative data, I've separated into what I'm calling two major findings that had these sort of corresponding sub-findings. And I can't tell you how many times again and again that the participants who had accessed this information had told me that they perceived that this genomic knowledge, knowing their carrier status, was a form of power, in the sense of they were able to make decisions, but they indicated, very little, almost no decisional regret with having done so. And they felt that, not only did it make economical sense, but in terms of for the healthcare system, but also, of course, in terms of minimizing disease burden when we think about cancer along a continuum. And I think this is illustrated by this quote: "Because I'm screened, I may never get cancer," whereas the guy down the road who doesn't screen and doesn't know. And in this quote, this participant tells me she actually feels less at risk than per se, somebody who does not. And I think I always share this quote as well down here: "I feel that I've cheated cancer, or I'm trying to cheat cancer. I don't have to die from breast cancer. I got rid of it. So now I'm living with the scars and emptinesses in places where I had organs before, but I won't have that cancer." So again, while this is power, and I think this will come out more in my sub-themes, it's power, when we consider the whole person and how this changes their supportive needs, which I'll speak to in a minute. So the sub-themes really speak to, yes, knowledge is power, but there are inherent complexities, inhibiting factors that are required to take into consideration in order for that knowledge to truly become empowering. And this is the translational part, because things can seem all very well interesting when we have a lab result, but what does that really mean to the person who's receiving those results, and what it means for their life in a way that isn't burdensome? Very interesting answers from the younger previvors who I interviewed in this sample. Just said, there needs to be a process in the way of we take some people have a high degree of agency. They're very comfortable in seeking out information on their own. They want to be involved in advocating for themselves, but others require a lot more guidance and explicit direction. But however, I think this is illustrated by this quote: "I don't think you should just put me in a cancer management category just like somebody who happens to be younger and had this gene mutation, because there are so many differences between me and that person as well." So as we integrate these recommendations, we have to be willing to work with the personalization in care as well, and have means where we can sort of tolerate that. And again, even within families, we have to understand that, like this person says, "My mom's experience and perspective of it is very different. So my mom is very helpful and she's been through a lot and is very supportive, but my decisions and her are going to vary." And when I interviewed the same woman, she told me that it was one of the first things that she was told when she went to go see a specialist. The first question was, when was she having a mastectomy? And she said, "I didn't think that was a very good thing to ask somebody. So I would hope that whoever you're speaking to are well informed on the variety of different choices and options that somebody may take, and are respectful of that." Sub-finding 2 also speaks to knowledge as power when shared with others. And I think this is very important for our clinicians to understand, is that a lot of times, particularly in the people who had cancer, even if this genetic testing might not have made a tremendous difference to them and their outcomes, their motivations for seeking this were for their loved ones, for participating in trials. And I think that, sometimes that can even be more of a driver than our own personal health decisions. And as you can see, as a mom of a child, of a young adult who is positive, well, I tell you, that's really close to my heart, to think that I don't have to worry about passing on this mutation. And of course, we're getting into, reproductive endocrinology and reproductive technologies and things like this. But it is very important to appreciate, all that comes when we understand why people are seeking this care. And again, knowing that this person who receives this care may actually be life-saving for their relatives. So this particular person was found to carry an APC gene, which is predisposes them to familial adenopolyposis, very highly penetrant. She said if her son hadn't got the genetic testing, it's scary to think about what might have happened, because he was 16 years old, having known because of his mother's status, and he was that full that fast. So we know that this has implications, and this informs why people are seeking this knowledge. And with new knowledge comes new supportive needs. And the first quote I shared on the, I'm living with scars in places where I don't have organs. This is sort of really relates to this as well. But this is the thing, when you give somebody that genetic test result, it doesn't just end there. And if you look at this quote up here, I feel like there are a lot of us out there who have these proactive surgeries that have this upside down world after that nobody really understands. And it's so hard to navigate. You don't fit in a box a lot of times. So, we assume that people, understand the information, but as I think if you can tell, especially these quotes on my left-hand side, there's a lot of psychosocial support that is needed as this is unveiled to ensure that, as we give this people, we are not providing burdens on them as well. So we need to think about how the healthcare system is prepared to absorb and support people. And I think, I remember the first time I heard this last quote, maybe this is just how it is, but I guess I thought there would be more support with the big life change I had to make. This is a woman who was, found to have a gene that one of the CDH1 gene mutations, and that requires removal of your stomach. And as you can imagine, that's the recommendation, that's a huge adjustment to your life. So thinking about, all these people who are really just left adrift to sort of figure out these very complex things for themselves. They were generally very happy in terms of the information they have from medical genetics when they received their results. But once you left that purview and that safety, the rest of the system just wasn't prepared to absorb it. And as we know, healthcare is not, a one and done transaction cross section. So this is the kind of things we need to be mindful of. And I guess speaking to this again, challenges seeking knowledge. Now, sometimes some participants, if they had a family doctor who was very willing to take this on, but for the most part, people again told me, once you had left the purview of you had your test result, you were set adrift. There was this one person who had been involved as a child in a study to find an APC gene, went on about her life, I guess, for many years, had gone through many different doctors. And so when she was going through a pregnancy, they're doing a health assessment. She said, okay, I have FAP, but I don't really know anything about it. The doctor says, well, when was your last scope? And she was the first doctor that ever had any idea of what it was. She said, if you have this, you should be getting colonoscopies regular. And I was like, oh, I didn't know. Meanwhile, what's even more interesting, I think, or startling, I guess, about this story is that since now that she's having these colonoscopies, on several occasions has been found to have a stage 0 cancer. So you just think about how easy it was for this woman to fall through the cracks, really because of silos and knowledge deficits. And now I don't know why that, okay. Or just extreme frustration. It's ridiculous how big of a battle is. It's terrible. And I think what's frustrating is that you've got medical professionals that should know about this. And when you mention it to them, oh, I've never heard of that. So my second big theme is the idea of expanding care through a genomic lens in cancer nursing as sort of one, and I don't purport that this is the only strategy, but it is certainly one that, as I spoke to these participants, they were enthusiastic about. And that is expanding care through a genomic lens in cancer nursing. If we expand to think about cancer care as a continuum. And, a lot of times we ask them about their ideas of a nurse navigator, but I think, as you see here, one person is going to be huge as in a start. But realistically, to me, to have them in the cancer centers would make the most sense because we're looking at pre-existing cancer syndromes. Patients need screening and follow-up for the rest of their lives. What better position for these nurses to be than located in the cancer centers? And the participants I spoke to who had a personal history of cancer, they spoke that the experiences they had accessing oncology nurses, cancer care nurse navigators within the cancer care continuum, they saw that the role that they provided as being very useful and applicable to the care that they didn't receive as it pertained to their genetic health. So considering cancer as a genetic disease, how do we integrate that in and use our oncology nursing workforce, our capacity to improve the quality of care? This quote down here as well, because I did disclose to these participants that I was a nurse. And she said to me, anybody you can reach out to would be wonderful. Do you know, even talking to you now about the things I've gone through and what I've done about them, it helps me because I'm prancing around with this genetic disorder, this gene disorder, and it's not like you can talk to everybody about it. Time and time again, I asked people, had they had any interactions with nurses as it pertained to their genetic and genomic health? And for the most part, no. Nurses, no, not so much. It's always been doctors. When you consider 40% of the entire healthcare workforce, 44% is nurses, that, you know, that's a huge missed gap in continuity of care. And we know we need continuity of care for care to be personalized and impactful. However, again, people had mentioned that sometimes the nurse navigators were involved in liaising with genetics and genomics. So how do we ensure that nurses have just that basic genomic literacy to be able to engage and to ensure continuity of care within cancer care? Because we do know that, you know, there are predictions. I work closely with the City of Hope in California. I've trained through there. You know, they do talk about perhaps in the future, whole genome sequencing may be standard of care in all cancer patients. And certainly we see that in clinical trials and stuff. So it's something that we definitely have to upskill and integrate. The second sub-theme relates to disciplinary contribution. So anybody who has had, I guess, to engage with the nursing theoretical literature, there's a lot of talk about holism and advocacy, and that sort of seems to be how the discipline sort of positions itself. And when you juxtapose that on the things that people were telling me, I think that anybody who can kind of point the way or clarify or humanize the whole experience would be such a help. Yes, I have a family doctor who is supportive and listens, but I definitely think that there are like gaps in the care. And of course, Lynch syndrome, you know, you have that multi-extracolonic involvement. You have a risk for a urinary, ovarian, endometrial, colorectal cancer. This person says, you know, there can be this sort of segmentation of the body instead of looking at the patient as a whole. So how do we have people who can bring sort of this whole together in our care? And while, you know, there was a consensus that this is something that is the responsibility of every oncology nurse, the participants did tell me that they did foresee that this needed to be a specialized, dedicated, advanced practice navigation role. You know, and there was a couple of reasons. You know, this person said, you know, if it became a specialized thing, I would definitely want that person to have some underlying knowledge that was specific to genetics, obviously. And I know that nurses are trained in all aspects, but I would definitely want them to be able to answer the more complex questions. So this is when, you know, you get a result. Is this mosaicism? Is this a variant of uncertain significance? What about the phenotype? What about the genotype? You point into basic, but there needs to be a clinical resource person as well. But not only for the level of knowledge and expertise, but also for the relationship building. And you'll see this evidenced in this quote on my right. It can be information overload. So when you receive this diagnosis, it would be really helpful to say, this is the person you're going to build your relationship with and who's going to follow you with ongoing. So, you know, hopefully one of these recommendations that comes out from this study is also a dedicated advanced practice role. And as I mentioned, I've been involved in training with the City of Hope in California. They do a interdisciplinary course in cancer genomic risk assessment. And these are the types of roles, and there are a lot of, within that community of practice, particularly in the United States, a lot of nurses working in an advanced nursing practice capacity, providing cancer genomic risk assessment and follow-up to these people found to harbor these conditions. So that was a lot about my qualitative data. So I'll just speak very quickly to the quantitative data. And I'm really not going to go into tremendous detail about this here right now, but just to show you that this sort of, you know, shows that we need to have a shift in our thinking. The majority of the people who I interviewed, who answered the survey, and I went around, I did sort of information sessions with the nurses in the oncology care pathways. Majority of them did work in outpatient cancer care clinics, and some were within our inpatient oncology units. Interestingly, the scores in the inpatient units were actually slightly higher. I don't really have a theoretical rationale for why that is, but yeah, so that was just sort of an interesting finding. And I used the genomic nursing concept inventory and select items from the genetics and genomics nursing practice survey, which are validated tools developed by scholars in the United States. So I'll just share sort of 3 key findings. So one of the questions I asked the participants was, in the last 3 months, has any patient initiated a discussion with you about genetics, so to the nurses? 46% said yes. So I find that fairly interesting, but concerning when you juxtapose that with, I asked, how confident are you? Are you confident or not confident at all to access reliable and current information about genetics and cancer within working in your, working within your institution? 64% say they were not at all confident. This isn't surprising, because most had reported that they hadn't had any foundational or continuing education since licensure, and many reported that it wasn't part of their foundational education in their nursing undergraduate training program. The GNCI, which is the thing that I use to measure genomic knowledge, like I said, it's a concept inventory, and the score is computed out of 31 questions as a continuous score. This is a specific one item from that instrument, but this is a fairly basic question about genome inheritance and genome basics, and only 14%, and this is a question about BRCA, so it talks about sort of the central dogma of DNA, only 14% of the nurses had selected the correct response. So we know that there's a lot of work that we have to do to align what patients are calling for and their needs, and to ensure that our oncology nursing workforce is prepared to do so. But I think that when you bring that patient perspective, it really solidifies the need and hopefully will drive the needle on translational change. So with a mixed method study, you have to work on the integration, so that is what comes next. But I think we do have patient-driven data to inform novel nursing models of care within the province, particularly as we do work with the Center for Translational Genomics to bring this into our existing cancer care program. And there are opportunities for both the specialized oncology workforce and the generalist nurses. And I certainly hope, in building a program of research to be able to pilot such outcome studies. Because it's a concept inventory that I use to assess knowledge, I hope to continue to build relationships, work with their clinical nurse educator to provide that education to the oncology nurses, as we see this integrated into oncology care. And one of my committee members, Dr. Angela Hyde, she's a medical oncologist, but also the oncology lead for the Center for Translational Genomics. So it's that interdisciplinary working together to ensure that, we get our nurses and our oncologists and the pharmacy team all on the same page. So, definitely, while I've spoken about nurses, I do realize that this is the work that will require many interdisciplinary professionals working together if this is to be truly harmonized. And as I mentioned, when I was working with the oncology nurses and going, trying to drum up support for my survey, I guess, in recruitment, they gave me a lot of very pragmatic ideas about where we can start. So it isn't such a daunting vernacular. They said we are doing very comprehensive intake patient assessments when we get a new oncology patient. But even if we had something within our electronic medical records, we talk about the 3-2-1 rule, like 3 affected family members over 2 successive generations, 1 diagnosis over 50. Little questions like that could prompt, that aren't so perhaps daunting, but may again ensure access. But of course, this will be an ongoing work. We know that genomics is the epitome of a complex competency. It has a language that most don't understand. And I think that was probably evident by some of these wonderful presentations that went before me. But we do need to have people who are willing to sort of break down that vernacular, because it is having shared language and shared understandings that are those sort of basic political tools that we need to change the world, so that all these investments that we make in these scientific advancements are pragmatic. They benefit everybody. So I've covered a lot, and it's high level. And like I said, I'm very much a learner and a student, but I certainly appreciate the opportunity to speak with all of you today, and I appreciate your time and attention.
Thank you very much, dear Rebecca, for your presentation and for your patience, and for all of our speakers that were very patient today and very understandable. So now I will check for the questions on the chat. You can see from the chat part If you go a little bit up, you can see that. And I'm reading the first one. In preparing for genomic-informed cancer nursing in Newfoundland and Labrador, how do you propose addressing the educational needs and training requirements of nurses to ensure that they possess the necessary competencies in genomic literacy, counseling skills, and interdisciplinary collaboration to effectively support patients and families navigating the complexities of genomic information and personalized cancer care?
Sorry, I think I've been having a little bit difficulties with my internet.
Sorry,
do you mind repeating the question?
Go to the chat part. Do you see the chat part? Where we write some question and the academician student have written. You go up there.
I think I understand.
Yes, it's related to addressing the educational needs and training requirements of
the
nurses.
Education. Yes, I'm sorry. Sorry, I'm just having some connectivity issues, so I think I'm having some. But yeah, so I think that it's a complex question that won't just be it won't just be the responsibility of universities. We do have to have the foundational awareness, but I think there is a really big need, to develop a advanced practice nursing workforce, doing interdisciplinary learning at the clinical education level. I think that perhaps we're not, I can speak to my province here, perhaps we're not great at doing interdisciplinary learning sometimes. You know, we're not great at breaking out of our silos. But I think if we get each other all on the same page, and I think that there's a recognition that the methodology of co-design is certainly a lot more appealing in terms of when you are designing your studies, involve people at the point of care who know I might come in with a great idea for an intervention, but maybe that doesn't work with that practice environment. So I thought that was really good practical information that was given to me by some of the nurses. I think that you do have to start with practice-based leadership and proof of concept, because a lot of times, like in the States, there's been a lot of studies looking at, you know, nurses' genomic literacy, but very little in terms of proof of concept, because I think it's still sort of talked about in a very way that isn't linked to outcomes. So I think if we can work towards, you know, that's why I've certainly focused in oncology rather than broader in nursing writ large, that we'll get more buy-in in terms of people seeing that this can actually benefit patients, which is why patients are a big sort of part of my work.
Thank you. And the last question, as we do not have so much time now, given the unique geographic and socio-cultural context of Newfoundland and Labrador, how do you anticipate overcoming the challenges of access to genomic testing services, genetic counseling resources, and specialized oncology care in rural and remote communities? And what strategies are being implemented to promote inclusivity and the delivery of genomic-informed cancer nursing services in across diverse population within the province?
Okay, so I think that, and like I mentioned, there has been reporting that up to 7-year wait, you know, obviously they're triaged, but I think that certainly, you know, talking with Dr. Hyde, who's the medical oncologist, there is hope that for some of the less complex things, if the medical oncologist can be ordering some of this genomic testing, and, you know, if there are more complex questions, utilizing our workforce of genomic counselors, but then some of the other questions, you know, some that maybe either an advanced practice nurse or a nurse within oncology would feel comfortable answering. But we also know that there needs to be considerations in terms of like a lot of, another question I asked the nurses was, what would be your concerns about integrating genetics and genomics? And they reported that, you know, they wouldn't want to increase patient anxiety and risk. So I think that there needs to be very clear direction about what we can tell patients in terms of the legislature and the protective. We do have pretty robust legislature and the Genetic Non-Discrimination Act, but I think that people want to know more about that as well. So I do think that mainstreaming is going to be sort of more it will be the way of the future. It has to be, because we know that there are deficits in the genetic counseling workforce. Like, that's been well documented in Canada and elsewhere. So, you know, my work is sort of futuristic in one sense, but we are on that cusp.
Thank you. And I remember from Professor April Pike, because when you are working with genomic data, you have very strict and health, in fact, policies, as I know, because in order when you share or ethical issues about your genomic data. So I know even this question is related with a rural, maybe a province that is a little bit geographical or socio-cultural, in fact, region. But I know that the genomic data especially is protected with very strict policy decisions.
Yeah.
And thank you again, Rebecca, for your presentation and valuable time. Now we will continue with our next speaker, our keynote speaker, that the last one, and from America. So I would like to open background information of our professor, distinguished professor.
Welcome.
Just a little bit. I'm sharing. Hello.
You're looking wonderful.
Thank you.
Yes. Before starting, I would like to welcome Professor Bahar Uslu. She's a very distinguished and esteemed scientist and medical doctor, and she is our honor guest today, keynote speaker. And before giving the stage to our professor, I would like to give a brief information. Of course, we cannot talk everything here because there is a lot that we can talk for Professor Bahar, but I just will give brief information. Before starting, I just, as everybody see here, the presentation will be related with when does life begin for lab-made kids? So title is this one. Professor Bahar Uslu graduated from medical degree program from Faculty of Medicine in Marmara University, and she has a PhD from the same university and continue her research doctorate studies in the School of Medicine at Yale University, United States. Professor's academic career is a lot, of course, but we just included some of them here. She worked as clinical senior embryologist, medical research scientist lecturer in Yale University, as a professor, again at Frank Natter School of Medicine, where she continues to work there. She is the chair of International Congress of Future Medical Pioneers. She is the professor, senior clinical and developmental embryologist, histopathologist in 2023 in the Infertility and Art Clinic Center. As a professor, again, at the Connected United States Visionary Professor of Medicine. And of course, beside academic career, professor has some major managerial experience as a founder of andrology laboratories from Ministry of Health in Turkey, founding presidents for the Society of Histology and Embryology since 2013. The main research interest of Professor Bahar is medicine, as you understand, as she is a medical doctor and a scientist, clinical industry, medical device designer, clinical and developmental embryology, IVF techniques, regenerative medicine, tissue engineering, and embryonic stem cell. Of course, these is just numbers, but professor has a lot of research activity. And beside research activity, she is also honored as the Academic of the Year and the 30 most successful woman abroad, talented by the American government. And of course, beside that one, she has a lot of honors that you can see here. Academician of the Year as the #1 among reproductive medicine specialists in Turkey, and another award and honors from other projects in Turkey and abroad. And of course, I would like to mention that Professor Uslu is the inventor of histomorphological ovarian follicle counting method and the owner of a medical innovation and design company. And of course, if you are interested in the research of professor, you can check from the news because she's in fact also a famous scientist and medical doctor that you can see here. So I would like to thank for accepting our invitation and be with us today, sharing her valuable time, and welcome again to our symposium. Now I will stop my sharing and I will give the stage to Professor Uslu. Welcome, Professor.
Oh, thank you so much. It is so embarrassing situation, too much detail. Your presentation is so hard for me. I mean, let me start sharing my screen, please, if you let me do first.
I think that presenter view.
Yes, I'm trying from the start. Is there any technical problem?
No.
Everything is okay.
Good, thank you so much.
Oh.
So, thank you for the invitation. I mean, you share all detail of me about, I'm so impressed about your university too, because the first, I saw some Anatolian center you made it the title of your center. This is so impressive. And the other one, you're talking about the handshaking, the clinical and basic science foundation of the medicine. This is so impressive. Is two things about me. So my current role, I'm giving some visionary role for creating some new education for the medical student. Actually, we are talking about at least 2 decades ahead. What is their role? What kind of literature we have to create for the futuristic medicine, advanced medicine. So I'm not connected only one university. I have a different role in different universities. So I hope I can give some nice contribution for your symposium. Congratulations and thank you for the invitation, Professor Erguven and Professor Armani. So let's start. I mean, this is the hot topic actually for this week, because I'm receiving lots of invitation to giving some contribution their symposium and congress. And next week, I'm going to Nevada. They're all the biologists and medical doctors in the reproductive medicine. We are collecting in the Congress and Symposium, and I'm the keynote speaker. I will present a similar presentation, but you are the first. The AYDIN University, right? Earlier than the older world. So let's start. I mean, I'm interested in the life beginning, especially embryologic life. So what we are talking about, this presentation, firstly, human embryo. And this is the ethical presentation. And I'm interested in pre-implantation stage embryos and their environment in the embryology laboratory. I mean, in embryology laboratory conditions, I'm talking about only for this entirety. I'm not for example, from the public perspective, they are confusing. Hey, where is the embryo? This is the human right. Or I'm not talking about the human right. I'm talking about some time, about at least 14 days nowadays with high tech. Inside the laboratory, like we are managing and manipulating like a father and mother and their doctors inside the laboratory. This is the main point. So I have some suggestion for the futuristic medicine, but also I'm so fan of the traditional, conventional Anatolian medicine, because the traditional medicine rules coming from the Anatolia, from the Hippocrates and Avicenna, we are the girls and boys and brothers from the Hippocrates and Avicenna, of course. And so I have some suggestion about the hierarchy in the laboratory clinics. The main point, I'm trying to protect not only the embryo, also the institution, also the personnel inside the laboratory. So if we are talking about the advanced futuristic medicine, new era, we will talk about the artificial organ tissue banking in the future. So you mentioned before, Professor Armani and her colleagues mentioned genetic imprinting. And next generation embryo development. And we will talk about these things, these topics in the futuristic advanced medicine. So we have to create some ethical issues and new rules for DNA repair. Please, I'm trying to remind you, Professor Turkish Nobel Prize winner, he created some DNA repair in the oncologic side, but also we can use this technology for the future, for DNA repair, for the embryo development. So this objective, identification of futuristic advanced medicine mostly. So we have some concern. I'm living in US. I'm creating some medical education developments, but we have some concern. I mean, I'm looking for some deontology classes, ethical classes. There is nothing in the US medicine. So if we are talking about the conventional medicine versus US medicine seems some deficits. I mean, there's no any oath, for example. This is the ethical issues, very important. I mean, there are mixing, matching the roles. For example, who is the medical doctor, who is the associate degree, or who is the, I mean, I have another concern. Traditional medical doctor has equity with the osteopathic medicine. So everyone, if you are enthusiastic with short-term certification, you can do anything in US. If you are electrical engineer or different type of engineer, you can be the dean of the medical faculty. I mean, it is confusing from the European traditional perspective. So there are lots of different rules, some of them federal rules, some of them state rules. They have some complexity, some difficulty, and in terms of the American legislator. But I have some still keeping my hope, because I remember the Pasteur Institute about 40 years ago, when we were talking about the first time the IVF, Louisa Brown, the Pasteur Institute teachers and scientists, they said, hey, hold on, wait a minute. We have to create the rules and regulation first. This is my hope. I mean, I think so. The US medicine still neglected these ethical issues. So I received invitation from Texas last week. It was a huge conference, the main conference and symposium about IVF and reproductive medicine. They gave me a small briefing about how can I follow up my idea or bill. This is the small example from Georgia legislator. Georgia is the small town, the conservative town, conservative state actually. And if you have some idea, it doesn't matter if it's reproductive or doesn't matter the other things, you have to pass almost 40 steps. It doesn't matter, I can pass all these steps. But you know, who are these Senate compiling from the different sites? Their occupation just only businessmen and businesswomen. So, I mean, this is the first time I understand where I am. So this is the capitalist world. And so, I mean, I would like to discuss some different perspective with scientists or lawyers, not with the businesswoman. And so it was so disappointed time for me. That's why the main reason I have to contribute to US medicine, because all the world is following the US medicine. I received an invitation a couple of months ago from Yeditepe University rector, and he said, hey, Professor Bahar, please come to me and share what is the dream country US? Share your idea. And I tried to explain to the students, I mean, the title was, my presentation was, the US is really a dream country. So last year, the almost big bomb is coming to the IVF world. I mean, you remember the abortus legislation and from the Mississippi court, right? And everyone focused to women's rights. Of course, the women's rights very important, but there are not only one leg to women's rights. I mean, there are three different legs. What about the embryo perspective? What about the institution perspective? What just only all the public concentrated on the woman, right? Actually, this legislation, I'm of course support the woman right. But this changing, showing the US lawyers and US medicine still has some deficit because there's no any detail-oriented constitution. I mean, they are creating some daily solving the problems, daily solution. So it was the good opportunity to create some regulation for the reproductive world, but nobody taking care about what's going on. And then a couple of weeks ago, from the Alabama Supreme Court, published and shut down the state's largest infertility clinics because some accident happened. One embryologist lost the embryo. And they are, I mean, the American people, American insurance companies also looking for the opportunity to sue the institution, getting some, I mean, they are waiting for your fault. They are waiting for your mistake to sue you. I mean, after this incident, they sue the institution. And because there's no any regulation, there's no hierarchy inside the laboratory, there's no protecting. That's why the Alabama court shut down these infertility clinics. And then a second provider also stops IVF care after the court deems. But this is the good side of this court decision. The court, this is the first time in the world, a couple of weeks ago, it says, hey, hold on, you are talking about the embryos. It means it is equal as a children. So actually, it is not good because as a creator, I'm waiting for at least 2 decades to put in some regulations, some hierarchy things, some rules. I mean, we have to handshake with the law and medicine together. It should have done at least 4 decades ago, actually. That's why I'm receiving lots of invitation from the not only reproductive medicine, I'm receiving invitation as a keynote speaker from the pediatrician. Can you imagine? Because they are interested in the futuristic kids. I mean, they're talking about the embryo from the starting point to do future kids. So this is the new entity. I mean, I'm not talking about the outside the woman body. I'm talking about in the petri dish. This is the new entity. So please remember these things. I mean, all about the money. So just, I have to remember your traditional medicine. I mean, what is the role? What is the ethics? And what is the hierarchic condition? Because this is my duty because I'm Anatolian. And Anatolian medicine starting to the world. I mean, you cannot destroy the ethical rules. You cannot destroy the handshaking law and medicine. So that's why I'm criticizing current status of the reproductive laboratory medicine. So there is no any consensus who is the clinical embryologist, who has the full authority, and what is the hierarchy inside. These are protecting the institution, not only the embryo. I mean, last 40 decades, this from the Louisa Brown, it's a legal problem actually, because all the shifting nature to the medical practice, we are talking about the embryo in the petri dish. This is new entity. And also we will talk about the differentiation of parenthood. Now we are talking about the 4 different parents, 5 different parents. We are getting the mitochondria from the other woman, cytoplasm from the others. There are different mothers, but we have no law and we have no regulation right now. So this is the starting point. What is the explanation of the embryo? Can we say that this is the ordinary tissue sample? Of course not. Is it possible to compensate? For example, just remember the Alabama court, someone lost the embryo, they cannot compensate this lost embryo to give the parents. That's why, but we cannot say that, hey, this is the totally full, mature child of human, but it has some kind of potential, of course. There are lots of different terminology about the embryos. The Webster dictionary says that they are talking about the inside the human body. They are not, they have no any explanation in the embryo in the petri dish. But there are lots of different definition, but they are talking about the natural reproduction. So I have some nice explanation, but all they are from the traditional medicine, from the European medicine. Thank God we have Dutch, French, and Belgian medicine. They are still following the traditional. It's good example from the US medicine. A Dutch Embryo Act, a couple of decades ago, they said, hey, this embryo has human origin and it needs some respect from the intrinsic value. Also, French National Ethics Committee explained it has some respect and must command universal respect. So also Belgian Senate bill published and declared the Embryo Production Act. It has capacity to develop into the human being. So also we have nice universal institution. It's called UNESCO. And actually 4 decades ago, UNESCO declared what is the embryo. It is heritage of the humanity. So these are, we have to take care of this explanation, of course, in this world. We can say that it has potential of the human being, of course, because this is the simple question, when the life began. So because we are not talking about the sperm or oocyte, gametes, we are talking about the new entity. This is the 3rd egg, and it has its own capacity to regulate and coordinate itself. I mean, this is the main point. This is the main spot of the life begin. And some authors is saying that the fertilization, the beginning of the life, some because there are lots of idea, because they says this is the key idea advanced by the defendants. The fertilization is different genotype, but the same individual human being from the start. So additional, there are lots of different perspectives. They are saying that zygote approach, or so conception or chromosome unit, this is the new chromosome unit. That's why the life begin from this time. And so but most extreme approach from the status from the zygote appears. Of course, they are referred to as conception, but presently doesn't essentially mean natural fertilization. Also, there are lots of nice examples, because we talk about the third egg or embryo has its own capacity, because it's an intellectual property. It has the stage of expansion, and it's using its own energy also. There are lots of different view also. Zygote demonstrate polarity. They are changing. This is also intellectual decision, the directional orientation, the inclined to develop. So these are all the starting point of the life. And we need some moral contribution, or theological maybe contribution, and legal contribution to this work. We have still obstacle, of course. And I love this station from the Kevlas. He's a good ethical scientist. He said we have very bad experience in the history, Nazi policy and the racial hygiene. I mean, without any regulation, we cannot do anything. So what is the current personnel for the reproductive laboratory medicine? I mean, if you ask in some public opinion, they're saying, who is the clinical embryologist, or who is the test tube baby makers? They can say that gynecologist. Actually, it's not, because I have to remind you, 90% IVF make it inside the laboratory, and there is no permission to gynecologists to come inside. So I'm not saying that forbid their rights or block their rights or something. No, it's physically, it's impossible. One person different in being in the different condition, physically it is impossible. But inside the lab, we have some biologists, we have some certified staff, some technologists, some MDs, some microsurgeons maybe, and nurses. So, but do they have full authority to manipulate the embryology? This is the question. So we have still some confusion about the roles and who has the full authority in terms of the traditional medicine, of course, because we have some guardian roles to not only mimicking the microsurgery, because the ICSI is a microsurgery manipulation. So we have some still confusion. And also, today's clinical embryologist terminology doesn't fit the traditional medicine. Alpha Society published a couple of years ago, this article is asking the definition of who is the clinical embryologist, but they couldn't find the conclusion at the end, because there are lots of different categories from different sides of some allied health professionals, some biologists, some medical doctors. They couldn't find the main reason to put the nice explanation as a clinical embryologist. So who can be clinical embryologist? Of course, it needs some technical skills, experience, of course, extensive medical knowledge, but we need some kind of crucial decision. So, of course, the other side, in the traditional medicine, if we are saying clinical, we are talking about the medical capacity and responsibility to remind you Hippocrates oath. as a microsurgery we are doing. This type of manipulation is microsurgery. It is not, will it be surprise, choosing the laboratory director among medical doctors received special education and from the authorized institution, carrying clinical embryologist specialist diploma. So also we need some hierarchical roles. Of course, this is the teamwork. I'm talking about like a hierarchy, like a orchestra. I mean, nobody can do everything himself or herself. Of course, we need nurses, we need technologists, we need definitely biologists, but we need to create some regulation between us. There is no any limit or border. I mean, we have to move border like this symposium, the title. So from the perspective of the US medicine, they try to create some certification standards for ELD, embryology laboratory director, also HCLD, high clinical laboratory director. So, but they're talking about the doctorate degree. Doctorate degree is confusing to everyone because from the scientists, they should know this doctorate degree, it is academical degree, it is not clinical degree. That's why still they couldn't get nice solution and nice example to show the lawyers. But we have some nice examples from the France. 90% they are their laboratory director qualified clinicians. So also Turkish medicine, they have some embryology specialty program. I mean, I will represent this program because the US medicine has no any embryology specialty program. I will represent what kind of program we need. So it's a good example, Turkish medical faculties. I will present this one. And still, US has tried to show 5 different levels of the certification. And please remember, all the other medical laboratories, they have already directed by the physician with the specialist training, the specific areas, laboratory medicine. Also, Turkish governmental system has a good hierarchical model because they are still protecting the institution. So because all the insurance companies and parents, they are trying to find out your mistake to sue the institution. So luckily we have Dutch, France, Belgium, and Turkish program. And also this program, Medical Histology and Embryology, belongs to Medical Histology and Embryology Department, a faculty of the medicine. So I'm the founder of this association, Society of Histologists and Embryologists. You can check the main page of this society. And I published, this is the first article in Turkish, because I'm creating some nice, I think so it's unique article. It's about the embryo rights. And then a couple of years ago, I think so 2 years ago, I established a congress as a chair. I didn't invite only medical doctors. I invite also lawyers, some ethical scientists, and some, I think so, I invited some artificial intelligence professionals to this Congress. I mean, we need handshaking. We need to find out some regulation for the futuristic medicine. While inviting them, I asked them, please, I'm not interested in your current status and your current project. I'm interested in your project at least 2 decades ahead, because I'm creating some new things. I have to publish some new things for the futuristic medicine. And we published, this is the first article about the embryo terminology in Petri dish. And so I received invitation from the nice journal, and they chose this chapter as a perspective chapter. Of course, you know what is the meaning of perspective chapter. This is unique, and the first subject to publish to the medical world. And the chapter's title, what about the embryo rise to remembering the older words? And this is the main page of the society. So thank you so much for the Yale University, Quinnipiac University, Turkish Tibetan program, and AYDIN University inviting me for nice symposium.
Thank you, Professor, for this presentation and for important points that you already mentioned, and take consideration about that, because a lot of people do not know these topics very well. Even you mentioned about the pre-implantation genetic diagnosis. Even it's very, I think, very important, because genetic diseases, when of course they are observed, it's not easy to treat them, in fact, because still gene therapy, other therapies are in advance or in research, not in clinic. However, we can prevent some genetic diseases. So embryology and genetics together in pre-implantation genetic diagnosis, I think, is one of the important techniques that, for me, I think, and I don't know how you think about that. Of course, the other topics here, because I know that the reason that I mentioned that, but of course, of other topics, I'm not so familiar about them. But maybe we have Mine Ergüven, professor here. She's also a clinical embryologist. Maybe she would like to add something. And before that, I can ask the question that come from the academicians and from the students that are in the conference hall. Yes, Professor Mine, would you like to ask now, or maybe I can ask and then you can continue with Professor Bahar?
Okay, first of all, I think that we must give first rank to audience, and then yes, I miss also Professor Bahar.
Thank you so
much. And I know her since
you are my childhood friend.
Yes, you're right. Yeah, but we are still teenager, you know.
Yes.
We miss you so much.
Yeah, me too. Please come to share your ideas with our university. Always, I mean, I'm giving some invitation.
Oh, thank you. My pleasure, my honor. Thank you very much.
Of course.
Thank you. First of all, let's get the questions from audience, Dr. Armani, and then I will ask 2 questions to Professor Uslu. Okay?
Yep.
Okay. Maybe some of them are already answered, but again, I would like to ask them because they would like to be answered. Professor Bahar, in the context of lab-made kits, how do you propose navigating the ethical and philosophical debates surrounding the definition of life and the implication for reproductive technology, particularly in terms of determining the moral status and rights of embryo or gametes manipulated in vitro, and how can these considerations inform the development of responsible regulatory frameworks and clinical guidelines for assisted reproduction?
Yeah, of course. Actually, I tried to explain in the slides. I gave some small explanation. I mean, sometimes I'm giving some objection from the scientists, as a biologists or nurses or something. They are thinking, hey, Bahar, Uslu tried to stop us. No, of course not. This is teamwork. This is teamwork. I'm trying to protect them and protect the institution, protect not only the embryo. Just you see the Alabama, they shut down all the states biggest institution, the clinic, and everyone lost their job. And this is the result because we have no any regulation. We should protect ourselves, not only embryo. Of course, it's not the idea, oh, this is the kid. I can manipulate myself. Hold on. I mean, please give me permission, just only me. Of course, I cannot say that. But we have to think about, we have to start to brainstorming, but from the traditional medicine side. I mean, because everybody talking about today, admiring the US, not only medicine, also US fashion, also US science, also US constitution from the lawyer side. I mean, still think about it. We have huge knowledge and we have huge background. Also, we have huge experience. It's a traditional experience. Why we are destroying all of them just following the US? I mean, we have to remind them, hey, we have something. This is the role why I'm here. I mean, because this is my occupation. I'm interested in the embryo, my institution, my colleagues, and my patients too. We have to find out nicest solution, not only for me, not only for the embryo, or not only for the institution. I'm interested in everybody's happiness.
What about another question? Dr.
Thank you,
Arman?
Professor. In fact, all of the questions are related with the moral and ethics issue. So as we, another one is, as we grapple with the question of when life begins in the context of lab-made kids, how do you envision incorporating diverse perspectives from bioethicists, religious leaders, policymakers, and the broader public into the dialogue surrounding reproductive autonomy, parental rights, and moral responsibilities associated with creating and raising children conceived through assisted reproductive technologies?
Yeah, of course, I agree with everyone. I mean, it is not, I cannot say that, hey, only only the scientists or medical doctors can decide to all of the reproductive medicine. Of course, I created nice World Congress. I invited not only medical doctors, I invited artificial intelligence professionals, lawyers, some ethical geneticians, everyone. We have to make a consensus. Actually, we should have done, I mean, at least 4 decades ago. We have to create, like Pasteur Institute warned the reproductive medicine. Hey, hold on. We have to, because science making larger steps ahead compared to law. I mean, the lawyers, a little bit, they are trying to figure out the daily solution in US, at least here. We are lucky as in Turkish or European legislator. I mean, they have nice solution for everyone. For example, in Turkey, there is no any insurance company suing the government, because all the government IVF laboratories has in the top medical doctors and lots of technologists, all the hierarchy inside. I think so a couple of years ago, the private sectors in Turkey, still they were keeping besides the medical doctors, but I think so because of the lack of the population of the medical doctors, now I think so they established the roles, but we need more regulation to show as an example to the world. Of course, my concern is not only ethical, also from the scientist perspective, we have to create nicest solution for everyone. I'm inviting everyone to our Congress. Please share your ideas. Maybe I'm wrong. Hey, this is the totipotent stem cells only. Stop it. They can say that. It is possible. This is the lobby. All about the money. Right?
Thank you,
I'm
Professor.
asking myself.
Yeah. And the last one, what opportunities exist for interdisciplinary collaboration and public engagement to foster informed decision-making and promote ethical practices in the realm of
Question. Love it. Love it. Thank you for the potential. You made, guys, big job really creating this idea and handshaking between the old institutions. Please let's do the next one, inviting with lawyers. Why not? Some ethical professors, lawyers, some artificial intelligence, some engineering.
We will organize it with you. Okay?
Of course, I'm ready. I'm ready because we have to do it.
Wonderful.
It's our role. Of course, we need some nice. But you can follow our association. They are still trying to create some congress. Maybe I can get some sponsorship from the institution also for this congress. Maybe AYDIN University is rich university, right? They can contribute us. So of course, the next time we can do it.
Yes, we can. I think we can.
Yes, next time and already host you here, and maybe in the future, all of our speakers, we can host you face-to-face in our campus here. So it will be fantastic to share the ideas here.
Right? Of course.
Okay, thank you very much. And first of all, I would like to tell you before, please accept our apology because we extend the time and you come too late for your presentation. Please accept our apology. Thank you very much. You know the situation.
So nice to see you guys. Bye-bye. Congratulations.
Thank you very much.
And we appreciate that we host here in view of clinic and fundamental science. See you.
Thank you so much. So nice to see you. Bye-bye.
Bye-bye.
See you, Professor. Bye.
Bye-bye.